Hello, my dear friends. I am Dr. Arun Swami, your physio faculty. Hope all of you are doing very good, better. Please confirm me whether this audio and video all are good. And I hope so that you are preparing for this FMG exam. Still, there is no date yet confirmed, but soon that NBA will issue the date. So don't worry about the date and just focus on your preparation better. And I hope so that the exam date will be in the June as well. So just do your preparation according to this June exam. क्योंकि कुछ भी हो जाए, चाहे exam बेटा कब भी हो, अपने को तो preparation अभी से करनी है, right? So why to waste the time and why to worry about the exam date? You have to start it as soon as possible, and we have to pass this exam at any cost. So I hope so that my voice is clear and my video is clear. Hope all of you can see me, all of you can hear me properly. So let's start with the physiology. And just starting before this physiology, but I would like to tell one thing, that's whenever you are starting any subject or whenever you are studying any subject, first you should identify that what is the importance of that subject, isn't it? Because that every subject which you are studying, first you should realize that what is the importance, why you are studying that subject, and what are the meaning of that subject. Then only you can able to understand that what we are going to study in particular in that subject, isn't it? So physiology means nothing. Why physiology is so important? Because whether you are going into the clinical part, whether you are going into the paraclinical part, everywhere this physiology, anatomy and biochem, that is quite important subject. These are basic subjects, and these basic subjects are quite important for your exam purpose as well. Because in current scenarios, as you know, that exam is quite changed, and right now there is no direct question is asking. Every time the examiner is asking you all the questions from the integrated part, means it starts from the pharma, asking about the physio, and ending on the medicine or like surgery or anything else. So this is how you should understand, because right now you cannot just mug up the things and you cannot go directly into the exam that you will get a same line whether you have studied or not. Means it doesn't mean better. You have to study from the basic. Your concept should be proper. You should understand those things, because everything, whatever there is inside our body, everything is always with you. So you should realize, you should think about it, that whatever you are going to study, that is thing already going inside of your body. So just by of your imagination, you can think about it. And that is how that makes it easy to understand that subject. Physiology means normal functioning of a living organism. Normal functioning of a living organism, that is the physiology. Means, suppose there is a motor car, there is a car, that car which you drive on day to day. So what is the importance of the petrol in the car? What could be happen if there will be no petrol or there will be the duplicate petrol will be in your car? So first you should identify what is the normal role of the petrol in your car. And you very well know that without petrol, you cannot start your car. It cannot function. So this is how you should understand. So if you don't understand the normal functioning of the body, but how you can identify whether it is a disease or not disease, whether it is a pathology or something else. So first you should always identify the normal functioning of a living organism. And that makes very easy understand for that particular organ, for that particular about that disease, and all the signs and symptoms that could be occur from that particular disease. So this is how you should understand it. So physiology means normal functioning of a living organism, and that's we are going to study in next 4 days. And we'll study about that. What's the normal functioning, how our body basically functions, what are the things present inside of our body that's we can do our day-to-day activity basis of these things, and what could be happen if those things could be in imbalance condition. So first, this physiology word is given by a doctor, and the doctor name of that called as Claude Bernard. He's a French doctor, French scientist. So basis on this French doctor, the name of this physiology is given, and physio means in the Latin normal function of living organism. And basis on this, we call that doctor a father of physiology. So father of physiology is none other. This is Claude Bernard. Right? This is Claude Bernard. But please confirm me whether audio video all good. Okay. Somebody is asking me about this workbook PDF. Right now I cannot send it, but yeah, please bear me with the first break. In the first break, I will tell to the management, and definitely they will provide you in the better quality. Okay, beta. So father of physiology is Claude Bernard, and just understanding of these things as well. And understanding of these things as well, beta, I would like also tell you that how you can understand about this normal functioning of our body. First, you should think, beta, like our Earth. In our Earth, there is a 70% is water and 30% is land, or might be a 75 or 25, something like this. But remember one thing, that's everything, whatever present on this earth, it is originated from this earth and it is ending into the earth. So everything, whatever you are seeing in the normal nature, that the things, everything is made up of that, and our body is also made up from this nature. So whatever we are seeing outside on this world, the same thing present inside of our body. Like, for example, outside of the world means there is a 70% is water and 30% is land, right? The land where we are living. Similarly, inside of our body, there is a 60% water. How much water? 60% is water and 40% is your solid mass. And this solid mass is nothing. This solid mass is cell. So, like similar on this earth, means if my weight is 100 kg, 60% is water, it means there is a 60 liter is water. Can you believe? 60 liter pani apni body ke andar. Yes, and that's the thing. That is why always in day-to-day activity, you drink more and you eat less. That is why every day you drink more than 2 liters of water and you eat only around 1 kg of the food. Why? Because inside of your body composition is also similar. There is a 60% is water and 40% is solid mass. And this solid mass, first of all, in this first 2 chapters, in the cell physiology and in body fluids, we are going about to talk only about these 2 properties of the body. Number 1, what is the solid mass? And number 2, the fluid. And we'll see that what are the chambers, what are the ratio of the fluid in different chambers, and how we can estimate of this body fluid, right, brother? So just starting of this one part, that is the solid mass, and brother, inside of our body, the solid mass is starting from the cell. So inside of our body, this body is started from the cell, and that is the smallest and very structural and functional unit of our life. It means the one cell came from the father side, one cell came from the mother side, and they both fused. And now this cell is having a copy. Now this cell is having information of genetic material from your father and information of genetic material from your mother. And that is why, when you grow, you look like similar your mother or either father. And that is how, if there is a two human cell is mixing together, then the new cell which is formed, that is also the new person will form into looks like a human being. And similarly, if there is some animal, the things which will come out or the outcome will be from the cell, again the same animal. So this is how जानवर से जानवर बनता है, इंसान से इंसान बनता है. Why? because due to the information which is present inside of the cell, and basis of this information, but this information is called as DNA, genetic material. And this genetic material actually decides that's what will be your height, what will be your body buildup, what will be the color of your skin, what will be your appearance, and this is how all things is decided by your cell. So this is how you should understand the things. And if I already told you about this, that minimum and the smallest unit of the life is cell. And remember, this cell is having a property. The property which having inside of the cell is dividing property. Means this cell can be divided. And this is how from one cell we have started our life, and then multiple cells are there. And after that, what happens? When these cells started to function together into a group, like similarly, I am a doctor, you are a doctor, and somebody else is a doctor, right? So what is happening? That you are working, we all are working into a group for the society, and we all are collectively called as medicos. It means we all are medicos and we all are doctors, and combined we are working for the same function. And this is how inside the body, when there are numerous cells present inside the body, and some of the cells started functioning into the group. This group now it is called as tissue. So what happens? First of all, the cell is there was a cell. After that, tissue. After tissue, there is organ, then system, and then the organism. So these are nothings. And cell is the basic structural functional unit of the life. And when the group of cells started functioning together, this is called as tissue. Then when group of tissue is started functioning together, we call this as a organ. And when the group of organ is started functioning together, we call it as a system. And when all the system functions together, it makes the organism. So this is how our body is started, right? So let's start understanding this. So first of all, the cell, and the cell is the basic structural function unit of the life, and this is the smallest part of the body. And remember, beta, in this physiology or in this medical, whatever we are going to talk about, the target area will be cell. Means you are taking oxygen inside. Where this oxygen should go? This oxygen should go into the cell. You are eating every day the food. Right now you had your breakfast, right? Whatever you had your breakfast doesn't matter. But you had your breakfast. Now all the nutrients from this breakfast, where they will go? They will go into the cell. So whatever you are doing or whatever you are eating, the target area is cell. Means the drug which you are giving to the patient for treating the cause, the drug it should reach into the cell. And this is how you should understand. Means inside the body, everything. Like suppose this is your cell, right? And this is your blood vessel. This is your blood vessel. Now, whatever, like suppose here is the oxygen is present, here is glucose is present, here is protein is present, lipid is present, fat is present, whatever you are eating, where they should all go? They should all go into the cell. And this is how your body functions. And this is how your cell can function. So but always remember, whatever you are taking inside your body, it always reach into the cell. And reaching into the cell, later on we'll discuss about that there are some barriers to entering into the cell. And this barrier number one called as cell membrane, right? So this is how you should think about it, and this is how you should learn about it, right? Then there is a tissue. Tissue means group of cells. And inside of our body, but remember, there are four primary type of tissue, primary tissue. And these primary tissue, number one, that is your neural tissue. Number two, muscular tissue. Number three, epithelial tissue. And number four, that is connective tissue. Neural tissue, muscular tissue, epithelial tissue, and connective tissue. So inside your body, there are four primary type of tissue. Means your whole the body, this is made up of only these four primary type of tissue: neural tissue, muscular tissue, epithelial tissue, and connective tissue. Right? So just write it down. Then there is an organ. Organ means group of tissue. Then the group of organs makes the systems, and group of system makes the organism. So till now we have understood that inside of our body the life started from the cell, and now this cell is the basic function unit of the life. So let's talk about first of all this cell, right? So this cell, the cell actually looks like this. In your school days, you have studied about it, and you have seen it. This is your cell, and inside of your cell there is some different organelles. Organelles. What is the meaning of organelles? But organelle is nothing. Like inside of your body, there are different organs. Basis of these organs, you are functioning. What would happen if I remove any of the organs from your body? Suppose if I remove the heart, can you survive? No, you can't survive. So similarly, these property, these organs making you a human being and making you a living structure. Similarly, for inside the cell, functioning of the cell, we also require the organs. Like what is the function of the lungs? Respiration. What is the function of the heart? That is the pumping. Similarly, what is the function of the kidney? That is filtration. Similarly, inside the body, all these organs, whether they are functioning like this, then only we can survive. So similarly for the cell, there is also inside of the cell, there are numerous type of organs. But as you know, the cell is actually very small in size. So whatever the organ which will be present inside the cell, these organs will be also very numerous in size, very small in size, and that will be called as microorgans, right? And basis of this, in the Latin, anything which is very small, we use the word ilis in the land. We use the word ilis in the end. So that is why organelles. We are using the word, means they are organs of the cell, but they are very small in size. That is why we are using the word cell organelles. So today onwards, whenever you will hear the word organelles, it means small organs. Organelles means small organs, and cell organelle means small organs of the cell. So let's talk about first of all, like similarly inside of our body, there are numerous type of different type of organs there are. And similarly inside of the cell, you have heard about there is biggest organelles. Biggest organ of the skin, biggest organ of the body is skin. You already know. Means I already told you, inside the body, everything is made up of the cell. It means this skin is also made up of the cell. And now this cell converted into the tissue, then tissue from the organ, and organ from the systems. It means this skin is also made up of the cell tissue, and that is why it is also becoming an organ, and that is why we use the word the biggest organ of the body, that is your skin, right? Similarly for the cell, the biggest organelles of the cell is nucleus. Biggest organelles of the cell is nucleus, and there are different type of organelles presents which you have heard in your school days as well. Number one, there is an endoplasmic reticulum, Golgi apparatus, lysosome, peroxisome, centrosome, secretory vesicle, mitochondria, anything. And remember, basis on the presence of membrane. What is the membrane? Outer structure, outer layer. Basis of this outer layer, which is called as membrane, these all organelles is divided into two types. Number one, with membrane. Number two, without membrane. All our organelles are with membrane. Only two organelles are without membrane. Always remember. Number one, that is ribosome. That is ribosome. Number two is cytoskeleton. Ribosome and cytoskeleton. Right? So now, going to the further bit. First, you should understand. Like inside of your body, there are different organs, and these different organs are having their different functions. So similarly, inside of the cell, these different organelles is also having the different functions. First of all, you should understand what are the composition, or what is the composition of our body. like the house where right now you are living. This house is made up of what, beta? This house is made up of the bricks, then the iron bars, then some cement, then some, there is a called as your doors, windows. So like many things are there. Similarly, inside the body, all the body parts is consist of the three things only. And these three things which you are consuming on everyday basis, beta. These three things, beta. Number one, that is protein. Number two is lipid. Number three is carbohydrate. Carbohydrate. So it means in everyday life, whether you are having your lunch, dinner, breakfast, whatever, you are vegetarian, non-vegetarian, what three things you are eating in every product, but only these three things. Number one, protein. Number two, lipid. Number three, carb. Means if you are eating a chips, if you are eating a biscuit, if you are eating the any tikka, paneer tikka, chicken tikka, whatever you are eating, what are the things inside of these products? That is number one is protein, number two is lipid, number three is carb. So that is why all the material which are consisting only these three things, that material only you consume on your daily basis, right? So now question comes. This is the protein, lipid, and carbohydrate you are eating every day. So beta, now when you are eating the protein, carbohydrate, and lipid, it is going inside of your GI tract. And what is the function of GI tract? Digestion. And now the question comes: this digestion is occurring. Why this digestion is occurring? Because beta, this protein which you are consuming, like you are eating an egg. So what is the size of this egg? Like this only. And now what is happening? First you are cutting with your fingers, making the two portion of this egg, then putting inside of your mouth, and then start chewing it. Once you are chewing it, you are making small particles. Now these particles are going inside of your stomach and then duodenum. And what is happening inside of your duodenum? Inside of your duodenum, it is digesting with some enzymes, chemicals. And now why these enzymes are digesting them? Because they are extracting the all related part from this protein. And these small particles, because this protein is bigger in size and we need a smallest part of this protein. And this smallest part of protein is called as the amino acid. So basically for the cell requires the raw material, and this raw material is not that bigger size of the protein. Cell is required the smallest unit of that protein. Cell is required the smallest unit of the lipid, and cell is required the smallest unit of the carbohydrate. So first you should identify when this digestion is occurring, at what stage this digestion should be stopped, isn't it? Because they cannot be like this. Means once you have put this protein inside of your alimentary canal, now this is digesting. Nobody is there to stop it. There is somebody. The body should realize that what is the smallest part from the protein. That smallest part is required for the cell, and this protein is smallest part is beta amino acid. Similarly for lipid, that is fatty acid and glycerol, and similarly for carbohydrate, glucose plus fructose. So these are the small particles of these substances. So now for the cell is required this amino acid first of all, then cell is required fatty acid and glycerol, and then cell required the glucose and fructose. And this is how all this digestion is occurs. So now when these particles are ready inside of the intestine, then what could be happening? Suppose this is your intestine, and now these all particles are ready. Now these particles should be absorbed inside of your intestinal gut, and then it will going into the blood vessels, and it will mix into the blood vessel. And from this blood vessel, the function of blood is always remember the function of blood is transportation. So now all these products will go into the cell, and now this cell will take all these products inside, and now this cell will start functioning of these products, all these three products. So now you know the function of the glucose is like a petrol, the petrol in your car. So this glucose, which is coming over here, this glucose will come over inside the cell, and now inside the cell, you know that there is one organelle called as mitochondria, and this mitochondria function is to generate the energy. That is how our cell can function, because without the energy, our cell cannot be function. So this mitochondria is generating the energy, and for generating the energy, this mitochondria requires the raw material. Like similarly, for making a fire, you require what? You require the woods, you require the wood blocks, and you require some fuel, or either you require the lighter or something else. So then only you can start the firing. And this is similarly for the glucose which is present inside of this blood. This glucose actually comes, and this glucose goes inside of this mitochondria, and then it metabolizes over here. And this is how this glucose actually produces the energy. Similarly, there is oxygen also present, like oxygen also there, and there is a cell, and inside of the cell again there is a mitochondria. So now for burning of this glucose, we require the oxygen. That is, isn't it? For burning of the petrol inside of the motorcycle or inside of the car, we require the oxygen. So similarly for burning of this glucose, we require the oxygen. So whatever oxygen you are taking, this oxygen where is also going? This is also going into the mitochondria. And now beta for glucose, see, that's how things are very simple if you understand them from the basic. Now you know that glucose is breaking into the mitochondria, and then it produces the energy. So it means the breaking of the glucose, we use the word beta always lysis. We use the word lysis. Lysis means breaking. And now at this moment, glucose is breaking down. So that is why we use the word glycolysis. And now this glycolysis is presence of the oxygen, isn't it? So that is why for the oxygen we use the word aerobic, because it is coming from the air. So we use the word aerobic glycolysis. So this is how you should understand the things, right? Why we are consuming the glucose? This glucose will go inside of our body, and then it will mix into the blood. From the blood, it will reach into the cell, and into the cell there is organelle, that is mitochondria. And in this mitochondria, there is oxygen which you are doing by the respiration. You are taking inside of this oxygen. This oxygen should also reach into the cell, and this is how this oxygen will also go into the mitochondria, and then the burning of this glucose will be happen. The process is called as aerobic glycolysis, and this releases the ATP. Similarly, this is the talking about this glucose. Similarly, what about the protein? What about the lipid? So now inside of your building, right now the house where you are living right now, so maximum part of the area is made up of what? The bricks in the walls of your room, walls of your bathroom, walls of your bedroom. Now everything is made up of the bricks, and basis of this, the different number of bricks is present in different walls, and that is how you should understand inside of the body. Similarly, these bricks, the bricks of life, is protein. So now always remember, inside of your body, whatever is made up of, that is made up of the protein, right? And the protein is smallest unit, that is the amino acid, and that is why in the biochemistry, sir also taught you, there are different number of chains of the amino acid. Why? Because like similarly, there is a bricks. Some of the wall is consisting around 500 bricks. The another wall, the bigger room wall, that is consisting around 1000 bricks. The toilet wall is around 100 bricks. So similarly, there is a different chain of this amino acid. That is how our body starts functioning. And this is everything made up inside of our body is made up from this protein. Now, what is the function of the lipid? So some of the part of the body, like inside of your house, there is a glasses, there is a window, there is a gates, everything is present. Similarly, inside the body, these window gates are like lipid. So inside of your body, everything is made up of either from the protein and second from the lipid. And this lipid also functions as a secondary energy provider. These lipid, when you are consuming more in number, it can be stored in your belly, and this belly fat can give you the energy whenever the energy is required, whenever there is a glucose is absent inside of your body. So these fatty acids will start providing the energy to the cell. And that is how, whenever you go to the gym, what your gym trainer says, start consuming the protein and don't eat carbohydrate. Why? Because already you are having the secondary, this part of the fat, secondary part of the energy, that is lipid, which is already present inside of your body. Now you want to consume, you want to remove this lipid, you want to remove this fatty acid from your belly. How you can remove that? Just using of them, you can remove them. Why they are present over here? Because body stores them. Why? Because body thinks right now you have consumed more things. Body doesn't require so much of energy. So now what body did? Body stores it. And where body can store it? The area which is very soft area. And this area is your fat, and this area is your belly, your abdomen. So that is why every time there is a fat deposition occurs on your peritoneum cavity. Now, if you are suppose going into the gym, using all the exercise, doing all the exercise, and putting your body into the aggressive pressure. Now, what is happening? Why doing the exercises? You require the more energy. Now, this energy you are not consuming. Suppose this carbohydrate, because gym trainer is just prohibited you. He said, don't consume more and more glucose. Don't eat sugar. Don't eat any sweets, and don't eat any rice. Don't eat any wheat. Why? Because everywhere there is a carbohydrate, and from the car, the end product is better. That is glucose, and that is this glucose. Actually, if you are providing this glucose directly into the body, first of all, your cell will consume this glucose. Once, if still you are working, and if this glucose is not present inside of your body, then the secondary energy source, that is your fatty acid, this will start consuming by the body, and that is how your belly fat will start going off. And similarly, inside of your body, now you are just doing more and more exercise like this, right? So now what is happening? This area you want to build up. You want to increase the growth of this area. You want to make a muscle mass. And how the muscle mass can be made better? If you want to making a more and more number of the floors in your house, what you require? You require the bricks. And similarly, inside the body, when you are doing more and more exercises, you want to increase the muscle mass over this area. What you require? You require the raw material. And this raw material is amino acid. And that is why your gym trainer always says you, please start consuming more protein and please less eat this carbohydrate. So I hope better now the thing is clear inside of your body. Now, let's go about that. The organoids part better. So now, similarly, inside of your body, that there are different type of organs present, and these different type of organs are having different type of functions. Similarly, inside of your cell, there are also different type of organs, and we call them organelles. And first of all, we should identify what are the function of these organelles. So number one, the information which came from your mother and your father, this information should always present inside of your cell. Then only your cell can be function properly. And actually, this information is in form of DNA, and this DNA is stored inside of the nucleus. So what is the function of nucleus? Function of nucleus is nothing. This is just a DNA storage. Now suppose, beta, if in the America there is a discovery of this Coca-Cola, the Coca-Cola which you drink, this is the carbonated drink. So now what happens? This Coca-Cola is actually invented into the America, but now this Coca-Cola company, actually, it's also started a plant inside of our country in India. Now what happened? Actually, the recipe of this Coca-Cola was presented to the America, but now they make a copy of this recipe, and this copy is sent to the Indian factory as well. Now, basis of this copy into the Indian factory, the same taste of this American Coca-Cola is now started producing inside of our country. How? Due to the information of this recipe. So now, similarly, from your mother and father, all the information is always present inside of your body, and that is why your body starts developing according to your information given by your parents. That is why you look like similar to your parents. Now, what could be happening? Suppose if there is 100 pages of this recipe of the Coca-Cola in the book, what could be happen if you remove the 2 pages from this recipe? Can the taste of this Coca-Cola will be same, or whether there is any defect? Yes, there will be the defect. Now this Coca-Cola cannot make with the same taste. Why? Because there is 2 pages gone. And now basis of these 2 pages, you cannot identify that what is the written inside of these 2 pages. And that is why there is some defect into the taste of this Coca-Cola. Similarly, what happens inside of this DNA, when in this chain, any of the area is removed or deleted via the body, now this is called as genetic disorder. This is called as genetic disorder. So now, if there is already a defect present inside of this nucleus is like a director. Now this director will tell to the other workers of the factory, like for the cell, what are the other workers? The other workers like other organelles. Now what is happening? If this nucleus information is already defected, whatever the production will be happening inside of this cell, inside of this factory, this will be also defective. And these defects will start appearing into a pathology, and those pathology we named as genetic disorder. So now, question comes in the mind, can you cure these genetic disorders? No, because from outside, you cannot give the information. This information is already present inside the body, so you cannot protect or you cannot cure this genetic disorder. But what you can do, you can slow down the progress of this genetic disorder, and that is why we are not having any disease for this genetic disorder, right? And in the pathology, Kunal sir already taught you about this, that autosomal dominant, autosomal recessive. This is how you should understand, right? So let's talk about. There is this organelles, number 1, smooth endoplasmic reticulum, number 2, rough endoplasmic reticulum, then Golgi apparatus, peroxisome, lysosome, mitochondria, nucleus, and many more. So mitochondria function you already know, it is a transformer of the cell, producing the energy. Nucleus function, DNA storage, and smooth endoplasmic reticulum. But smooth already name is suggesting. Smooth means lipid are smooth, oil are smooth. So that is why the smooth endoplasmic reticulum function is lipid synthesis. The function of this is lipid synthesis. Because you are consuming only 3 things: protein, lipid, carbohydrate. From carbohydrate, it is utilized in mitochondria. Lipid will go into the smooth endoplasmic reticulum, and now comes to the protein. This protein, inside of the body, everything is made up of the protein now, you know that. So now this protein, suppose rough endoplasmic reticulum, actually what happens? This is your nucleus, and inside of this nucleus, outside of this nucleus, there is something like this, and this is called as your endoplasmic reticulum. On this endoplasmic reticulum, there is like dots are present like this, and those dots are ribosome actually. That is why this ribosome is always present in attachment with this endoplasmic reticulum. That is why this ribosome never contains its own cell membrane, and that is why if you remember on the above table, we have talked that this is a without membrane organelle, right? And the function of this ribosome, which is present over this rough endoplasmic reticulum, is protein synthesis. The function of rough endoplasmic reticulum is protein synthesis. Protein synthesis, and this protein is actually for export out from cell, right? Now Coming to the next part, that is Golgi apparatus. In Golgi apparatus, now that you have heard about this Golgi apparatus, now suppose inside of the factory, what happens? First of all, you have given the raw material. Now, basis of this director information, these two workers, number one, smooth endoplasmic reticulum and rough endoplasmic reticulum, they both started to making the product. Now this product is ready. Suppose this is a shirt. Now this shirt is ready. Now, what you want with this shirt? You want to this shirt, send it out from the factory. And for sending out from this factory, first you should do the packing of this shirt. Once you will pack it, then what is present inside of this packet, you will put a label on it. And then after you will do the delivery of this packet. So similarly, inside the body, whenever this lipid and protein are ready for using for another cell, now these protein and lipid should be packed and then put a label on it, and then it should be delivered out. And this functions similarly like Amazon, the Amazon which is functioning inside of your city or inside of your state. What is the function of Amazon? It takes the product, pack it, put a label, and deliver it. Similarly, these functions are carried by this Golgi apparatus. So, what is the function of this Golgi apparatus? This is first of all packing, number two labeling, number three is delivery. So, packing, labeling, and delivery is a function of Golgi apparatus. Now, question comes: how the packing can be occurred? Because inside the body there is no cardboard. बाहर तो आपने डब्बे में पैक कर देते हो. But inside the body, from where you will bring this cardboard? So inside of this body, whenever you are packing to this protein or lipid, the third substance which is present inside of your body is glucose. So some of the glucose is taken by this Golgi apparatus, and now this glucose will attach with this protein or will attach with this lipid. And now when any glucose will attach with the protein and lipid, now sensed by the other cells or for understanding for other cells, these protein and lipid now are ready to consume. And now what is happening? You are adding protein with glucose. So now this protein is called as glycoprotein. Similarly, when you are with the lipid, you are adding this glucose, you are called as glycolipid. So this is how these names is originated. So whenever you will see the glycoprotein or glycolipid, means this is a ready protein or ready lipid, right? Now question comes, the packing. Packing inside the body is done via carbohydrate, and labeling is done via mannose 6-phosphate. Labeling is done via mannose 6-phosphate. MCQ. Always remember. Right, brother? Packing is done via carbohydrate and labeling is done via mannose 6-phosphate. Now the next already, that is peroxisome. What is the function of peroxisome? Question comes. Peroxisome is nothing. Suppose if there is this shirt is defective, क्या आप इसको कंपनी से बाहर भेजना चाहेंगे? No. Do you want this shirt? Send it out from your factory. If this shirt is defective, no, you don't want to send it. So now what should be done with this shirt? This defective shirt. This should be cut it down back and should be sent into the recycle. And then the new shirt will be formed from this part. And then when it will be ready, then only it can be delivered. So similarly, if there is any defective lipid synthesis, this lipid will be destructed via this peroxisome. So function of peroxisome is nothing. This is the lipid destruction. And if similarly for the protein, this protein is destructed via lysosome. So now in microbiology, I hope that microbiology is already finished. In microbiology, Salman sir will teach you that any bacteria, when it gets inside of the body, what is the composition of this bacteria? This bacteria is made up of the lipid. This bacteria is made up of the protein. And now this bacteria is killed via the cells. How? Because remember, again, this is your cell, right? Now this is your blood vessel. This is your blood vessel. Suppose this is the bacteria. Now what is happening? This bacteria will come out into this space. Now this is called as interstitial space. And in this interstitial space, nobody is there, no army is there to kill this bacteria. Now what is happening? Inside of this your cell, there is one organelle that is called as lysosome. And function of this lysosome is nothing. This is actually the protein destruction. And your bacteria is also made up of the protein. So now this bacteria will be killed via which organelle? Via peroxisome, via lysosome. And this lysosome present is where? This lysosome actually present inside of this cell. So now what happens when this cell sees that there is this bacteria is there. So this cell comes like this, and this cell engulf of this bacteria inside the cell. And then this lysosome actually splashes the toxic substance on this bacteria, and this bacteria is dead. And these cells are called as macrophages. So this is how all these macrophages, first of all, taking all this bacteria inside of the cell, the process which is called as endocytosis. If cell is drinking pinocytosis, if it is eating phagocytosis, and then only with the help of this organelle, this bacteria can be killed. So बेटा always remember, again and again, I'm telling you, बेटा बच्चे बार-बार मैं आपसे कह रहा हूँ, बॉडी के अंदर, शरीर के अंदर सबसे जो टारगेट एरिया है, वो है बेटा this cell. जो टारगेट एरिया है वो क्या है बेटा? This cell, right? So whatever is coming inside of this blood vessels, this will reach into where? Into this cell, right? So whenever you start assuming the things, better always remember the target area inside the body is cell, right? Now, coming to the next part. Suppose question comes: if in some cell there is no mitochondria is present, then what could be happening? If there is no mitochondria is present, what could be happen? If no mitochondria, it means still it will use the glucose. It will consume the glucose via some secondary process, secondary metabolism actions. So now what is happening? But this time for lysing of this glucose, there is no oxygen is required. So now this glucose is breaking down without using of the oxygen. That is why we use the word anaerobic glycolysis. And this mitochondria is not present inside this RBC cell. So there is no mitochondria. Hence uses anaerobic glycolysis. MCQ. This question is asked in your exam. Quite easy questions. So if you know the basic, you can answer them very easily. Coming to the next part, that is cell. So let's talk about detailed manner about the cell. This cell, you have seen the cell is like this. Now, inside of the cell, this is the biggest organelle. So for understanding of the cell, we will understand the three components of the cell. Number one, that is outside layer, and this is called as cell membrane. Then biggest organelle, that is nucleus. And already I have told you, inside the body there is 60% is water. So the water which is present inside of the cell, we called it ICF, intracellular fluid. And the water which is present outside of the cell, this is called as ECF, extracellular fluid. This is how we differentiate. Later on we'll see there are different chambers into the ECF, and we'll talk about those chambers as well. So now you know that inside of the cell is water, outside of the cell is water. So everywhere inside the body is water. So now this water which is present inside the cell, this water is actually very clear water. And that is why we use the word cytoplasm, because this is the fluid of the cell. So we use the word cytoplasm. So this is the third component is cytoplasm. So for understanding of the cell, there are three components. Number one, cell membrane. Number two, cytoplasm. And number three, nucleus. Now you already know that. whatever is coming from this blood vessels, whatever is coming from this blood vessel, this should reach where beta? Suppose whatever is coming from there, this should reach into the cell. And for reaching into the cell, what we require beta? There should be some channels. Via through this channel, these product can enter inside of the cell. And now these channels are made up of something. Later on we'll discuss about it. Second thing, inside of the body, everything is made up of what? Either it is made up of glucose, either it is made up of the protein, either it is lipid, or either it is carb. So similarly, this cell membrane is also made up of this three component. Number one, protein, lipid, and carb. And remember, these protein, lipid, and carb, they are just making this cell membrane together, and they are present into the alternative ways like this: one lipid, one protein, one lipid, one protein. And this alternative pathway, we use the word mosaic. But we use the word mosaic, and that is why we use the word. This cell membrane is having a model, and this model name is called as fluid mosaic model. Right, brother? So now let's talk about when any products need to go inside of the cell. First barrier is cell membrane. So let's talk about the cell membrane. First of all, as already you know that if any substance need to go inside of the cell, so now you should understand, brother, what is the thickness of the cell membrane. Because as much as there is a smaller thickness, these products can be easily enter inside the cell. But if thickness is more, these products cannot enter inside the cell. So thickness of the cell membrane is 7.5 to 10 nanometer. And if the person is asking to the Armstrong, remember this is 70 to 100 Armstrong, right? Then the structural model of cell membrane. The structural model of cell membrane is called as fluid mosaic model. Fluid mosaic model. And this fluid mosaic model is given by two doctors. Number one, Singer. Number two is Nicholson. And this is given in 1972. This is given in 1972. So fluid mosaic model and given by S.J. Singer and G.L. Nicholson in 1972 MCQ. Coming to the part, what are the substances of the cell membrane? And substances of the cell membrane is nothing. Number one, that is protein, and the protein is around 55%. Then 40% is lipid, and 5% is carbohydrate. 55% is protein, 40% is lipid, and 5% is carbohydrate. Now, this is your cell membrane. This is how it looks like. This is your cell membrane. And now inside of the cell membrane, as you can see, this is the one layer, and this one layer is something looking like into the double layer structure like this. Then you can see this navy blue color. These are the protein area, and in this protein area, you can see there are two types of protein. Number one, protein, which is coming to the entire thickness of the cell membrane, and these proteins are called as integral protein or intrinsic protein. Later on we'll discuss about it. We'll write it down, don't worry. And these proteins, as you can see, this is present only on the peripheral side. So we use the word peripheral protein. And now you already know that these proteins are basically packed with this carbohydrate. So that is why this carbohydrate is also consisting inside of the cell membrane. Now as you know that this area is also having water, and this area is also having water, right? Suppose this is the outside area and this is the inner area. So inside and outside, everywhere there is a fluid, everywhere there is a water. And you know that the lipid, that is fat, it never live or never make the friendship with the water. It always repels with the water. So now how this cell membrane, now how the question comes, now how this cell membrane can be remain attached inside of this water? So for making of this, that is why this lipid layer is actually your bilayer in structure. Why? Because this is your lipid molecule. And in this lipid molecule, this is your head and this is your tail. This head is actually having the property of hydrophilic property. This is your hydrophilic in nature, and hydrophilic means water loving nature. And this tail is actually hydrophobic in nature. This is in hydrophobic in nature. So that is how inside of the cell membrane, as you can see, these tails are inside. This is not making any contact with the water, but outside and inside, only the head is remain attached with this water. And that is how this lipid layer can sustain inside of this water area. So that is why this is all the functions, and you should understand one by one that what are the functions of different protein, lipid, and carbohydrate. So let's talk about number one, the lipid layer. But this lipid layer is, now you know, that's all the product which is present over here and inside of this blood vessels. This product should enter inside of the cell. Now you know that. Now how these products can enter inside of the cell? There is a one barrier, number one, that is your cell membrane, and this cell membrane is actually made up of your lipid, protein, and carbohydrate. So now question comes, what you are eating? Similarly, you are eating only three things. Number one, protein, lipid, and glucose. Lipid, protein, carbohydrate, or glucose. Now, whatever the lipid part, whatever the fatty acid part which you are eating, this fatty acid is present over here. Now this fatty acid should get inside of the cell, and this fatty acid actually goes via this lipid layer. And that is why we use the word, this lipid layer is a semi-permeable in property. So this is a semi-permeable layer. This is bilayer in structure, and this is fluid in nature. This is fluid in nature. Question comes. Now the question comes, this major component, what are the component of this lipid? And inside of the body, when lipid synthesis are there are different type of lipid. And actually when this lipid makes an attachment with the phosphate, this is called as phospholipid. What we call this? Phospholipid. So fatty acid when makes the attachment with the phosphate, we use the word phospholipid. And for making of this lipid layer, this is actually the main component of your body. So for main component, the phospholipid is making this lipid layer, and this is the main component, right? Number two is cholesterol, and number three is glycolipid, the glucose which is attaching with the lipid called as glycolipid. And now remember, when this lipid start breaking down, the biggest part of this lipid is called as triglyceride. Then from triglyceride, it will make the diglyceride, and then it will convert into monoglyceride. Right, beta? So from tri, then di, and then mono. So now you know that this triglyceride is the biggest part of this lipid, and this cell membrane is very thick or thin. This cell membrane is very thin. So beta, that is why this biggest molecule of this lipid never composed or never makes this cell membrane MCQ. So my question asks, beta, that's lipid layer is consist mainly by the phospholipid, and which do not contain the lipid layer, that is triglyceride. That is your triglyceride MCQ. Now, what are the functions of this lipid layer? So, first of all, it provides the structural integrity of the cell, structural integrity, then provide the flexibility, solubility, and it allows lipid or fat soluble substance through the membrane. And question comes now, what are the lipid or fat soluble substance? But remember, all the gases which you are taking inside through your lungs, these gases should also reach into your cells. Now, these gases from the blood vessel, when they will enter into the cell, which layer they will use? They will use this lipid layer, because all these gases are in lipid soluble. So, oxygen, carbon dioxide, nitrogen, carbon monoxide, and also your Saturday night product, alcohol, Similarly, you are eating some vitamins as well, like vitamin A, vitamin B, vitamin C, vitamin D, vitamin E, vitamin K, like similarly. So now the question comes, 4 vitamins are actually lipid soluble, and whenever you are eating those vitamins, they should also reach into the cell. And now when they will process this barrier, they will cross this cell membrane through this lipid layer, and that is why we always study that lipid soluble vitamins are. And remember, beta, this is ADEK. This is ADEK, ADEK. Similarly, some fatty acid, and then the thyroid hormone and steroids. Question comes, beta. Question comes. My question is that what are the hormones? What are the hormones? Write me on the chat box, beta. What do you understand with the hormone? Write me in the chat box. What is the hormone? What is the function of hormone? Yes, बेटा. देखो बेटा, जब भी मैं आपसे क्वेश्चन पूछ रहा हूं तो बेटा मेरे को पता है हार्मोन क्या है, बट आपको वो चीज पता होनी चाहिए, राइट? आई ऑलरेडी नो दैट, आई एम ऑलरेडी डॉक्टर, यू शुड नो दैट. इसलिए बेटा जब भी मैं आपसे पूछूं, प्लीज ट्राई टू आंसर. मैं मानता हूं वी आर थ्रू द ऑनलाइन मोड, सो आई कैन नॉट सी यू राइट नाउ, बट डेफिनेटली बेटा यू कैन सी मी वेरी वेल. सो प्लीज ट्राई टू आंसर ऑलवेज इन द चैट बॉक्स. व्हाट इज द हार्मोन? व्हाट आर द फंक्शन ऑफ दिस हार्मोन? द फंक्शन ऑफ दिस हार्मोन इज नथिंग बेटा. व्हाट इज नथिंग? व्हाट इज हार्मोन? Yes, yes, Anu. Very good, beta. Very good. It is a chemical messenger. Beta, what happens? Remember. What happens, beta? Remember that this is your brain, suppose. And you all know, beta, inside of our body, everything is controlled by the brain. Means right now I'm talking. Why I'm talking? Because this brain is controlling all the part. Now I'm doing my hand like this. Why? Because my brain is controlling this area. So that is why. So inside of your body is everything is controlled by this brain. Now what happens, beta? This brain can control all over your body area via two things. Number one, either it can control via some wires. Suppose this is wire which is going over here, starting from the brain, and then it is going into the organ like this, right? So this is going like this, and these wires we call as a nerve. So this brain, number one, this brain can control all the organ via the nerve. But beta, as remember, if suppose this is a classroom, and inside of this classroom there is a Wi-Fi. This Wi-Fi is coming via the wire. But now, is it possible to provide you all of this, all of you, the single wire to use the net? It's quite impossible. So that is why we use the word Wi-Fi. So this Wi-Fi is going inside of this classroom, and now you are connecting via this Wi-Fi. So similarly, what happens? This brain, the area where this brain can send these wires, this area can be controlled by the nerves. The another area where this brain cannot send all the wires up to that cell level. Now this brain is actually gives the signal to this cell via some chemical, and this chemical we use the word hormones. So what brain did, beta? Brain asked to some cells to make a group, and now this group of cells start functioning together. That is why we use the word gland, and these glands function is to produce the chemical. Now this brain actually attach with this area with the using of a nerve. Whenever via this nerve, this brain actually gives the signal to this gland. What this gland will do? This gland will release the chemical outside the cell, and here it will release, beta. This will release into the blood vessel. This is the blood vessel. Now this chemical will release into the blood, and through this circulation, this blood will take this chemical up to this cell. And now my question is, beta. Now my question is, suppose this is the cell, right? Now this chemical is coming from like this, and this chemical should give the signal to this cell now. Now my question comes, where this signal should reach? Means if you are giving to the any message to any company, but where this message should reach? Up to the director of this company. And who is the director of this cell? The director of this cell is nucleus. So now this hormone is actually should go up to the level of this nucleus, and then this hormone should tell to the nucleus that please listen, man, I am coming over here, and this is the message for you from the brain. Please do something like this. And basis of this message given via this hormone, this nucleus start giving all the orders to their different organelles. Please do function like this, and this is how this cell start functioning. So now this is a chemical, beta. Now ideally this chemical should reach up to the nucleus, but reaching up to the nucleus, what are the barriers? The first barrier, which is this cell membrane. So there is a possibility that this cell membrane cannot allow to come inside of this hormone into this cell. Similarly, there is a watchman inside of your house. Similarly, if any person comes to your house, this watchman do not allow to come inside of your house. So now this message is reached up to where, beta? This reach up to this cell membrane, and now this cell membrane is having the receptor like this. This is having a receptor like this, and this is bind with this receptor. And once it binds with the receptor, it is having a tail, and this tail will send the signal to the nucleus like this. And now this tail will release the another chemical, and those chemical which gives this signal, this message, this hormone signal up to this level of this nucleus, beta. So this is called your primary messenger. This hormone is coming from the brain side, from the gland, to give the message to this nucleus, beta, like this up to here, right? Now this hormone can reach only up to the cell membrane, and that is why we use the word this hormone. This is a primary messenger. Now this message is still not received by this nucleus. Now this receptor will leave or will secrete some chemical, and these chemicals will take this message up to this nucleus, and this is how this nucleus will get the information. So that is why these chemicals, our function is to giving the message to the nucleus, and this message is given by the primary messenger. That is why these chemicals are called as secondary messenger. Secondary messenger. And those secondary messenger, beta, always you have studied CAMP, CGMP, IP3, DAG, and calcium. सुनिए आपने नाम सारे CAMP, CGMP, IP3, DAG, and calcium. So this is how they are functioning into the secondary messenger. And what is the role of secondary messenger? Nothing but just take the message from the receptor and will give to the nucleus. And this is how once this nucleus will get the information, this nucleus will start giving signaling of this cell accordingly. Right, beta? So whenever the hormone will come, where this hormone should reach up to this nucleus. So now maximum hormone they never enter inside of the cell, and that is why they function with the receptor. But 2 or 3 hormones can enter inside of the cell, and those receptors, one of the hormone receptor present inside of the cytoplasm, and few of the receptor present directly on the nucleus. And these questions are asked, beta, many times. The receptor which is present on the for which hormone the receptor is present on directly on the nucleus, and these hormones are your steroid hormone, the receptor which is present into the cytoplasm, and number 2 is thyroid hormone. The receptor is directly over this nucleus. So this is the MC question. So now these two hormones are entering into the cell, and to entering into the cell, now from which area they are entering? From the protein area or from the lipid area? But remember, always these hormones enter inside of the cell via this lipid layer. Okay, via this lipid layer. So I hope now it is clear. This thyroid hormone and steroid, each and every word we are going to discuss about it, because each and every word is having its own meaning, and if we don't understand the meaning of that word, we can't understand the topic, right? So this is all about this lipid layer. Second is protein layer, and this protein layer mainly composed of glycoprotein, which we already discussed about. And function of this protein, but protein is well dissolved in the water. So function is this: allow water soluble substances, allow water soluble substances. There are total three categories. Number one, that is integral or transmembrane protein, and this is also called as intrinsic protein. Intrinsic protein. Number two is peripheral protein, and this is also called as extrinsic protein. Extrinsic protein. Number three is, and this is very less in number, lipid anchored protein. So this integral protein is nothing. It is passed through entire thickness. Pass through entire thickness. And peripheral protein present on peripheral side only. Peripheral side. Then integral protein tightly bound with the membrane. Tightly bound with the membrane. And peripheral protein is loosely bound with the membrane. All the example like that is enzymes, receptors, and pumps. These are made up of this integral protein. And supporting proteins like dystrophin, ankyrin, and spectrin, they are actually this peripheral protein. Now the question comes with the highest protein content per gram of tissue. What is the highest content protein per gram of tissue? First you should write it down, then we'll discuss about it. Highest protein content per gram of tissue is your inner mitochondrial membrane. Inner mitochondrial membrane. And the lowest protein content per gram of tissue, that is your myelin sheath. That is your myelin sheath. Highest protein content per gram of tissue, inner mitochondrial membrane. Lowest protein content per gram of tissue is myelin sheath. I hope that you have noted it. Understanding of this, what is highest protein content and lowest protein content. Suppose inside of your CT, there are some places, ideally, there should be the 55% protein in the cell membrane, then 40% is lipid and 5% is carbohydrate. So, for 55% is protein, 40% is lipid and 5% is carbohydrate. So, like similarly inside of your CT, what is happening? There are more number of your walls is present, which is made up of the bricks, but less number of the windows and less number of these gates are there inside of your house. So now if I ask you the questions, the area where there are no windows are present, it means there are more number of bricks, it means protein is more. And similarly inside of your bank, there is a locker room. Inside of locker room, क्या windows ज़्यादा होती है क्या? नहीं होती. Whether there are more windows? No. There is only one gate. It means there are more number of bricks. Similarly inside of your body, the inner mitochondrial membrane, but inside of this, there is a more than 80% is protein. Ideally it should be 55, but more than 80% is protein and less is lipid. That is why we use the word highest protein content per gram of tissue. The question comes the lowest protein content per gram of tissue. This lowest protein content per gram of tissue, but similarly like in myelin sheath. In myelin sheath, there is 80% is lipid and around 18% is only protein and 2 or 3% is only carb. So now question comes, what is the myelin sheath? You should understand what is the myelin sheath. So for understanding of this myelin sheath, you know that this is your neuron. This is your neuron, right? Beta? Now on this neuron, what happens? Beta? There is every time the function of this neuron is to send the signal. So whatever the signal, this is your dendrite and this is your axon, right? What happens? Beta? All the signal will come from this dendrite into this neuron body, and then through this neuron body, this signal will transmit ahead, transmit forward via this axon, and this area is called as axon terminal. And sending of these all the information, beta, we require the energy, we require the electricity, and electricity which produces inside the body, that is called as action potential, right? So this actually what happens, beta? This axon is having a covering over there like this, covering over here like this, and this is called your beta myelin sheath. This is called your myelin sheath, right? What happens? Beta, remember one thing, I didn't make this myelin sheath like this, okay, or like this. I made it like one boxes, like number one from here to here, then here to here, and then here to here, like this one. And there is a one space in between this. Can you see like this? This space is actually called that's nodes of Ranvier. Nodes of Ranvier. Question comes, why this myelin sheath present over here? What is the function of this myelin sheath? Beta, what happens? Inside of your body, remember there is a 2 ion, 3 ions we are going to discuss later on. Right now we are discussing 2 ions, sodium and potassium. What happens, beta? Inside of your cell, it is always negativity. Remember, your cell in resting condition always negative inside this. Now, you just take a cell, like cell in this remote, the cell in this remote, beta, like this. Now, this remote is getting the electricity with the help of this cell, and inside of this cell, what you have joined these two cells via? If you will join this minus to minus, will it produce the electricity? No. So when the electricity will be produced, beta? When this minus will be attached by the plus, then only the electricity will be produced. So this is plus and minus, plus and minus. Similarly, so what happens, beta? In regular condition, inside of the cell is negativity, and outside of the cell is positivity. When the electricity will be produced, beta? When the electricity will be produced when some of the positive ions will get enter. Some of the positive ion will get enter inside of the cell. Then only this area will become the positive. So this is now positive, this negative, and this produces the action potential. And this is how the action potential starts. So outside there is which ion is present? Question comes. So but outside there is sodium is more, and that is why this sodium gets entry inside of this area, and this inner area becomes positive and then negative, and it makes the action potential. Now if you are going from one side to the another side, if you are going into the jumping state, beta, you will reach very fast. But if you are going to the 1 by 1 foot like this, then you will reach very lately. Similarly, these neurons, beta, this is the biggest cell of the body, and this can as biggest as up to the 1 meter. 1 meter, can you imagine? And now, beta, if Suppose if somebody is pricking you right now, it means this information should be reached up to your brain first of all. Then from your brain, this information or the affecting condition should reach up to the hand, and then you will decide whether you need to remove the hand or not. Right? So this information is going very fast, coming very fast, and for making this faster conduction, this myelin sheath plays the role. So what happens? Actually, this myelin sheath does not allow to go this sodium like this. This myelin sheath allow to go this sodium from this side, then only from this side, then only from this side, and then only from this side. So now you can see that this is coming like a jumping state, and basis of the jumping state, this conduction of the message, this action potential goes very faster. And basis of this myelin sheath, we have divided this nerve fiber into two types: myelinated nerve fiber, non-myelinated nerve fiber. So now you know better. That's what is the function of this myelin sheath. Function of this myelin sheath to give the faster conduction. So now if I remove this myelin sheath from this area, what would happen? The conduction will be slower. Now there are two diseases. Now you know the normal function better, and you know inside of the brain, this brain is having the two parts. Number one, that is central nervous systems. Another is peripheral nervous systems. If these myelin sheaths are destructed into the CNS area or CNS nerve fiber, this disease we call as multiple sclerosis. And if this myelin sheath is destructed into peripheral nervous systems area, this disease we call as Guillain-Barré syndrome. Right, Buddha? So this is how you should identify. So multiple sclerosis you have heard many times, Guillain-Barré syndrome you also have heard many times. So this, if CNS myelin sheath is destructed, multiple sclerosis; if PNS is destructed, Guillain-Barré syndrome. Right, Buddha? So now what are the signs and symptoms will be in multiple sclerosis, Guillain-Barré syndrome? There will be the slower conduction, muscle fatigue, tingling sensation, all the scenario will be occur. So this is how you no need to mug up the things. If you have understood those things from the basic concept, things are going to be very easy for you, right? So this is how you should understand. So this myelin sheath is actually made up of more part of this lipid and less part of this protein. So that is why we use the word lowest protein content, or either I can say the highest lipid content per gram of tissue. Let's talk about the cytoplasm. Cytoplasm is nothing. This is a water, and there is 80% water, and that beta, this is a clear fluid. So that is why it is called as cytosol, and it contains the carbohydrate, lipid, and electrolyte. Number 3 component is nucleus. So before, until now that we have talked about the cell. In cell we have talked about 3 components. Number 1, cell membrane. Number 2, cytoplasm. Number 3, nucleus. In cell membrane we have seen it is made up of these 3 things: protein, carbohydrate, and lipid. Then we are talking about cytoplasm, and in nucleus now the third component is nucleus. But remember, for your exam point of view, the size of this nucleus is 10 to 20 micron, and this is the largest cellular organelle. So till now we have understood the function of cell membrane is: number 1, protection; number 2, selective permeability; number 3, absorption; then activity functions, gas exchange, and maintain shape and size of the cell. So all the points we have discussed about it. Now coming to the next part, and that is called as membrane transport. For understanding of this membrane transport, there are 2 types of transport which you should understand. Number 1, the transport which are using the energy. The transport which are using the energy called as active transport, and the transport which are not using the energy called as passive transport. So remember, if I drop this pen right now from here, what will happen? Where it will go? It will go downside. So this means high to low. Is there using of any energy there? No. Inside of, whenever I am just leaving this pen right now, this is not using any energy. इसमें कोई energy थोड़ी लगी बेटा. So now what happens? Suppose if I take this pen from low to high, now it is using the energy. So means anything substance which is going from low to high, it is always using the energy, and any substance which is going from high to low, it is never uses the energy. And divide basis of this energy, we have divided this membrane transport into 2 types: passive or active. Why? Why we are studying better? Because suppose this is the cell again, and this is the blood vessel. So if any substance which is present over here, if it want to go inside of this cell, means either this substance can be more outside of this cell, so then it can go easily from high side to low side very easily. But either similarly, from inside of the cell, any substance can go outside of this cell as well. So this is how this is using of the energy without using the energy. Basis of this, we using the word membrane transport, and called as passive membrane transport, active membrane transport. So first of all, just write it down. This is energy not required, and this is energy required. Energy not required, and in this energy required. This is from high to low, and this is from low to high. Now understand better. In passive transport and in active transport, there are 4 sub-transport are there. 4 subtypes are there. Number 1 talking about the passive. Number 1, simple diffusion. Number 2, facilitated diffusion. Number 3, non-ionic, and number 4 is osmosis. Again, beta, simple diffusion, facilitated, non-ionic, and osmosis. Similarly, in active, there is a primary active, secondary active, exocytosis, and endocytosis. So now let's talk about one by one. For understanding of these things, beta, first of all, you should understand any negative ion, we use the word anion. So what is the meaning of anion? Anion means negative ion. Any positive ion, we use the word cation. Always remember that. So cation means positive ion, anion means negative ion. Now question comes, this is your cell, and this is your extracellular fluid, this is your intracellular fluid. Now the question comes, what is that? There are 4 questions. Number 1, listen very carefully. Most abundant means more in number. Most abundant intracellular cation is. So remember, inside of the cell, always potassium is more, and outside of the cell, sodium is more. Inside of the cell in the negative ion, phosphate is always more, and outside of the cell, chlorine ion is more. So now question are there. Most abundant intracellular cation, that is potassium. Most abundant intracellular anion, that is phosphate. Most abundant extracellular cation is sodium, and most abundant extracellular anion is chlorine. So 4 questions we have covered. Number 1, most abundant intracellular cation means positive ion, which is positive ion more inside of the cell, potassium. Then most abundant intracellular anion, phosphate. Most abundant extracellular cation, sodium. Most abundant intracellular anion, that is most abundant extracellular anion, that is chlorine. So this is how you should understand. Now question comes, if this sodium, and before that we already talked about inside of the cell is always negativity, and this is called as resting membrane potential, and outside of the cell is always positivity. Now we want to make the positivity inside the cell. Which ion should go inside of the cell? Which ion should go? So sodium should go inside of the cell for making the positivity, right? And this is how. Earlier it is the resting membrane potential, then sodium is always getting inside for making the action potential. And beta, whenever anything that gets inside of the cell, we use the word for inside of the cell, we use the word influx, and for everything which is getting out from the cell, we use the word efflux. So beta, what is the meaning of influx? Influx means outside to inside. What is the efflux? From inside to outside. Now what is happening with the sodium beta? This sodium is influx or efflux. That is, as you can see right now, it is influx. So this is how this sodium is getting inside. Now what happens beta? When this sodium is getting inside, this earlier, suppose the resting membrane potential was minus 70 millivolt. Now this sodium is getting inside, and now this minus 70 is changing into minus 50, then minus 10, then plus 30 millivolt. And this is how this is formation of this action potential. Once this plus 30 is happens over here beta, this sodium channel is actually closes. And once this sodium channel will closes, what could be happen? Now this cell again want to become the negative, and now for become the negative, what this cell should do beta? This cell should remove some positive ion from inside to out, and which is more positive ion inside of this cell? That is potassium. So now this potassium gets outside, and this is called as efflux of potassium. And this is how each and every cell this sodium gets in and potassium gets out for doing the action potential. But ideally beta, what is the home of the sodium? This sodium is always should remain outside of the cell, and this potassium is always should remain inside of the cell. So now what is happening? Earlier there are more sodium, and this is getting very easily inside of the cell, and inside there is more potassium, so that is why potassium gets out very easily. So now what happens beta? When this resting membrane potential again become minus 70, when this become again minus 70, now all the potassium which is getting out from the cell, and all the sodium which is already trapped inside of the cell, this sodium needs to get out and this potassium needs to get in. And whenever this sodium needs to get out, beta, already you know there is more and more sodium is present right now. So when from low to high side, if you want to send anything, you require the energy, and you require the pump. From similarly inside of your house, when you want to send the water from downside to upside, you are using the motor, and this motor is like a pump. So similarly you are using a pump, and this using a pump, actually this pump is having beta 6 sides. This is having 6 seats inside of this car, and 3 seats are reserved for the sodium, always remember beta, and 2 seats are reserved for the potassium with the help of 1 ATP. So energy is form of ATP inside the body. So now beta, what is happening? 3 sodium. What is happening? These 3 sodium is getting efflux. Earlier it was influxing, but now it is effluxing, and 2 potassium is getting influx with the using of 1 ATP, and that is why we use the word beta sodium potassium ATPase pump. What we use the word? Sodium potassium ATPase pump. So now you know that beta, the pump function is, what is the function of this pump? The pump function is nothing. Whenever you want to send from high side, whenever you want to send from low side to high side, you require the pump, and that is why in each and every cell there is a sodium and potassium pump, and this pump is always required the energy. So now you know that. Please write it down beta. Whenever the sodium potassium ATPase pump activates, this is having 6 receptor sites, 3 for sodium, so sodium efflux, 2 for potassium influx, via using 1 ATP. And this is how beta, what is happening? 3 positive is getting out and 2 positive is getting in. It means there is a 1 net loss of positive ion. 3 positive ion बाहर जा रहे हैं और 2 अंदर आ रहे हैं, तो 1 का loss हुआ ना बेटा. So what is happening inside of the cell? Inside of the cell is always become negative, and this is how it becomes a negative. Coming to the part beta, simple diffusion. This is the passive transport. In the simple diffusion, all the gases means oxygen is coming. So outside oxygen is more. So from high to low, this oxygen should reach into the cell. So from high to low, it gets easily inside, and inside there is due to the metabolism, CO2 is producing. So this CO2 gets out again high to low. So this is called as simple diffusion. So gases, example is gases. Number 2, facilitated diffusion. But remember, for understanding of this facilitated diffusion, this is your cell, and this is your blood vessel. Inside of this blood vessel, this is glucose. Now this glucose should reach inside of the cell. But there are 2 types of transporter for the glucose, because this glucose should get inside of the cell. So this glucose is using the transport channel, right? Brother? This is using a channel. So this glucose is divided. These channels, glucose channel, divided into 2 types. Number 1, this glucose when enters with the help of sodium, this is called as SGLT. SGLT stands for sodium glucose linked transport. Sodium glucose linked transport. Number 2, that is GLUT, glucose transporter. Glucose transporter. And remember, these GLUTs are numerous in number, like GLUT 1 to GLUT 5. GLUT 1 to GLUT 5. Okay, always remember. So this is how this glucose getting inside of the cell via the channels. Number 1, that is SGLT. Number 2, that is GLUT. Right? Now what happens, beta? Remember, whenever this GLUT getting inside, how this GLUT is getting inside, beta? Remember, this glucose will not get directly inside of the cell. This glucose will get inside of the cell only when there will be the message from the boss. This is the boss. This brain is boss. When this boss will give the message to this particular cell that please allow to this glucose come inside of your cell. And how this cell will get the message, beta? This boss is having only 2 ways, either through the nerve, either through the chemical. So now this boss will release the chemical, and this chemical is producing by your beta cell in pancreas, and that is beta your insulin. That is your insulin. Now what happens, beta? This insulin is also present inside of this blood. Now this insulin will come, and here is a 1 receptor. Here is a 1 receptor for insulin. Now this insulin will come out, and this insulin will bind with this receptor, and this receptor will send the information to the nucleus via some secondary messenger. And once this nucleus will get the message that please open the channel for the glucose, that boss is saying कि please इस glucose को अंदर आने दो, इस glucose को अंदर आने दो. जैसे ही ये message nucleus को मिलता है बेटा, this glucose starts getting inside of the cell, and that is how outside of the cell, this is your blood vessels, your glucose level is drops down, and this is how your insulin helps to get inside of your glucose, this cell. And that is how this glucose will enter inside of the cell, and it is utilized by the mitochondria, and outside of your blood, outside of your cell area, this glucose level is dropped down. What could be happen, beta? If this insulin is not there, suppose if this insulin is not there, क्या कोई इस receptor को बोल पाएगा क्या कोई ये मैसेज न्यूक्लियस तक लेके जा पाएगा? Can somebody will take this message up to this nucleus in absence of this insulin? No, this receptor is not activating. And once this receptor is not activating, this nucleus is not getting the message from the brain. And once this nucleus is not getting the message from the brain, this channel is not opening, and that is how this glucose is not able to enter inside of the cell. And this glucose will remain inside of this blood vessel, and that lead to hyperglycemia. And this condition called as diabetes mellitus. And that is how this diabetes mellitus can be two types. But there can be two possibilities. Number one possibility, either insulin is not there itself. Again, this receptor is okay, but insulin is not there. Again, this glucose cannot enter inside the cell. This is called as diabetes mellitus type 1, insulin dependent. So now you know that insulin is not there inside the body. So what you can do from outside, you just give the insulin. Once you mix the insulin inside this blood, what could be happen? This will reach again up to this receptor, and all this process will start occurring, and this will lead to the hyperglycemia, to the normal glucose level. Similarly, the another possibility, suppose if this receptor is not functioning properly, hormone is there, but again this receptor is not functioning properly, again this message cannot reach up to this nucleus, and again if it cannot reach up to the nucleus, again no channel opening for the glucose, and again it leads to the hyperglycemia, diabetes type 2. This is called as diabetes mellitus type 2. Right, brother? So this is how all the hormone functions. In the kidney, we'll see, how this hormone is functioning. Remember, this SGLT, sodium glucose link transporter. What happens? Suppose this is your intestine, and this intestine is also made up of the cell. So this is the cell. Remember, inside of this cell, there are total 4 walls. This is number 1, number 2, number 3, number 4. This wall, number 1 wall, the present which is on the apical. This is called as apical wall. What we call this? Apical wall. This is called as apical wall. This apical wall is present to the side of the lumen of your intestine, and behind of this, what is there? There is a blood vessel. So the wall which is present behind of this lumen, this is called your basal wall. This is called your basal wall, and both sides, this is called your lateral wall. So this is how 4 walls present inside of the cell. Number 1, that is basal, that is apical, then basal, then lateral. So now, whatever glucose you are eating, first of all, this glucose should reach inside of the cell, and this cell is called as enterocyte, because this is a cell of the intestine. And for using any word for the cell, we use the word cyte, C-Y-T-E, and this is for the intestine, so called as enterocyte. So now this glucose will enter first of all into the enterocyte, and from this enterocyte, it should reach up to this blood vessels. So now, this should cross via 2 membranes. Number 1 membrane on this apical wall, number 2 membrane on this basal wall. Remember, first of all, this glucose where it is present inside of this lumen, inside of this intestine, because right now you have just consumed rasgulla, and this rasgulla is reaching into this lumen, and now this glucose is present over here. First of all, this glucose should enter inside of this area, and this is called as your SGLT. This transport called your SGLT, and this glucose cannot enter directly. This always with the help of sodium, because there is a 2 receptor present over here, and this is how once the glucose will bind and then sodium will bind, then only this glucose will enter inside, and that is why in the ORS you always mix the glucose with sodium, and that is why 75 and 75, the equal quantity of this glucose and sodium is present inside of this ORS. So this is how this glucose enters inside of your body. Then it mixes into the blood, and through the blood it reaches up to the different type of the cell, and then in the cell it is using via this mitochondria to produce the energy. So this GLUT is always, remember, this GLUT is actually providing there is some facility for this GLUT via the insulin. So that is why we use the word GLUT. Similarly, some water also via the help of ADH. Then there is something called as non-ionic diffusion. Non-ionic diffusion, this is ammonia in kidney. Example is ammonia in kidney, bile salts in ileum, and salicylate in stomach. Ammonia in kidney, bile salts in ileum, salicylate in stomach. These are the 3 examples, but always remember these examples. These are called as non-ionic diffusion. And what is the non-ionic diffusion? Suppose if any substance which is toxic for the cell will not allow to come inside of this cell. So what happens? This ion will become into the different form and become the non-ionic. Then this cell cannot recognize this non-ionic part, and then it can enter inside of the cell, and this is called as non-ionic diffusion. For your exam point of view, just remember all these 3 examples. Number 1, ammonia in kidney. Number 2, bile salts in ileum. Number 3, salicylate in stomach. And then the osmosis, you know the water goes with the help of some solid particles. So this is osmosis. This table is quite important for your exam purpose. Number 2, that is primary active. In the primary active, you already know that sodium, potassium, ATP is formed. In secondary active, remember the SGLT which we talked about. This is also with the using of this energy. So this is SGLT, and this SGLT with 1 and 2. There are 2 types of SGLT, because first of all you are eating this rasgulla and it is going into the intestine. So through the intestine it should absorb inside of this gut. So this is SGLT1, and then with the kidney this glucose can be filtered out, but this glucose is essential for us. So remember, from this kidney again this glucose will be reabsorbed. So there will be SGLT2. For understanding and for remembering these things, this 1 looks like the intestine. So SGLT1 is present into intestine, and there are how many kidneys are there? There are 2 kidneys. So SGLT2 present is always in kidney. Then sodium hydroxide import. Coming to the next, that is exocytosis and endocytosis. What is exocytosis and endocytosis? Remember, suppose this is the cell. If any substance which is more size or bigger size inside of the cell, now this cell wants to send this substance from out of the cell. Can this substance accommodate inside of this gate? No, it cannot accommodate. And once it cannot accommodate inside of this gate, how this cell can be get out this part? And what are the part which is very bigger in size? But all the chemicals, your chemical cells banati na, like neurotransmitters, like pancreas, beta cell, like all these hormones, they are packed into a bag, and these bags is called as secretory vesicles. So whenever inside of the cell, the continuous production is occurring of the cell, but not all the time this insulin will get out from the cell. So that is why this becomes a bigger size of this bag. And now when the cell want to send out this bag, but what would be happen? This cannot fix into this gate. So ideally, this wall should be removed, and then first of all it will attach. This wall should be removed, and there will be a new wall will be formed like this, and then only it get out from here. And this is process called as when anything getting out from the cell, this is called as exocytosis. And remember, this is the 3rd ion, calcium dependent ion. Okay, so it is a calcium dependent process. This is called as exocytosis. Similarly, if any bigger substance present outside of the cell, and which is the bigger substance outside of the cell, any pathogens like bacteria, it should be killed via the cells. And when it can be killed, once it will be get inside of the cell, then only it can kill via the lysosome. So this is the process when cell is eating anything, that is called as phagocytosis, and when cell is drinking any toxic substance, this is called as pinocytosis. And this process, when anything getting inside of the cell, this is called as endocytosis. This is called as endocytosis. And this is protein dependent. So exocytosis is calcium dependent, insulin release and neurotransmitter in the endocytosis is lectin protein dependent, and there is pinocytosis and phagocytosis. So I hope now it is very much clear that passive and active transport, and there are 4 subtypes. So every time the question comes from this table, so please remember all these examples, right? So remember this photo is given in your exam, and they will remove all these names, and they will ask you, they will just put you the arrow, and they will ask which type of transport that is. So remember, now as you can see, this is your lipid part, and this is your protein part. So this is your cell membrane. When any single substance is moving inside or outside of the cell, this type of transport we call as uniport. If any two substances is moving into same direction, two or more than two substances going into the same direction, this is called as symport. And if two or more than two substances is going opposite to each other, that is called as antiport transport. What we call this antiport. So these are nothing. These are basic of the switch ions getting inside of the cell and outside of the cell. If going inside together, symport, opposite, antiport, single substance, uniport. So transport only single substance. Then symport is two substances in same direction. Antiport is two substances in opposite direction. So this is how we should understand, beta. Same direction, opposite direction, and single substance. Single substance. This is uniport. Okay, beta. So uniport, symport, and antiport. So now let's have a break, beta. Break for just for 20-minute break. So let's have a break for 20 minutes, and then we'll continue the next part of the cell. Okay, beta. I hope that still now very much is clear now, and now you know that target area of the body is inside of the body. Target area is cell, and inside of the cell again the target area that is nucleus. So that is all for this, beta. So let's meet after the break. Thank you very much. Welcome back, beta, after the break. Welcome back, beta, after the break. So I hope my voice and my video is very much clear. You can all hear me properly, and you can all see me very clearly, beta. So let's start. Before the break, we have discussed about the cell, and in the cell we have seen that's how the cell functions and how all the different type of transports are there, and we have named that. So in exam, if they will give you this type of images, don't get confused. As you can see, this is the lipid part, and inside of this lipid part, after this, you can see very easily, this is the protein part. So this is the substance which is getting inside or outside. So if two or more than two substances is getting inside or outside in going into the same way, this is called as symport, opposite to each other, antiport, and if single substance is going, this is called as uniport, right, bro? So this is how you should understand. Now coming to the next part, that is called as cell signaling. So now how the cell can talk to yourself. Now let's assume that's you. You can also talk to yourself. आप भी अपने आप से बात कर सकते हैं बिल्कुल. मैं अगर मैं चाहूँ मेरे खुद से बात करना, तो yes, I can do this. Now suppose I hope that you have appeared in this last FMG exam on the 20th January 2023, and what was the scenario on the 3rd of Feb in evening 8 o'clock when the result came out. But you were talking with yourself. आप अपने से खुद से बात कर रहे थे कि मैंने ऐसा क्या कर दिया कि मैं ये पास नहीं हो पाया. So this is how when if you can also talk with yourself. Similarly, the cell can also talk itself, and this is this type of cell signaling. That's in the picture, as you can see. The cell is sending some things and it is getting into that cell only. So if cell target itself only, this cell signaling is called as autocrine. This cell signaling is autocrine. Similarly, when cell target itself, this is called as autocrine cell signaling. Number two, but remember, when suppose two cells are very close to each other, like similarly in your colony, there are two houses which are very close to each other. There is a single wall. So now, if you want to talk with your neighbor, how you can make a talk with your neighbor? So you can make a window in between of this wall, and this window is like a gap. This is called as gap junction. What we call this gap junction. So when the cell, similarly like in picture, as you can see, when the cell talks to this gap junction, this is called as signaling through the gap junction. This is called as signaling through the gap junctions. Next one. Similarly, if the one cell is sending the signal to the nearby cell, and this cell is having a little bit on the distance, means this is not very close to each other, so they are like very little bit in the distance. So now, if the cell is targeting to the nearby of the cell like this cell signaling is called as paracrine cell signaling. What we call this paracrine. So this is all our cell signaling. How the cells talks to each other, because they are function into a group. So definitely they have to send the message that I am functioning this group, I am doing this function, so you also do this functions as well. So this is how similarly when the one cell sends the signal to the another cell nearby to the another cell, this cell signaling is called as paracrine beta. And remember, in the last year PG exam, this question is asked. They have given this picture and they just asked in the given image which type of cell signaling this is, and in the option there was autocrine, paracrine, signaling through gap junction, or endocrine. So our right answer of this question was paracrine cell signaling. So don't be confused. If there is everything inside the body that is cell, and now this cell is targeting to the nearby cell, so this is called as paracrine signaling. So this is when cell target to nearby cell. When cell target to nearby cell, this is called as paracrine cell signaling. Next is endocrine. So now suppose if one cell is present over here and another cell is present over here. So now this is very far, like from here up to this part, right? Now how this cell can send the signal for functioning of this cell? Now this cell can mix this chemical into the blood, and through this blood, the circulation, this chemical can reach up to this cell, and this cell will also get the signal. And when any chemical mix into the blood, as you can see in the picture, this is mixing into the bloodstream, and through this bloodstream, it is reaching up to the target cell. This is called as endocrine cell signaling, and that is why we are always in the hormone. Hormone is a chemical for sending the message. That is why we use the word endocrine systems. So endocrine means when a cell target to the distant cell through the bloodstream, right? So till now you have understood. Inside of the body, cell is the smallest part, and that is the main target area for the body. And in the cell, there is also the nucleus. To entering into the cell, there is a barrier that is called a cell membrane, and this is how we should understand whatever we are eating. It should reach up to where? Into the cell. Into the cell, there are different organelles. Those organelles will start functioning with these products, and anything, the message should reach up to the cell from this brain. This should reach up to the nucleus part, and that is how there are different receptors present on the different site, right? So now let's talk about the next topic, that is called as intracellular junction. In intracellular junction, remember what is the intracellular junctions? Like similarly, when you are making a wall, you have put the bricks. Then this brick, first this brick, you have put it on the floor, and now in between this floor and in this bricks, you have put the cement. That then only this brick can be सस्टेन ऑन दैट पोजीशन, इजंट इट? पहले आप अगर सीमेंट लगाओगे, अगर आप सबसे पहले फ्लोर के ऊपर सीमेंट रखोगे, उसके ऊपर आप ब्रिक रखोगे, तब वो सीमेंट उस ब्रिक को उसके साथ फ्लोर के साथ रोक के रखेगी, और फिर उसके ऊपर क्या करोगे बेटा? व्हाट यू विल डू आफ्टर दिस? यू विल पुट अनदर सीमेंट एंड देन वन मोर ब्रिक, अनदर सीमेंट, वन मोर ब्रिक, एंड दिस इज हाउ यू मेक the wall. So similarly inside the body, remember beta, there are different type of junctions, and these cements are actually called as junctions, and these junctions are present in between the cells, so we use the word intracellular junctions. So number one junction, it is called as anchoring junctions, which actually holds all these cells for a proper structure. That is how your heart is always maintained via this structure, always, right beta? So this is number one, the cement which is called as desmosome, and number two, that is hemidesmosome. So the cement which you are putting on the floor and then putting the first brick, so what is happening with the cement? What is the function of the cement? Actually this cement is attaching this brick with the floor. So similarly this floor is like your matrix and cell is your brick. So now this cell when attached with this matrix, this is with the help of hemidesmosome. This is with the help of hemidesmosome. And now what happens beta? Inside of the body, this cement, this is cell to matrix. This is cell to matrix. And the protein which is used in this, because inside the body, as we already know that inside the body everything is made up of the protein. So now the question comes, the protein which is used in this, that is called as integrin. That is called as integrin protein. Right beta? Next is, now one brick already there. Now what you will do? You will put more cement and then another brick. So now that cement which is present between two cells or the two bricks, this is called as desmosome. So this is cell to cell, and another name of this desmosome is called as beta macula adherens. And the protein which is used in this, that is cadherins. Cadherins. If this desmosome is gone, this can lead to beta. You have studied in the skin pemphigus vulgaris disease. And if this hemidesmosome is gone, this is called as bullous. This can lead to bullous pemphigus. Bullous pemphigus. So this is how you should understand beta. If desmosome is gone, this can lead to pemphigus vulgaris, and if hemidesmosome gone, it can lead to bullous pemphigus. Next is gap junction. This gap junctions, beta, signaling intracellular junctions are called as gap junctions. The protein which is used for making this gap junction, this is called as connexins. Connexins. And remember beta, inside of our body there are three types of cells. Number one, that is skeletal muscle, and inside of the body there are total three types of muscles. Number one, skeletal muscle. Number two, cardiac muscle. Number three, smooth muscles. So your complete body is made up of three types of cells, three types of muscle, and that is number one, skeletal. Number two, cardiac, and number three, that is your smooth muscles. Remember beta, this gap junction is present in all the muscle except skeletal muscle. MCQ. So it is not found in skeletal muscle. Skeletal muscle. Then coming to the next, that is called as tight junction, beta, like your water filters. What happens in the water filters, beta? When you will put the very dirty water, what could be happen? It will pass through the filter, and inside of the filter there is very tight junctions are there, and this water will cross these tight junctions, and the outcome of this water will be there will be neat and pure water. So this is similarly inside the body. What happens, beta? Inside of your body there are many things are there. Similarly, there is called as tight junctions. There is called as tight junctions, and these tight junctions actually for the selective permeability. Like similarly for the brain, this is your brain, and inside of the brain this is a hollow part. And now inside of the brain there is also required the proteins and required the nutrition and everything. Now what happens, beta? This all the nutrition will be provided with the help of blood, but inside of this blood there can be a lot of wastage material can be there, and also there are some pathogens can be there. Now this box is the main part of the body. We don't want that if any infection is occurring over here, from here this pathogen can lead up to this brain. So what happens on the level of this brain? There is a form of choroid plexus. There is a form of choroid plexus, and this choroid plexus is a type of makes the blood brain barrier, and this blood brain barrier actually do not allow to come all the blood cell inside of this area of the brain. So now what happens inside of the blood? As you already know, the blood composition is what, beta? Inside of this blood there is blood cells and plasma. Blood cells and plasma. Plasma is 55% and blood cells is only 45%. Remember beta, in this blood cells there are three types of blood cells. Number one, RBC. Number two, WBC. And number three, your platelets. In plasma content there are, number one, there is a water, which is 92% of total plasma. Then there is a protein, this is 7%, and inside of this protein, remember beta, there are total three types of protein. Number one, albumin. Number two, globulin. And in this globulin again it is alpha, beta and gamma, and this gamma globulin is actually your immunoglobulin. This gamma globulin is actually your immunoglobulin, beta, which you have studied IgG, IgA and everything. And then 1% is other substance. 1% is other substance. So this is how your plasma is formed. So now when the blood cells from the blood cells, this RBC, WBC, platelets will be removed. What is happening, beta? What is remain? This plasma is remain, and in this plasma there is 92% is water. So what is the color of this plasma? It is transparent. And that is how when the blood cell will be removed, whatever is remain, that is always like a fluid. So now we do not call this is a blood. Now we use the word fluid, and this fluid is present inside the brain, cerebro, and inside the spinal cord using the cerebrospinal fluid. So what is the CSF? CSF is also produced from this blood, right, beta? Now this protein, what is the function of the protein? Beta, protein is always, remember, protein is having tendency to hold the water, and this is making a oncotic pressure. This is making the oncotic pressure. What happens, beta? This is your blood vessels. Inside of this blood vessels, what happens, beta? Understand over here. This is your blood vessel, right? This is your cell. Suppose the fluid which is present inside of the cell is intracellular fluid, and outside, that is from here up to here, beta, that is called as your extracellular fluid. Now what happens? This extracellular fluid is having two components, one area from here to here and one area from here to here. The area which is present between the cell and this blood vessels, this area is called as interstitial space, and the fluid which is present over here we call as interstitial fluid. And the inside of this blood vessel, beta, you already know there are two components, number one blood cell, number two plasma. So if we are talking about the fluid where this water is present, into the plasma, so that is why we use the word plasma, right, beta? Now what happens inside of this plasma? Inside of this blood vessel there is a plasma, and this is a water. And what happens with this water? It creates the pressure. This water actually wants to get out from this blood vessels, and this pressure it is producing by the water, and we use the word hydrostatic pressure. What we use the word hydrostatic pressure. So due to this hydrostatic pressure, this water want to leak out from this area, and which area this is? Interstitial fluid. But remember what happens due to the presence of the protein. Due to the presence of the protein inside of this blood vessels, what happens? This protein actually holds this water. This protein holds this water, and this protein do not allow to get this water out from this area, and that is why this water always remain inside of this blood vessel, right? So what could be happen now? This protein can be filtered out from the kidney, because inside of the body there is only one place from where this blood component can get out from the body, and that is the kidney. During the filtration, suppose if this protein leaks out from the kidney, what could be happen now? This oncotic pressure is gone, so only the hydrostatic pressure will remain. And what could be happen now? This water will start leaking out. This water will start leaking out, and where it will come? It will come into the interstitial fluid. Where it will come? It will come interstitial fluid, and once this water will accumulate inside of this interstitial fluid, this will lead to the edema. This will lead to edema. So this is how this edema occurs, and the protein which is leaking out, this condition we call as nephrotic syndrome. What we call this? Nephrotic syndrome. So that is how in nephrotic syndrome there is a feature of this generalized edema, right? So I hope now you have clear about it. So this is making it tight junctions. In this tight junctions, remember another name of this tight junctions is called as zonula occludens. Zonula occludens, and protein which is used in this, that is occludin and claudin, etc. Many type of protein there, occludin and claudin. And example BBB stands for blood-brain barrier. Blood-brain barrier, right? This is called as blood-brain barrier. Number 2 is Sertoli cells. Number 3 is renal tubules. That is how, because from the kidney you don't want to get out everything out from the kidney, so this is renal tubules. And similarly, if you are eating anything non-hygienic food, from this food there can be a lot of pathogens can be there, a lot of microbes can be there. So these microbes should not get inside of your body. So that is why this intestinal mucosa is also made up of this tight junctions. Hope so, so this is how all these junctions can be present. Coming to the next topic, that is called as homeostasis. So now inside the body environment, you always know that inside the body there is normal parameters are there. Means inside of your blood vessel, for example, there is a pressure, and this pressure we use the word blood pressure. Now this blood pressure is always 120 by 180. Now this 120 by 180, this is the normal limit. What could be happen if this blood pressure is getting rise from 150, 120 to 150, 170? What could be happen? Your body will try to do something that this blood pressure can be bring back into the normal condition. So what happens when your body is having its own parameter, and always your body try to maintain your inside parameter, and when your body try to maintain your inside parameter, this condition called as homeostasis. But use the word homeostasis. So homeostasis means homeo means that is same, homeo means same, and stasis means standing. So that is how your body always try to maintain into the same condition. Like your temperature, 37-degree temperature. When you are going outside into the snow area or into cold area, what happens? Your body starts shivering. And why you are shivering? Because when you are shivering, it produces the energy. So you are going outside, there is a cold, and you are start shivering to increase the temperature of your body. That is why this is something called your homeostasis, right? So now this term is given by W. Buchanan in 1930. This is given by W. Buchanan. And now the question asked in your exam: what is the component of this homeostasis? So what could be the component? There are 3 components. Number 1, similar, simple. First, there's something stimulus, means some stimulus should be there. Either there should be some cold, there should be some hot, or there should be something else. So some stimulus should be there. Then this stimulus will be sensed by some receptor, isn't it? Because if I'm touching my phone, for suppose this is my phone, I'm touching over here. What is happening? This is phone is showing this fingerprint scan, and this is how this fingerprint scan is showing, because there are some receptors, there are some sensors. So why are these receptors? Actually, they are sensing. So first should be stimulus, then receptor, and then it's sent to the brain, and coming to the brain, then there are the muscles. So these muscles should start accordingly to functions, and these functions can be to remove the hand or to slap that person. What could be the happens? Anything can be happen. Which be ho sakta hai. So 3 components are there. Number 1, that is stimulus, then receptor, and effector. MCQ. That is stimulus, receptor, and effector. And there are total 3 type of mechanism. Number 1 called as negative feedback, and this is, remember, this is more common. And what is the negative feedback? Suppose similarly, what happens? Suppose you are anything like for your BP. Your BP should be ideally always should be 120 by 80. What happens when the BP rises? But when the BP rises, it means more amount of volume inside of your body. So your body will start to excrete out more and more amount of volume, and that is how you're starting passing the more and more urine. Similarly, your vaso, there is a vessels. These vessels are pipelines, and now the pressure in this pipeline is very high. Now what could be happening if there's high pressure inside of this pipeline can be ruptured. Now for protecting of this pipeline, what could you do? What your body can do? This body can increase the size of this pipeline, and this is called as vasodilation. So this is how whenever there is a change, whenever there is any change to the normal limit, your body will try to maintain this normal limit, and any changes, your body will pull back all these changes, and again it will come into the normal position, and this is called as negative feedback. It means any changes is occurring, and your body will try to pull back all changes into the normal condition. This is called as negative feedback, and remember that this is more common. Next is positive feedback. It means some changes is occurring from here, some changes is occurring, and this changes is occurring above the normal limit. Now your body is also trying to push that this changes is occurring. Let do this changes more and more. Why our body wants these changes? Why your body wants to going beyond above the limit or beyond the limit? But this could be happen. Similarly, for a pregnant lady, for example, for a pregnant lady, what happens when a baby delivers from the cervix? What is happening? Ideally, the cervix should be always into a contact position. What happening when this head of the baby comes with the contact of the cervix? This cervix start dilating, and your body now the cervix is dilating. This is not normal condition for your body, but what your body is also trying. This body is also wants to baby this get it out. Now what happens? Your body starts secreting more and more oxytocin, and this more and more oxytocin will increase the contraction, increase the cervix dilatation above the normal limit, and this is how this baby can be delivered. And this is when the changes is occurring, and your body is also pushing these changes. This is called as positive feedback. This is called as positive feedback. Like similarly, blood clotting, milk ejection. How the blood clotting? Normally there is a platelet present, but now normally in general condition, platelet do not form any clot inside the body. What could be happen if they will form the clot? This clot can be going inside of your coronary arteries, and it can block the coronary arteries, and you can lead to the infarct. So now what happens, beta? Suppose you are cutting some fruits, and now due to by accident you just cut down your finger. Now there is a bleeding. Ideally, no clot formation should be there, but now there are two conditions: whether to make the clot or not to make the clot. If you will not make the clot, your blood will start losing and losing and losing. So now your body will start forming the clot, and once the clot will start forming, what your body will try to do more and more and more and more clot. So this blood clotting factor, this is also a type of positive feedback. There is a milk ejection reflex, childbirth. This is not Ferguson, this is Ferguson. Make the correction. This is Ferguson reflex. Then the LSS during ovulation. Number 3 type is feed forward. Number 3 is feed forward. In the feed forward, remember what is the feed forward? Suppose if somebody will tell you that today in the evening you will eat the biryani, right? So just hearing the name of biryani, still your mouth already getting the water. Biryani is not here, but even just by the anticipating, that's yes, after the end of the class you will have the biryani. So just by the anticipation, your mouth starts watering. And what is happening? Why this is? Due to this, this is called as feed forward mechanism. This is called as feed forward mechanism. It means stimulus is not there, but still you are showing some response. This is called as feed forward regulation, right? And this is also called as, this is why the anticipation, so called as anticipatory system. Anticipatory system, like saliva secretion before the food or body temperature regulation. So this is all from this cell part, beta. So this cell part we have covered, and we already know about, I already told you, beta, that 60% is water, 40% is solid mass, and this solid mass is made up from the cell. So in the cell we have seen each and every condition. So now after each and every chapter, beta, there are some remember points. Please do not forget to revise these remember points just before your exam, and they are quite important for your exam purpose, right, beta? So this is exercise questions. So what is the thickness of cell membrane? Now you should answer what is the thickness of cell membrane. This is 8 to 10 nanometer. Then human cell is the basic structural function of the life. It contains cell membrane, cytoplasm, nucleus. All are true about lipid layer of cell membrane except. Except means it's semi-permeable. Yes, it is true. Mainly composed of phospholipid. True. Doesn't contain triglyceride. True. Allowed to pass vitamin C. That is false, beta. It is allowed to pass A, D, E, K. Vitamin A, vitamin D, vitamin E, vitamin K. Yes, beta. Which is an example of calcium-dependent transport. Calcium-dependent transport, beta. We already talked about, that is exocytosis. Protein associated with zonal occludence. Zonal occludence means tight junction. This is tight junction, and the protein which you use in the tight junction, that is claudin. A 30-year-old boy met an accident on examination, general condition, stable, conscious, oriented, having minor lacerations or elbow bleeding. After some time, his bleeding stops. Identify what type of feedback this is. So bleeding is stopping. So this is which type of feedback, beta? This is your positive feedback mechanism. Positive feedback mechanism. So this is how all this chapter is complete, beta. Coming to the next part, and the next chapter, that is your body fluid. In the body fluid, we already talked about, beta. That's your total body water is 60% of total body weight. Total body water is 60% of total body weight, right? So now remember, beta. In adult, there is around 60% is water. In newborn, there is 75% is water. इसलिए बच्चे बेटा ज़्यादा puffy नजर आते हैं अपने को. In female, the water content is less due to more adipose tissue, and this is only 55%. So suppose for a mean weight, the person which is weight around 70 kg, beta, what is the total body water? So 60% of 70 kg is 42 liter. And now I have told you, this water is present. This is the cell. This is the blood vessel. So water which is present inside the cell, this is intracellular fluid. Outside of the cell, this is called as extracellular fluid. And in extracellular fluid, two compartments we have talked about. Number 1, that is interstitial fluid, and number 2, that is plasma. Remember, beta, this ICF is more in number, and this ECF is less in number. So out of this total 42 liter, or out of this 60% water, 40% water is present inside of this ICF, and only 20% present in this ECF. So this ICF is 2/3, ICF is 2/3, and ECF is 1/3. And in ECF, again you have seen there are two components, that is plasma and interstitial fluid. So plasma is around 1/4 of total ECF, or 25%, and interstitial fluid is 3/4 or 75% of total ECF. This interstitial fluid, remember one thing. Whatever the exchange will be occurring, this exchange will be occurring in between this cell and in this blood vessel via only this interstitial fluid. So this interstitial fluid is also called as tissue fluid, and this area is also called as internal environment. What we call this? Internal environment. Because whatever the exchange will be occur, this exchange will be occur only with the help of, or only from through this interstitial fluid. So that is why this is called as internal environment, and this internal environment is having a French word called as milieu interior. Right, beta? So this is tissue fluid. This is your tissue fluid. Right? Talking about the composition, remember, beta, this is your ECF, and this ECF is contained both these two things. Okay. This ECF contain both these two things. Number 1, that is plasma. Number 2, ISF, interstitial fluid. Now remember, in this plasma, there are nutrients is present. There is any other substance is present. And what is happening? There is also the presence of the plasma protein. Remember, all the substances which is present inside of this blood, it is also with the same amount of inside of this interstitial fluid, except protein. So plasma protein is always remain inside of this vessel. Okay, beta? Remember, plasma protein will never leak out into the interstitial fluid. It is always remain inside of this plasma. Right, beta? So this is, write it down. So composition of plasma and interstitial fluid are same, like all the ions and everything except plasma protein. Remember, beta, all the plasma protein like albumin, alpha, beta globulin, and fibrinogen, they all are synthesized inside of this liver. They all are synthesized inside of the liver except gamma globulin. This gamma globulin also called as immunoglobulins, and it produces into the B lymphocyte. All exchange between vessel and cell occur between interstitial fluid, and this is called as internal environment, called as milieu interior. And this term is given by, again, the father of physiology, that is Claude Bernard. Coming to the next, the measurement of body fluid, beta. So now this is the body fluid. Now you should identify that what is the body fluid in which compartment, that is how much is there. So the indicator how you can measure the body fluid, beta, you will put a dye, and this dye will mix into the water, and then you will take the sample and you will find the concentration. So this is called as dye dilution method. So now not all the dye you can use, beta. The dye you can use only there are something called that is eligibility criteria. Like you, beta, सारे लोग FMG दे देते हैं क्या बेटा? सारे डॉक्टर FMG दे सकते हैं? No, only those doctors can give the FMG which are completing their MBBS degree from outside this country. Only those are eligible to sit in this exam. Similarly, not all the dye we are using for measuring of this body fluid. The dye which is uniformly distributed or acquired equally, that dye only we can use for measuring this body fluid. And the rate of this dye is, beta, Once we will give this dye to the patient, we ask the patient to sit for 30 minutes to 1 hour. Just make a correction over 30 minutes to 1 hour. And formula for this is V is equal to M upon C. V is volume, M is mass of total marker substance, C is concentration in sample fluid. Now, something called correction factor. What happens, beta? If you will ask the patient to sit for half an hour or 1 hour, this patient want to go in between the urination. And once this patient is going for the urination, there can be a possibility that this guy can be leaked out from this urine. So now, whenever you take the sample from this patient, might be there's something error because you cannot measure all the concentration accordingly. So now what happens, beta? Whenever the patient wants to go for the urination in between this mean period, you ask to this patient, please urinate into a pot. So now you are having two samples, one sample from the veins and one sample from the pot. And now you will measure both the concentration area and accordingly we will calculate that how much amount of substance marker you have given and how much of that concentration of that substance into this both sample. This is called as correction factor. So in this correction factor, amount injected minus amount excreted upon concentration. Right, beta? Now coming to the dye, which dye we used for measuring the different volumes. So for total body water, this TBW stands for beta, total body water. So for measuring the total body water, we use the word heavy water, and this heavy water is actually deuterium oxide. This is most common, D2O. Also, we can use the T2O, titanium oxide, or antipyrine. So in the exam, if they will give you all this dye and if they will ask you the total body water, the most appropriate answer will be deuterium oxide, D2O. Simply for ECF, again for ECF, you will use beta inulin. And remember, beta, tomorrow we'll discuss this inulin we also use for measuring the glomerular filtration rate, GFR. Right? Also, we can use the sucrose, mannitol, sodium thiosulfate, sodium thiocyanate. Then for the plasma, we use Evans blue. For plasma, we use the Evans blue. Also, we can use the radioactive iodine, which binds with the protein like I131 and 125. This Evans blue is most common. Remember, this Evans blue is most common. Next is ICF. You can't measure this ICF directly. Why? Because you are giving the dye into this, first of all, the vessel. Now this should enter into this area, then this area. So now directly you cannot measure this area. So now how you can measure this area better? So this is called as indirect method. And you are having a dye for total body water and you are having a dye for ECF. So first calculate total body water and then calculate the ECF. What is remain? That is ICF. Similarly for interstitial fluid, you have dye for ECF, dye for plasma volume. What is the remain? That is interstitial fluid. There is something called that is transcellular fluid. This transcellular fluid is a part of ECF and this is 5% of total body water, like all the synovial fluid, CSF, pleural fluid, anything. Now coming to the next part, that is osmolarity and osmolality. Those two words, but every time it makes a confusion, osmolarity and osmolality. For osmolarity and osmolality, for understanding, first of all, you should understand what is the solution. What is the solution? So the solution is nothing. It is a combination of solute plus solvent. It is a combination of solute plus solvent. So when the solvent will mix with the solute, this will become now the solution. Right, beta? Now, osmolarity. Osmolarity means number of solute particles in per liter of solvent. If any solute particles is present in the solvent and this solvent is liquid, so we use the word liter. We use the unit liter. We use the word osmolarity. But we use the word osmolarity. And you know, in plasma there is a water. In plasma there is a water and there is also the ions. So now for water we use the unit, that is liter, and ions is present in this water. So that is why we use the word plasma osmolarity. But we use the word plasma osmolarity. Similarly for plasma osmolarity, what is the meaning of osmolarity? Number of solute particles in per kilogram of solvent. Coming to the next part. Inside of the body, there is more sodium present in ECF part. And now we already know that's whenever we are putting the namak inside of this vegetable, we use the word gram. How much gram namak you are putting, how much salt you are putting into this vegetable. But for the body, we never use the word gram. We use the word mole. And in 1 mole, there is 23 gram per liter water. And plasma osmolarity is roughly 285 to 295 milliosmol per liter. And for just remember, this is around 300 milliosmol per liter. It means in 1 liter of plasma, there are around 300 moles of solute is present. Coming to the next, that is tonicity. Whenever you are measuring the effective osmolarity, whenever you are measuring the effective osmolarity, this is called as tonicity. And basis of this tonicity, as you already know, what is the component of the other substance into the plasma? Plasma में कितने substance है बेटा? That is 1%. As you can see, this is the other substance is 1%. 92% is water, 7% is protein, and 1% is other substance. In other substance, there is an ion, and these ions are around 0.9%. And that is why it means inside the body, the fluid osmolarity is 0.9%. That is 0.9%. So if any water, suppose this is the water, inside of this glass is the water. If this water is having the same composition, like that much of water and that much of solute, that the solute which is present inside the body, it is also having the same property like 0.9%. Then this fluid is using the word. For same, we use the word iso, and this is the tonicity. So that is why we use the word isotonic fluid. Means same fluid, that the fluid which is present inside the body, this is the same fluid composition which is present outside. Right, beta? So that is why in the NaCl, in sodium chloride, normal saline bottle, have you seen every time there is a mentioning of 0.9% and there is mention also NaCl. Why NaCl? Because now you know in ECF there is more sodium and chlorine. So that is why this is NaCl in this water, and this NaCl is having same composition like the body. That is why we use the word isotonic fluid. If any solid particles which is having more than 0.9%, we are called as hypertonic fluid. Anything which is less than hypotonic fluid, that is hypotonic fluid. So isotonic fluid is same tonicity like normal saline ringer lactate, and recommendation is beta diarrhea, trauma, excessive vomiting. Hypertonic fluid is more tonicity as body fluids. And what happens beta whenever, suppose this is the cell, and now you are giving more sodium outside of the cell. So what is happening beta? Now this more sodium will also enter into more number into this interstitial fluid. Now this area is having more sodium. This area having more sodium, but our body always maintain the balance. So now how the balance can be maintained beta? Either this more sodium should be little bit uptake via this cell, and this sodium should reach inside of the cell. So now if some of the sodium will get inside of the cell, what is happening beta? The number of sodium particles will be less, but now you know if this sodium will enter inside of the cell is already negative, and once the sodium will enter inside of the cell, it will lead to the action potential. So this cell don't wants to make this action potential right now. So now what is another choice with this cell? This cell is having more water. ICF, you know, that is 2/3 of total body water, and this cell is having more water. So this cell will donate some of the water outside, and once this cell will donate the some of the water outside, what will be happening? This sodium will be dissolved in more water, so the tonicity will be decreased. And now, if the water is getting out from the cell, what about the size of the cell? The size of the cell will be shrunken. So this is how the recommendation of this hypertonic fluid. Now, why we are giving the hypertonic fluid to the patient? You know there is a raised intracranial pressure. Now, what could be happen? This raised intracranial pressure can squeeze up all the blood vessels, and once this blood vessel will be squeezed up, there will be no blood flow through these vessels, and this will lead to the ischemia into the brain. Now, what is the initial or very rapid treatment for this patient? That you just introduce the hypertonic solution. This hypertonic solution will reach into the brain, and this will reduce the cell sizes. Once the cell sizes will be reduced, the pressure will be decreased. So this recommendation is head injury. So this is how the water leakage lead to cell shrinkage, and this is in head injury to reduce ICP. Similarly, hypotonic, that is less tonicity, and this is similarly what happens when there is more water. Now opposite you are doing. Now you are doing opposite. Suppose you are giving more water over here, more water, and this more water also comes like here. Now again the homeostasis should be maintained. What could be happen? This cell cannot take this cell. What can do? This cell will take some of the water inside of the cell. So now the water content outside of the cell will be little bit less. But now what will be the size of the cells of the cell? The size of the cell will be increased. So whenever you are giving the hypotonic solution to the patient, the cell size will be increased. Cell swelling will be there. And whenever you are giving the hypertonic solution, there will be cell shrinkage. And whenever you are giving the isotonic fluid, there will be no changes in cell size. So this is how this recommendation is occurs. So this is water absorbs, and this can lead to cell swelling, and this is maintenance fluid in the acute infection. Coming to the next, that is what daily fluid intake and output. So now question comes here. There can be two possibilities. Number one, either you are doing the normal functions. Number two, you are doing the prolonged or heavy exercise. Prolonged or heavy exercise. So now we are talking about the per day water loss and per day water intake. So number one condition is normal condition. In normal condition, what happens? There is a fluid which you are drinking every day. That is around 2100 ml. How much? 2100 ml. Then fluid from the metabolism. From the metabolism, fluid is forming, isn't it? But what happens? Remember, this is your cell. Inside of the cell, there is a metabolism, and through this metabolism, CO2 is produced and water is produced. When the CO2 and water is mixed together, this is done via carbonic anhydrase Ca, and this will lead to into S2CO3. This S2CO3 is very weak substance, and this is called as carbonic acid. What do you call this? Carbonic acid. And this S2CO3 will be divided into H plus and SCO3 minus. And this is how this hydrogen ion is formed and bicarbonate is formed. So this is how inside of the cell every time there is a production of the water, and this is around 200 ml per day. And that is how you are consuming in a normal condition 2300 ml per day water. So now if 2300 ml you are getting inside your body, then suppose definitely you will maintain the homeostasis. So 2300 ml also you want to excrete out from the body, isn't it? So let's talk about the output. Number one, that is insensible loss of skin and lungs. Insensible loss of skin means water losing through the skin. How is possible? Sweating? No, beta. No sweating. Sweating is already mentioned over. So means, what happens? Right now your skin is very dry or it is moist. If it will be very dry, it can be ruptured. So now, right now your skin is very moist. And if your skin is very moist, why? Because of water. And this water is continuously evaporating as well. But now this evaporation, you cannot feel that. That is why we use the word insensible water loss through skin. And this is every day with 350 ml. This is every day 350 ml. Similarly, if you are standing into a very hot environment, what happens? You are taking the very dry air from outside to inside. Now this dry air can be injured to your alveoli, and now you don't want to injure your alveoli. So what you want? You just want to humidify this air, and for humidify this air, we require the water. And again, you cannot feel the water loss, and this is called as insensible lungs water loss. And this is again 350. That is we are talking about in the normal condition. Then through the sweat 100, through feces 100, and urine examination is 1400, and this is total is 2300 ml. 2300 ml. Similarly, if a person is doing a prolonged or heavy exercise, how much water he is drinking, you cannot calculate. But remember, from the metabolism, it is always again 200. Then the insensible loss of skin, but skin loss will be the same. But now when the patient is doing or the person is doing exercises, he is hyperventilating. And once the hyperventilating, more air come inside, more humidify is there, more water loss is there, and this water loss is around 650 ml. Now sweating will be increased around 5000 ml, and again with the feces 100, and through this urine. Now this urine is less, and this is only the 500 ml. But always remember, less than normal urine output is 1 to 1.5 liter. That is normal. Less than 500 ml oligouria, and more than 3 liter. That is called as polyuria. That is called as polyuria. Right, beta? So this is how again all this water losses happens, and all this homeostasis can be maintained. So this is all about this body fluid as well. Coming to some questions, but again the remember point, don't forget to remember. So 30-year-old boy having 70 kg, identify the interstitial fluid. This interstitial fluid is 10 liter only. Plasma and interstitial fluid are component of extracellular fluid. Composition of plasma and interstitial fluid are same except sodium, yes, potassium, yes, glucose, yes, albumin, no. But no protein can be come out into the interstitial fluid. The glucose absorbed intestine by the GLUT. But GLUT already you know this is a facilitated diffusion. Yes. Secondary active transport is transporter across cell membrane is ATP, which is example of this type of transport. But a secondary active, that is SGLT, and SGLT is glucose transport through SGLT. Now coming to the next question, all are false about symport except. All are false about symport except. But a symport means two or more than two substances are going into the same direction. This is called a symport. And now this is everything is false about the symport except. It means one thing is true and one thing, two substances moves in the same direction. This is the right answer. So make the correction, but this is not C. This is the B. Right? This is your B. So this is how the cell and body fluid you already finished. I hope that many things is clear now in your mind. And if you know the basic concept that whatever you are consuming, whatever it is going inside your body, where it is reaching, this all reaching inside of your cell, and inside of your cell, where it is going into your different organelles, and the message should go into the nucleus, right? So coming to the next chapter, and that is your muscle physiology. That is your muscle physiology. And inside the body, already I have told you there are three types of muscles. Number one, skeletal. Number two, cardiac. Number three, smooth muscles. In this chapter, we will go into detailed discussion about the skeletal muscle, and we will something talk about this smooth and cardiac muscles, right? So now let's talk about. Our body is having more than 200 types of muscles, and depending on different methods, we are divided into this muscles into different types. Number one, depending upon the striations. But what is the striations? The striations is nothing. It is a straight line. A straight line in between the muscle. This is called as striations. If there is some straight line present inside of this muscle, this is called as stratified, and if not present, this is called as non-stratified. Now you already know this skeletal muscle is very solid. Why? Because due to these lines. So that is why this is called as stratified muscles. Similarly, this cardiac muscle is also having these lines. So again, this is stratified muscles. But smooth muscles, they are, as name is suggesting, very smooth. So this line should not be present. So that is they are non-stratified muscles. Similarly, transverse line, skeletal muscle is smooth, and skeletal and cardiac, and non-stratified that is smooth muscles. Then depending upon the control, but some of the muscle is in very well control in your So they are called as somatic control, and they are which muscles in your very well control? This all the skeletal muscles, right beta? Another that is the involuntary muscle, that is your cardiac and smooth muscle. Can you ask your heart? Please don't beat right now. आप क्या पूछ सकते हो कि अभी भैया अभी मत धड़कना, गर्लफ्रेंड भी जा रही है, उसको चले जाने दो, जब वो चली जाएगी उसके बाद धड़कना, क्या ऐसा पॉसिबल है? Is it possible for you? No beta, this is not at all possible. So that is why this is the involuntary muscles. Now coming to the question, this voluntary muscle is supplied by the somatic nerve and this is supplied by the autonomic nervous systems. Understanding of this, first you should understand the nervous system beta. So just write it down. This nervous systems is divided into two types. Number one, that is central nervous system, CNS, and number two is PNS, peripheral nervous system. In peripheral nervous system, there are the nerves, and in CNS there is number one, that is brain, and number two is spinal cord. Spinal cord. Any nerve which is taking sensation from organ to brain, what we call this? This is your sensory. This is your sensory. From brain to organ, this is called your motor. So now this PNS is also divided into sensory and motor, and the sensory is also called as afferent. अंदर आती है सारी की सारी nerves, and motor is also called as efferent. Exit होती है, so efferent. Now this motor is going into two areas. One is voluntary area, one is involuntary area. The motor nerves, which is going to into the voluntary area, first of all, they are called as somatic nerve. They are called as somatic nerve. And involuntary, we are using the word autonomic nervous systems. And this autonomic nervous systems again divided into two. That is into flighting condition, that is sympathetic, in resting condition, parasympathetic. Right? So this is how your nervous system is divided. If you don't understand these all methods, you can't understand why we are using the word somatic and why we are using the word this. Why we are using the word somatic and why we are using the word this ANS. Right? So I hope now it is clear. So if you want to draw this, you can draw this. Now, coming to the situations, this situated on the skeletal area called the skeletal, on heart muscle, cardiac muscle, and for the visceral organ, we use the word smooth muscles or visceral muscle. Right? Visceral muscle or smooth muscles. Let's talk about first of all the skeletal muscle. This skeletal muscle is stratified and voluntary. And suppose first of all this skeletal muscle should remain attached with the bone. So this is my bone. So this is the one muscle. This should remain attached with this bone, and attach it with this bone. There should be some structure, and this structure is called as tendon. So tendon is nothing. This is muscle to bone attachment. Muscle to bone attachment. And if you stretch this tendon, this will now become the aponeurosis. So what is aponeurosis? This is flat stretched tendon. Now coming to the topic of this muscle, as you can see, this is your muscle. Have you seen the cable on the roadside? There is a bigger size cable. What is happening when you cut down this cable? You will see there are the bundle of the wires like this, and these bundle of wires similarly present inside of this muscle as well. When you cut this muscle like this, what happening? You can see there is like these bundles are there. Now if you take out the one bundle of this wire, what could be happen? You are taking out one. Again there is a small bundles inside of this different wires, red, blue, black, anything. So now what is happening? You are taking out this one red color bundle as well. So suppose now when you cut down this red color bundle, again there is a copper wire inside of this wire. There is a copper wires like very small wires, and this is small wires like this. And this is called as microfilaments. What we call this? Microfilaments. Now what I did, when I zoom this area, and if I see like this, what I found there are some lines over there and some dark lines like this as well. And now inside the body, everything is made up of the protein. Everything is made up of the protein. So similarly, this is also made up of the protein. So now let's talk about what are the proteins present inside this muscle. So what I've seen, when there is two lines like this, these two lines. So these two lines, actually what I did, these two lines, I call that Z line. What we call that? Z line. In between two Z line, another similar structure is again repeating like this. This is repeating and this is also repeating like this. So what I've seen, that I should identify between these two Z line, whatever is present inside this two Z line, this is everywhere it is repeating in all the muscles. So now I have focused on this two Z line. Now in between of this two Z line, as you can see, there is a one line in between of this, and this is in the middle portion. That is why we use the word this is M line. This is your M line. So Z line and M line. Now as you can see, if you will do the zoom in between of this, what you have found? So this is your Z line, this is your Z line, this is your M line, and this red color filament, this red color, this is a protein. This is your protein. Also, this is also your protein, right? So this red color protein, this is called as your myosin. What we call this? This is your myosin. And another, this protein is called as your actin. So now this is the main protein part of your muscle. So your muscle is made up of this actin and myosin. So this myosin is actually your thick filament. As you can see, this is very thick, red in color. And another one is thin filament, that is your actin. Now, as area, where you can see the area from this, where this complete myosin is present, this is called as A band. What we call this? A band. And the area where no this myosin is present, only actin is present. So this actin is present from here up to the next actin is also starting from the here. So this area is actually completing consisting of this. Similarly, this side of area as well. Now this area is called as I band. What we call this? I band. This is called as I band. Now in between of this area where no actin is present, only myosin is present, this is called as H zone. This is called as H zone. So now I hope that you have drawn this complete. So first of all, 2Z line, then in between M line, then the complete myosin area. This is called as A band. Except of this myosin, there is also the actin. So exclusively actin area, this is called as I band. And now the area between these two Z line, this is actually your functional unit of your muscle. This is how your muscle can do the contraction. Because what is the function of the muscle? It can do only the contraction and relaxation. Contraction, relaxation, contraction, relaxation. So this is the function of this muscle. Now this 2Z line area, this is called as sarcomere. What we call this? Sarcomere. And it is the structural and functional unit of the skeletal muscle. So now can I call this sarcomere? The sarcomere is nothing, it is present between 2Z line. Another thing also I can use the band which is coming into this one sarcomere is half band of this side, half I band of this side, and half I band of this side. So how I can use? This is half I band, full A band, and half I band. This is your one sarcomere. This is your one sarcomere. So this sarcomere is nothing, it is the functional unit of skeletal muscle. It is enclosed by 2Z line called as sarcomere, and sarcomere is nothing, it is half I band plus A band plus half I band. This is our one sarcomere. This is your one sarcomere, half I band plus A band plus half I band. This is your one sarcomere. Okay. Coming to the next part, how the muscle contraction is happens, beta. Look at over the screen. What happens, beta? This actually, this myosin, as you can see, beta, this myosin is having some heads over here. As you can see, there are some heads, right? And this is your actin, and this actin is having, there is some depression, which is called as binding site. So what happens, beta? Actually, this is your myosin actin like this, and there is the head of this myosin. Whenever this myosin head is get attached with this actin, this myosin head is actually tilt like this. This will tilt like this, or this is attaching like this, and it is tilting like this, and this will lead to the muscle contraction. This will lead to muscle contraction. So what is happening, beta? Whenever they both will bind, they will lead to the muscle contraction. This will lead to the muscle contraction, right, beta? So this is how this muscle can be contract and relax, contract and relax. Now the question comes: this actin binding site is not always very free. This is not always free. Why? Because our muscle should not always get the contract. So in relaxing condition, your muscle should not be in the contract position, right? It should be the relax. And for making the relax, this complex, this is actin, this is myosin. This should not be bind. And for not binding this, not allowing to binding, this area of actin should be covered by someone. And if this area should be covered by someone, this myosin cannot get inside of this actin. And this covering area is another protein. And this protein is actually your tropomyosin. This protein, beta, tropomyosin. So what is the function of tropomyosin? It will not allow for the muscle contraction. It will not allow for the muscle contraction. So now in this picture, as you can see, beta, in this picture, as you can see, this is your actin. This green portion is actin, and now this actin is having a binding site, right, beta? This binding site is completely covered by this area, this protein, and this protein is called as tropomyosin. This is your tropomyosin, and where it is present with the actin. Another thing, this dot are you can see, beta, these dots are present over here, and actually these dots are another protein called as troponin. It is called as troponin. Now, beta, we already know that's two ions. Already we have discussed about sodium and potassium, which we have already later on. Before that, we already talked about it. Now there is another ion which I already told you, that is calcium. Now the role comes of this calcium. So beta, for your body, these three ions are very much important, right? So now what happens when this tropomyosin will be get out from this binding site? Once, because if the tropomyosin will not getting out, what could be happen? This actin and myosin cannot be bind together, and muscle cannot be contract, right, beta? Now, when this muscle want to get contract, question comes in your mind. The muscle will get contract only when there will be the message from the brain. This brain will tell, yes, please contract. And how this message will tell? Why to this cell? How this message will tell to this cell? Now what happens, beta? Now the role comes of this calcium. What happens? This is actually your troponin. This troponin is having three binding sites. This troponin is having three binding sites like this. This is having three binding sites, one for troponin T, troponin I, and troponin C. So what happens, beta? First of all, this troponin should always remain with the actin. It should always remain in contact with the actin, as you can see in this picture, right? So this is attached with the help of this troponin I binding. So troponin I for attached with the actin. Then troponin C. This troponin is also should remain attached with the tropomyosin. And to attaching with this tropomyosin, there is another binding site for the troponin, that is troponin T. And now when this troponin will attach with the ion of the calcium, actually this calcium will comes only when there will be the signaling. I have told you about a secondary messenger. So whenever there is a signal will come, this calcium will attach on this troponin C, and once this calcium will attach this troponin C, this troponin will immediately sense, oh, might be this boss is telling me, this boss wants to this contract of this muscle, and once this calcium will bind with this troponin will remove this tropomyosin from the binding site, and once this troponin will be removed from this binding site, as you can see in the picture, beta, what is happening? This tropomyosin is removing, and this binding sites are now free, and this myosin is immediately attached with this actin, and there is having one. Over here, there is a one site for the ATP, and this ATP phosphorylation is occurs. Phosphorylation means this ATP will remove one phosphate, and this will become now a triphosphate to diphosphate, so ATP, and this will produce the energy. And this is how this myosin head will be tilt ahead like this. It will be tilt ahead like this, and this is how this muscle contraction will be happen. So I hope now it is clear. Now you know that how the muscle is contracting inside of your body, because due to this is actin and myosin. So this actin and myosin main the contractile protein. So this is called as contractile protein. Number two, that is troponin and tropomyosin. They are regulating this contraction, so they are called as regulatory protein. Another thing, beta, another thing. Also, as you can see in this picture, this Z line, suppose this is the basal lamina. So now this Z line should also attach with this basal lamina, and this is called as supporting proteins, and this supporting proteins is your desmin. Everything is written in your workbook. Don't worry. This is your desmin. So what is the desmin function? It attaches Z line to the basal lamina. Then this actin will also should attach with this basal lamina. This is your dystrophin, and this was the question, beta. This is your dystrophin. So what could be happen if dystrophin is not there? This muscle cannot be properly contract, and if this muscle cannot be properly contract, this person cannot do day-to-day activity, cannot properly stand up, cannot properly climb up, and this is how all these features can become up, right, beta? And this is a dystrophin. This is a supporting proteins, and we already know this is also everything is made up of the protein inside the body, and this dystrophin is also synthesizing by the cells, and why the cells is not producing this dystrophin due to some genetic disorder, and this is lead to the muscle dystrophy called as Duchenne muscular dystrophy. This is called as Duchenne muscular dystrophy. Now tell me, beta, one thing: what could be happen whenever the muscle contraction will be happen? This both Z line will become closer, and once this Z line will come closer, what could be happen, beta? What could be happen? This Z line comes closer, so this I band will also be decreased. Now what about this A band? This A band is having the head, beta. This A band is having the head. Now what is happening, beta? This is actually myosin, and this is the head. Only this head is moving. This hand is not moving. So now what will be the effect whenever the muscle will be contract? This A band will never change. This A band is always remain constant. Similarly, beta, this is called as your H zone. So what could be happen? This H zone might be decreased, and this H zone might be disappears, because both this actin can come closer, and once they will come closer, this H zone will be disappears. Okay. So this is how all these things are there. So now start writing down. This light band consisting of your actin, and this is your I band. Dark band is myosin. This is your A band. A band contains myosin filament, I actin, H band non-overlapping part of myosin, and M band is middle line of the H band. Coming to the next, that is the myofilaments. So protein in the skeletal cell, contractile protein is actin and myosin. Supporting protein desmin, dystrophin, titin. Relaxation protein is sarcopam. Later on we'll discuss about this, what is sarcopam. And regulatory protein is troponin and tropomyosin. Troponin and tropomyosin. This actin is thin filament, and this is a group of actin plus troponin plus tropomyosin. Actin plus troponin plus tropomyosin, and myosin is thick filament, and it is only the myosin contains. It contains only the myosin. Coming to the next part, this troponin is having 3 binding sites. Number 1, that is trop I, trop C, and trop T. Trop I for the actin, trop C for the calcium, trop T for the binding with the tropomyosin. Trop T with the tropomyosin. Other proteins are like actin, it's attached actin to the Z line, desmin Z line to sarcolemma, nebulin parallel to actin, and titin. This is the question. This is the strongest and hardest protein of your body, and it connects to M line to Z, myosin to Z line. This is the strongest and hardest protein. So question comes, strongest and hardest protein of the body, that is our titin. Then the dystrophin connect actin filament to the sarcolemma, and if this dystrophin is not there due to the destruction of dystrophin, this can lead to your Duchenne muscular dystrophy. This will lead to Duchenne muscular dystrophy. Suppose if this dystrophin is present but not properly functioning, so it means ये बेकार है, ये destruction जो हो रहा है इसका properly नहीं हो रहा है, complete नहीं हो रहा है, partially हो रहा है, so now it is also not functioning again properly. So if due to the partial destruction of dystrophin, this dystrophy is called as Baker's muscular dystrophy. So if the question is asked Duchenne muscular dystrophy, which protein deficient? Dystrophin. If the question is asked Baker's muscular dystrophy, which protein is deficient? That is your again partial destruction of the dystrophin, right? Coming to the next, how the skeletal muscle contracts. How the skeletal muscle will be contract. This skeletal muscle will be contract, just remember. Bas, थोड़ी देर बेटा, once we will complete the skeletal muscle contraction part, because now you will understand better. Then we'll take the lunch break, and after the lunch break, we'll finish all this muscle physiology, and we'll start with the GIT part. So I hope that till now you have understood all the things, and now you are started already assuming that how the body is functioning, because inside of the body, everything, the target area is that is cell, and inside of the cell, that is your nucleus. And now you already know only 3 things you are consuming on every day. That is number 1, protein, number 2, that is lipid, and number 3, that is carbohydrate. Now what happens? These 3 things you are consuming, these 3 things is breaking down, and they are making the raw material, and actually everything inside of the body is made up of only these 3 things. So now everything inside the body, most of the part, that is your protein. That is how you should understand. Okay, beta. So now coming to the skeletal muscle contraction. What happens? Suppose this is your skeletal myocytes. Skeletal myocyte, myocyte means muscle cell, myo means muscle, and cyte means cell, and this is skeletal muscle, so skeletal myocyte. In skeletal myocyte, beta, what happens? The layer of this myocyte is not like this plane. Actually, the layer of this myocyte is something like it is having a depression like this. It is having a like this depression completely everywhere. And actually, these depressions are present over there. So these depressions are into a transverse plane. This is into a transverse plane, and these depressions making a tube-like structure. As you can see, this is a tube-like structure. And this tube-like structure, we use the word tubules. What we use the word tubules? And these tubules are actually onto the transverse plane. So we use the word T-tubules. What we use the word T-tubule? So this is your T-tubules. Now what happens, beta? Inside of this T-tubules, there is a presence of one receptor. Because already you know, if somebody is coming outside of your room, there should be some bell. That via this bell you will understand that somebody is outside. So this is similarly in this T-tubules. What is happening? There is a presence of one receptor, and this receptor is called as a DHPR, dihydropyridine receptor. This is called as your DHPR, dihydropyridine receptor. Now what is happening? Inside of the cell there is something called, this is your sarcoplasmic endoplasmic reticulum. What we call this? Sarcoplasmic endoplasmic reticulum. And this sarcoplasmic endoplasmic reticulum is actually having calcium inside of this, and this is actually the warehouse of this calcium. So what is the function of this sarcoplasmic endoplasmic reticulum, beta? This is actually the warehouse of this calcium. So now there are a lot of calcium present. Now we know that what is the function of this calcium, beta? So don't even just don't think that calcium is only required for your bone. That calcium is required only for your bone formation. Calcium is not only for your bone. Calcium is also required for your muscle contraction. So if there is no calcium, there will be no muscle contraction. And remember, whenever there is a patient will get the MI, heart attack, what you do? First you will give the adrenaline because you want to stimulate via this nerve, this heart. And once for contraction of this muscle, you introduce again a calcium gluconate injection. Why you are giving the calcium gluconate? Because now you know for muscle contraction how this muscle can be pumped up when it will be contract. And for contraction, which iron is needed? The calcium ion is needed. So that is why from outside you always give this calcium gluconate injection. So now what happens? This calcium is more number over here. What happens, beta? This is sarcoplasmic reticulum, and actually this is the warehouse of your calcium. And again, this warehouse is not having this plane channel. This is also having, beta, this type of tubules over here. And now these tubules are into a longitudinal plane, so called as L tubules. This is called as L tubules. And again, one more receptor will be there, and this receptor is called as RYR receptor. This is your RYR receptor, and called as ryanodine receptor. This is called as ryanodine receptor. Now what happens, beta? Whenever there is action potential comes, so this is action potential means signaling from the neuron, and whenever this action potential comes, this action potential enters into this DH, into this T tubules, and in T tubules, which receptor is there? DHPR is there. So now what will happen? This action potential actually stimulate this DHPR, and once this DHPR will be stimulated, this DHPR is mechanically connected with this ryanodine receptor, and this ryanodine receptor will also activate. And once this ryanodine receptor will be activated, beta, what will happen? This ryanodine receptor will immediately sense, oh, might be there is this message is coming from the brain, so might be this DHPR is activated, and this DHPR is also activating this ryanodine receptor, and this ryanodine receptor will be open. And now, beta, this ryanodine receptor will be open. What will happen? This calcium will get out from the here. Why? Again from high to low. High to low. So this high to low calcium will move out, and once this calcium will moving outside, where it will go? It will bind with the troponin C. And once this troponin C will bind with this calcium, this troponin C will immediately remove this tropomyosin. This tropomyosin will remove from this actin, and this actin is now having an open binding site, and immediately this myosin head will get attached with this actin, and it will start doing this contraction. And this is how this muscle contraction happens. So now you understood Action potential is coming. This is activating DHPR. This DHPR activating ryanodine receptor, and this once is open, this calcium is getting out, and this is all doing the contraction. Now the contraction is over. Now we don't want to more contraction. So what ideally we should do? All the calcium which is outside of this sarcoplasmic reticulum, this calcium should send back into the sarcoplasmic reticulum. Because who was the culprit for making this contraction? This calcium ion. So this calcium ion should ideally send back. And now this area is having low, and this area is having high calcium concentration. So now you are sending from low to high. You require the ATP, you require the energy, and you require the pump. From downside to high side, you are sending the water. You require the pump, and this pump is called as SARCA pump. Why it is SARCA pump? SARCA stands for sarcoplasmic endoplasmic reticulum calcium ATPase pump. Means you are sending into the sarcoplasmic endoplasmic reticulum, you are sending to the calcium with the help of ATP. So why are you using the word SARCA? So that is why in the contractile protein beta, I have told you the SARCA pump is a in the relaxation protein I told you that SARCA pump is a relaxation protein. And why? Because this SARCA pump is reverse back, or this SARCA pump will send back all this calcium into this sarcoplasmic endoplasmic reticulum, and muscle contraction will be stops. So now write it down, beta. One more thing, just listen over here. Now what happens, beta? In cardiac muscle, this ryanodine receptor is absent. In cardiac muscle, the ryanodine receptor is absent. So now what could be happen, beta? Now what could be happen? How this ryanodine receptor will be activated? Because there is no DHPR, so DHPR is absent. How this ryanodine receptor will be activated? Now what happens, beta? There is a channel. There is a channel, and these channels are for the calcium. Now what happens? Whenever this action potential comes, these channel will be open, and these channel will be open. This calcium will start moving inside, but this calcium is not much enough for making the muscle contraction. But this calcium is getting inside. Once this calcium is getting inside, what happens, beta? This ryanodine receptor will immediately sense, I didn't open. From where this calcium is coming inside, and now this ryanodine receptor will immediately think, oh, might be this boss is telling. Due to this boss, might be this calcium is getting inside, and this is the signaling for me. And this ryanodine receptor will be open. And what will be happen, beta? This ryanodine receptor once will be open, this calcium will get out, and again the muscle contraction will be happen. So now what is happening in the cardiac muscle, beta? In cardiac muscle, DHPR is absent, so calcium is getting entry, and this will leading to open this ryanodine receptor. So this is something called a phenomena called as, due to this calcium is getting out. So this we call that, beta, calcium-induced calcium phenomena. Calcium-induced calcium phenomena. And this calcium-induced calcium phenomenon is seen in which muscle, beta? In cardiac muscle. In cardiac muscle. Now what could be happen? Suppose if this ryanodine receptor is getting overactivated due to the mutation, there is some mutation, and this ryanodine receptor now, beta, overactivated. Now this ryanodine receptor is functioning its own. It is not listening to the boss. And now what could be happen, beta? This ryanodine receptor will open itself only, and what could be happen? This will lead to the release of this calcium, and muscle contraction will be happen. And beta, you know that when there is a more and more muscle contraction, every time muscle contraction will increase the level of temperature, hyperthermia. And due to this malignancy, due to this genetic defect, this ryanodine receptor is getting open itself only. This can lead to the malignant hyperthermia. So why the malignant hyperthermia? Due to the overactivity of the ryanodine receptor. So beta, ryanodine receptor responsible for calcium release. Ryanodine receptor activated by DHPR, dihydropyridine receptor in skeletal muscle, and ryanodine receptor activated by calcium in heart muscle, called as calcium-induced calcium phenomena. If ryanodine receptor is overactivated, this causes increased temperature, called as malignant hyperthermia. Treatment of this, you just block this ryanodine receptor, and the treatment of this can be done via dantrolene sodium. So dihydropyridine receptor, beta, DHPR responsible for mechanical interaction with ryanodine receptor. And what will be happening, beta? When this muscle will be contract, so this actin will bind with this myosin. So beta, it is something like a bridge formation. And why this bridge formation is happening? This bridge formation is happening due to this myosin head, beta, and this is called a sliding filament theory. So whenever there will be the muscle contact, this Z line comes closer, length of H and I band will be decreased, or H band disappears, MCQ. Length of A band remain constant. Always remember, beta, remain constant. This is always remain constant. And calcium releases from sarcoplasmic endoplasmic reticulum. Right, beta? And the muscle will be relaxed. This calcium ion goes back into the sarcoplasmic endoplasmic reticulum via SARCA pump. And this is the cross bridge formation. Why the cross bridge formation? Attachment of myosin head to the actin filament. In the rigor mortis, beta, you already know the rigor mortis seen into the death, and this is due to the absence of ATP. Because there is no ATP, and this myosin head is remain attached with this myosin actin filament, and that is lead to the muscle into the spasm condition. This is called as rigor mortis. So beta, this is all for this all for till this break. And now we'll take the lunch break, but I hope that now you have cleared very much about the skeletal muscles. So if you'll find any questions, in the end I will give you my WhatsApp number. So in WhatsApp number we can connect with each other, beta, and just have a break now, lunch break, and lunch break will take lunch break for 45 minutes, beta. Lunch break for 45 minutes. So it's 2:25 now. It's 2:25. So we'll start the class at around 3:15 p.m. So we'll start the class at 3:15. Okay, beta. Thank you very much. So guys, welcome back to after the break. So let's start with the next topic, that is the property of skeletal muscle. So I hope that all of you had your lunch. Yes, guys. Please tell me, beta, in the break whether my speed is okay or not, or are you okay with my speed or with my pace? I hope so that all of you are getting the points and you are understanding well this physio. So till now we have discussed from the cell, then we have discussed about the body fluids, and then we have entered into the muscle physiology. So before the break, we have talked about that skeletal muscle, how the skeletal muscle actually functions, and we have seen in the skeletal muscle actually the ion which is responsible for the muscle contraction, that is the calcium, isn't it, beta? So now you know that about the sodium, potassium, and calcium. So what is the role of sodium, potassium to forming the action potential inside the body? And where is the potassium is mostly lives inside the body? That is more in ICF, intracellular fluid, and sodium is more is extracellularly. And also we have seen the calcium, how the calcium regulates the muscle contraction. So in skeletal muscle we have seen that there is a T tubules. In the T tubules, there is a DHPR receptor, and there is a sarcoplasmic reticulum. There is the L tubules. There is the ryanodine receptor, and in this ryanodine receptors actually mechanically interact with this DHPR. And once the axial potential reaches up to DHPR, this will activate this ryanodine receptor, and calcium is leaking out and going up to the troponin C. This troponin C, when binds with this calcium, this troponin will remove this tropomyosin from the actin binding site. And once this binding site is free, this myosin comes and attached with this actin. Okay, and this is how the muscle contraction can be happen. So for understanding this property of the skeletal muscle, we have understood that first of all there are two things. Number one, excitability. Number two is contractility. Because first of all you have to excite the muscle, isn't it? And once you will excite the muscle, it will start contracting. And this is how this property you will understand. So what happens, beta? To excite any muscle, you require the stimulus, isn't it? For that. To excite any muscle, you require the stimulus, and this stimulus can be different type. Means there can be low grade current, there can be higher grade current, there can be mid grade current. So similarly, that's current which you are giving to this muscle. This is for how much time you are giving. So this is how we actually count or how we access this actually excitability of this muscle. And the muscle excitability we can be done via two things. This method is called as strength duration curve. What we call this? This is called as strength duration curve. Remember beta one thing, strength duration curve. Remember one thing. What is excitability? Excitability is nothing. This is the reaction of tissue to stimulus. For excitation of tissue, stimulus has to give beta, right? And this, there are two words which we are going to discuss about. Number one, beta, Rio base. Number two is chronaxy. Number one is Rio base, and number two is chronaxy. But it might be these words you are hearing first time in your life, but every time, many times, our question is asked from these two words. What is the Rio base? What is the chronaxy? So remember one thing. Whenever you just want to check this excitability of the muscle, all right? So you are giving the current. This current actually you are denoting with the name strength. So strength means nothing. This is the current. And what is the duration? The time interval, the time which you are giving this current to this muscle. So this is called a strength duration curve. Actually, this strength we use the word Rio base. So what is the meaning of Rio base? Rio base is nothing. The current which you are giving to this muscle. This for this current we use the word Rio base. So this is a synonym of this. So Rio base is nothing. It is a current minimum strength, right? Now what happens? Suppose you are giving a minimum amount of current and giving for a particular time period. So this time period, after some times, what you have seen, this muscle gets excited. Once this muscle gets excited, so the time period which was taken by this muscle to getting the excite, this time period is actually called as duration time. What we call this? Duration time. Now what happens? The minimum current which you are giving over here, for the current we use the word Rio base. So this is beta your Rio base, right? So Rio base is nothing, it is just a current. What happens? When you double the current, means earlier you were giving the 1 millivolt, right beta? And suppose it was taking the 10 second at the 1 millivolt current, what could be happen? When you increase the current or you are doubling the current, means now you are giving a 2 millivolt current. So this is Rio base you are doubling. So this is Rio base x2. This means that you are increasing the current. Once you will increase the current, what about the time? Earlier it was taking the 10 second. Now it will take half time because you have increased the current. And this half time actually we use the word chronaxy. So this half time is nothing, it is the chronaxy. So this is how you should understand Rio base and chronaxy. In your exam, they can ask you the definition of this Rio base and chronaxy. Either they can ask you the muscle excitability is best measured by which method, and this is the answer: strength duration curve. This is your strength duration curve, right? So this is how you should understand, right? Now coming to the definition. Rio base means minimum strength of stimulus which can excite the tissue. Rio base means minimum strength, utilization time, minimum time required, that is duration time, and chronaxy is minimum time required for double the Rio basic strength. This is the definition. So you just remember these words, okay? Next is contractility. So once the muscle gets excited, after that what happens? Muscle gets contract. So contract is nothing. This is the response of muscle to stimulus. And one contraction is there. Means how the contraction is there? This contraction is there like this. This is a contraction, then relaxation. So this is called as twitch. What we call this? Twitch. So this is called as one twitch. So this period is one twitch. Now this contract can be two types. First of all, if I'm holding this remote like this, right? Now what is happening? My muscle is contracting or not? Yes, my muscle is contracting. But right now the length of this muscle is same. Length of this muscle is same. But the tone of the muscle is changed. So now what is happening? If length is same, for same we use the word iso, and for length we use the word meter. So this is an isometric contraction. And what could be happen if I will do this arm like this? Now what is happening? Tone is same, but length is changed. So this is called as isotonic contraction. Now for understanding this thing, first of all you should understand the total twitch period is 0.1 second. So muscle will take only 0.1 second to first contract and relax. So this is called as total twitch period. What happens? First of all, you are giving this stimulus. So this is the point of stimulus, right? After this, sometimes this muscle get contracts. So this is called as point of contraction. PC stands for point of contraction. And the time period between taken by this is called as latent period. This is your latent period. PS stands for point of stimulus, point of contraction. After this muscle gets contract. So this is called your contraction phase. After this is the point of maximum contraction. Then it will relax. So this is relaxation phase, point of maximum relaxation. And after that, it is again into the polarized state. So this is how your muscle gets contracts and everything. So the latent period is exists around 0.01 second, contraction for 0.04, and relaxation for 0.05. So total twitch period is 0.10 second. This is how simply you can understand. The next question comes. The next topic, that is refractive period. But what is the refractive period? This is very important because question is asked in your exam every time from this. So this is quite important. Frequently asked question. The refractive period. Refractive period is nothing. If your muscle started to contracting, suppose this is your muscle. It started contracting like this. What could be happen if in between at this point, if you will give any other stimulus to this muscle? What could be happen if you will give any other stimulus to this muscle? This muscle will not show any response. So what is the refractive period? During the one twitching period, if you will give any stimulus in this mean period, this muscle never show any response. This is called as refractive period. So what is refractive period? Refractive period का मतलब है कि मसल अगर एक बार कॉन्ट्रैक्ट हो रही है तो उस पूरे पीरियड के अंदर मसल दोबारा कॉन्ट्रैक्ट नहीं हो सकती है जब तक कि पहले वाला कॉन्ट्रैक्ट खत्म ना हो जाए. This is called as refractive period, right बेटा? Now this refractive period is divided into two types. Number one, absolute refractive period, and number two, relative refractive period. So this period, question comes from where to where this absolute period is there. So better remember, when this contraction starts from here up to the 1/3 period of the relaxation. up to the 1/3 period of the relaxation. This phase is called as your absolute refractive period. Means in this condition, at any point of view, means whatever the current you will increase, this muscle will never show any response. From where? From starting from this point up to this point, right? So this is called as absolute refractive period. But what happens from 1/3 part of this relaxation period up to this downwards area? This area, if you will increase the strength, if you will increase the current, this area can show the stimulus. This area can show the response. So this is called as relative refractive period. So this is relative refractive period. Absolute refractive period, no response at all. Or relative refractive period is few response when strength increased. Few response when strength increased. Coming to the next, that is all or none law. But all or none law is nothing. Whether this muscle or whether this neurons will show the stimulus completely from one end to the other end, either it will not show any response. So this is called as all or none law. It is not something like that. Means suppose this is your neuron, this is your neuron, right? So if stimulus is started from here, it is 100% it will come up to the other end. It will not going like this. Means this stimulus is started from here or it can end over here. It is not shown like this. So this is called as all or none law, right, brother? So this is the graph. In this graph, just remember one thing. This is the graph. In this graph, I already told you, brothers, that resting moment potential is always negative. So this is minus 70. What is happening now? Now what is happening? Axial potential is generating. So how the axial potential is generating, brother? Now it is becoming the positivity. So this is called as your threshold value. Means up to this level, the positivity will be slower. But once this level is reached, this positivity is going to be very fast. This positivity is going to be very fast. And this is how this axial potential is happens. So now the question comes, what happens, brother? Why this negativity is becoming the positivity? As you can see, it is coming to the plus 30. Due to which ion? Which ion is getting inside right now? So you already know that the sodium influx or efflux. Right now, sodium influx is happening. So this is in this phase, sodium influx is there. After some times, at this maximum point of action potential, what happens? This sodium channel will be closed. Once the sodium channel will be closed, which ion channel will be open? So now this will again become into the negative. And now at this point, which ion will be getting out? This is potassium channel and potassium efflux. Here is potassium efflux. So now just remember these 2 points. Number 1, that is sodium. This is sodium influx and this phase, and this is potassium efflux in this phase. Right now, ideally, what is happening? Here you can see it is going more negative. This is going more negative, and this more negativity is actually called as hyperpolarization. Hyperpolarization. But this phase is called as depolarization, and this phase is called as repolarization. This is called as repolarization. But remember, so this phase is called as depolarization, and this is called as repolarization. And this more negativity period, as you can see, ideally it should stop over here, but it is not stop over here, and it is going further down, and then it is coming to the normal resting potential. So this stage is called as hyperpolarization. And question comes, why this hyperpolarization is happening? What happens during this phase? During this phase, what is happening? During this phase, this potassium is getting out from the cell continuously because the gate is open. What happened? When this minus 70 is achieved, ideally this gate should be immediately closed like this, but this gate never closed like this. What happens? This gate actually closed very slowly, and due to the slow closure, still the more and more potassium getting out. And once the more and more potassium getting out, it will lead to more and more negativity. And this is condition called as hyperpolarization. So this hyperpolarization is due to slow closure of potassium channel. Slow closure of potassium channel. So this is how you should understand it. Coming to the next, that is the muscle fiber. So inside of your body, there are total 3 types of muscle fiber. 3 types of muscle fiber. Number 1, that is type 1, type 2A, and type 2B. As you already know, inside the blood, this is the blood vessel, this is your cell. What happens? Inside of your blood, there is a hemoglobin. With this, oxygen is binding. That is why the color of this blood is red. And now this oxygen will be delivered into the cell because this oxygen is required at the level of cell. Now what happens? When this oxygen delivers, this oxygen should be bind with someone in the muscle. And here is the function of myoglobin. So what is the function of this myoglobin? Myoglobin function is nothing. It is actually binds with the oxygen. And now question comes, how many molecules of myoglobin, how many oxygen binds with the myoglobin? Beta? So always remember only one oxygen molecule binds with the myoglobin. And due to this myoglobin, the color of muscle becomes the red. Okay, beta? So type 1, there is a myoglobin is present. And that is why oxygen storage is present. Type 2A, again the same story. And type 2B, there is no myoglobin, so no oxygen storage. And that is why due to the storage of oxygen, this is color in red, and this is in white. This contraction is slow. They are fast, and they are also fast. Size of this nerve fiber is small, muscle fiber. That is small, that is large. Again, this is large, right? This is fatigue resistant due to the presence of oxygen. Same story. And they are more prone to the fatigue because no oxygen is present over there. Now the function is, this is for standing. So that is why you can stand for a longer time. This is for walking or slow running. And this is for fast running or sprinting, right, beta? So this is how you cannot run faster for a longer time, but you can stand for a longer time. Yes, beta, due to the presence of this myoglobin, due to the presence of oxygen. So this is all story about the skeletal muscle. Coming to the cardiac muscle. In this cardiac muscle, beta, remember one thing. In the cardiac muscle, this is an involuntary muscle, and this cardiac muscle, always remember, cannot be titanized. This cannot be titanized. And this cardiac muscle is made up of actin. Again, this is not myosin, but this is myosin. Then troponin, tropomyosin, and titin. Titin is the hardest and strongest protein of the body. And always remember, cardiac muscle is a functional syncytium. What is the meaning of functional syncytium, beta? Suppose this is my one hand, right? If I am raising my one hand like this, ideally this hand should also be come up, isn't it? Have you seen that toy? खिलौना देखा है आपने? जिसमें वो एक हाथ उठाता है तो उसके दोनों हाथ उठ जाते हैं. So what happening in this limb? If you are raising one hand, or another hand is not moving. So this is not a functional syncytium. But what happens in the heart, beta? In the heart, this is the chambers, right? This is the chambers. So these both chambers, they are contact together, and these are contact together. So what is happening, beta? Both atria contacts together, and both ventricle contacts together, and this is called as functional syncytium. What we call this? Functional syncytium. We already talked about that this shows calcium-induced calcium release phenomena, and there is a well-developed sarcoplasmic reticulum. There is something called Frank-Starling law, but very important for your exam purpose. What is the Frank-Starling law? This is nothing. What happens? Frank-Starling law says whenever you increase the length of the muscle fiber, what is happening? This will increase the muscle contractility. Have you seen the rubber band. If you will stretch the rubber band as much as possible, and what could be happen, if you will release this hand like this rubber band is stretched, contracts very fast, and it hurts on your another hand. So this is similarly, this is actually the Frank-Starling law. So whenever you will increase the muscle length fiber, this will increase the power of contractility. So this is called as Frank-Starling law, and this Frank-Starling law is for measuring the cardiac output. This measures cardiac output. Coming to the next, that is smooth muscle. In smooth muscle, this is the involuntary muscle. Make the change. This is no visible striations. These smooth muscles are present everywhere inside of your peritoneum cavity. So there is the interstitial cell of Cahal. There is a pacemaker called as interstitial cell of Cahal. Now what happens, beta? Inside of this smooth muscle, there is no sarcoplasmic reticulum. There is no sarcoplasmic reticulum. It means no sarcoplasmic reticulum. This is the sarcoplasmic reticulum. It means if this is absent, calcium is absent, and without calcium, how the muscle can be contracted? So now what happens? In smooth muscles, this calcium actually comes from the outside. And once this calcium comes from the outside, it binds with the protein, MCQ protein name, called as calmodulin. Beta, yaad rakhna, calmodulin. And this makes the complex of calcium calmodulin, and it will activate the myosin light chain kinase, and this will lead to phosphorylation of the myosin, activation of ATPase, and muscular contraction is happening. So this is how your muscle contracts. Next is maintenance of muscle tone. But maintenance of muscle tone, skeletal muscle, always remember, this is always your neurogenic. This is a neurogenic control. Later on we'll discuss about this gamma motor neuron in anterior gray horn. Cardiac muscle is again myogenic, and smooth muscle is also myogenic. Now the electrical changes in muscles. But always remember, in electrical changes in the muscle, the resting condition is called as polarized state, which we already discussed about. Polarized state, and this is due to ion balance. What happening during the resting condition? But this is the cell. Suppose this is cell. So there is a potassium, lot of potassium outside, sodium, and also calcium. Now what happening? Now what is happening, beta? Normal condition. In normal condition, this potassium can leak outside, because this potassium can go only from inside to out, because outside there is a low concentration, inside there is a high. So what could be the effect if the potassium is getting out? It will become more negative. So no problem with the cell. So cell says no problem, man. You can go outside. It will lead to more negativity. I can become the more resting condition. But what could be happen if sodium gets entry and calcium will get entry? It will excite the cell. So that is why in a resting condition, nil permeability for sodium and calcium, but potassium moves freely in and out of the cell. MCQ. Which ion moves freely in and out of the cell? That is potassium. So in resting condition, as already you know, in resting condition, ICF is always negative. So this is negativity, and ECF is positivity. The resting membrane potential in different tissue, that is in neuron minus 70 millivolt, in skeletal muscle minus 90 millivolt, smooth muscle minus 50, rodent core minus 30, and MCQ RBC is minus 12 millivolt. Minus 12 millivolt. Now coming to the next beta. This anion means negative charge ion, cation means positive charge, depolarization due to influx of sodium ion, hyperpolarization due to slow closure of potassium channel, and reestablishment by the sodium potassium ATPase pump. Immediately tell me beta, what is happening in the sodium potassium ATPase pump? Who was influxing, who was effluxing, and how much? So you know that potassium was going outside in the pump. So this is sodium efflux, and how many sodium? 3 sodium, and potassium coming in. So this is potassium influx, and how many potassium? 2 potassium with the help of 1 ATP. So till now we have discussed about the primary functions of the ion. Sodium is more in ECF, which is depolarization, passes through sodium channel. Potassium is more in ICF, maintain RMP, initiate repolarization, passes through potassium channel. Calcium more in ECF, contractility of muscles, conductivity of repolarization, and secretion in gland. That is exocytosis, beta. That is processes exocytosis, which we already talked about. And chloride in more in ECF and passes through chloride channel. Now coming to the next beta, that is called as NMJ, neuromuscular junctions. What is the neuromuscular junctions? In neuromuscular junctions, beta, there is nothing. This is how, as you already know, this brain actually controls all the muscle. So now this brain is controlled by 2 things. Number 1, that is nerves. Number 2, that is hormones, right? Chemical. So now, as you can see in this photo, this is the bone, and with this bone, this attachment is tendon, and then this is the muscle, and over this muscle, there is coming up this nerve fiber, and this nerve fiber is coming over here, and this is the axon part, which you can see. This is a myelinated portion, which is having more lipid. And now what is happening, beta? This is the endings, different endings, and basis of this one ending, if we will do the zoom out, this is called as synapse. What we call this? Synapse. So this is how it looks like. So this is your cell membrane, and this is your axon part of this neuron. Right? Now what is happening, beta? In this neuron, always remember what happens, beta. Whenever understanding for this, what happens? Suppose this is your neuron. This is how your neuron looks like. What happens? Or in your neuron, there is a presence of something called as knee cell granule over here. So these are the knee cell granules, right? Function of this knee cell granules is protein synthesis. And the protein it is synthesizing, that is the neurotransmitters. What is the neurotransmitter? Now the question comes. But a neurotransmitter is nothing. Actually, what is happening? When this neuron is sending all the information over here, this information must be passed from here, and then it should start from the next part. So this information passing through this area via a chemical, and this chemical is called as neurotransmitter. Right? But now what happens? Whenever these knee cell granules make synthesis of this protein neurotransmitter, they will pack it into a vesicle, and these vesicles come down and it starts dropping over here. Now if you remember, this is a type of exocytosis where there is a neurotransmitter in the nerve. I have already told you, beta. Now what happens? Always remember. Also I told you there is a myelin sheath like this. Myelin sheath is there, and sodium gets entry over here, and action potential reaches over here. Now what happens, beta? Look at on the diagram. What happens when this axon terminal sending all the action potential up to here? So this exocytosis is calcium dependent. I already told you this exocytosis is calcium dependent. Now what happens when this axon part getting this action potential over here? This calcium channel is open. This calcium channel is opens, and what could be happening, beta, when this calcium channel is opens? This calcium from outside getting inside of the cell. Once it will get inside, what is happening, beta? Here are the bag neurotransmitter bags are present, and now these bags will start attaching with the cell membrane. And what is happening, beta, when they are attaching with the membrane, there is a lysis of this membrane like this. Can you see over here? And what is happening? All these chemicals is getting out into the synaptic area. What is happening, beta? As you can see, this is the muscle layer, and this muscle layer is having like this appearance. And I already told you, beta, these muscles is having PTBs, DHPR. And now what is happening, beta? When this acetylcholine, this neurotransmitter is acetylcholine, beta. This acetylcholine comes over here. Here is a actually receptor present over here like this. Receptor is present over here like this. Now what happens, beta? When this receptor binds with this neurotransmitter, when this neurotransmitter binds over here, this will immediately, because inside there is already negativity and we want to excite this area. So what will happen when this neurotransmitter binds with this receptor will immediately open the channels, and these channels who will come from the outside now for making the positivity, which ion? That is sodium. So now sodium will start entering inside, and this will become the positivity, and this will lead to the action potential into the muscle. So this is how this neurotransmitter actually functions, and this is how this neuromuscular junction is functions. Now there are 5 things. What could be happen, beta? What could be happen if this calcium is not getting inside? If this calcium is not getting inside, this is due to the mutation, and this disease is called as Lambert-Eaton syndrome. Lambert-Eaton syndrome. So what will be happening in the Lambert-Eaton syndrome? In Lambert-Eaton syndrome, there is a defect in the calcium channel, and this defect, especially the fast calcium channel, slow calcium channel, still they are working, but they are allowing only very slowly all the calcium to get inside. So that is why the acetylcholine which will be released at the synapse area, this is also very slow, right? Beta? Now what could be happen next? Suppose this calcium is okay. Now these vesicles, they cannot attach to this membrane, and this is due to the botulism toxin. So botulism toxin will not allow to bind this neurotransmitter with this presynaptic membrane. This is the beta synapse, so this is called as presynaptic area, and this is called as your postsynaptic area, right? Presynaptic and postsynaptic. Number 3 thing, suppose if this neurotransmitter is releasing more and more due to the toxin, again the tetanus, and this tetanus lead to more and more releasing of this condition, and that is why this will excite more and more muscle, and that is why in the tetanus there is a muscular spasm. Number 4, suppose if the receptor is not functioning well, the receptor which is present over here, this is not functioning well. Now what could be happening? What happens when this acetylcholine is released? There is something called acetylcholine esterase. This acetylcholine esterase actually degraded all the remaining acetylcholine from here and allowed to only few acetylcholine binds with this receptor. Now what could be happening when this receptor is gone? Now this acetylcholine cannot bind with anyone, and once it cannot bind with anyone, can the sodium get entry inside? No. And this condition is called as myasthenia gravis. So in myasthenia gravis, there is nothing. There is a receptor is defective at postsynaptic membrane, hence no neurotransmitter can be bind up this, and this condition is called as myasthenia gravis. Now what was happening? You cannot produce the new transmitter, or you cannot produce the new receptor inside the body because you are not having any rights to formation inside the body. So now what could you do? You can do the decreased activity of this acetylcholine esterase, and that is why the drug of choice for myasthenia gravis is acetylcholine esterase inhibitor, and this inhibitor is your neostigmine, and that is why the drug of choice in myasthenia gravis is neostigmine. Number 5, suppose there is some venom. This venom is coming from the snake, and this snake is actually bite to you, and due to the bite of the snake, what is happening? This venom reaches up to the synapse, and this will again this dysfunction of this receptor, and again condition can be happen with the myasthenia gravis. So this is called as cobra venom. Which one, beta? This is your cobra venom. Again, the drug of choice for cobra venom is, again you can use this condition. So this is again neostigmine is cobra venom, right? So I hope, beta, this much is clear to you. This much is clear. All the diseases which you have discussed, this is clear, beta. So this synaptic cleft is having acetylcholine esterase. Subneural cleft is postsynaptic membrane contains the receptor, right? Now how the muscle contraction was happening, beta? First of all, the motor nerve fiber action potential was coming. At axon terminal, what was happening, beta? Opening of voltage gated calcium channel. And remember, beta, there are 2 things. Number 1 is voltage gated, and number 2 is ligand gated. Beta, any gate, if it will be open due to the action potential, due to the current, for the current we use the word voltage gated. But if any gate for any ion is open due to the chemical, this is called as ligand gated. What is happening over here? The sodium gate is ligand gated. So opening of voltage gated entry of calcium from ECF, opening of vesicle release of ACH. Then the postsynaptic membrane binding it is with the receptor, opening the ligand gated sodium channel, entry of sodium from again ECF, development of end plate potential, and this will lead to muscular contraction. So this is how all this muscular contraction happens inside of your body. This is how I hope that you can easily understand all these things, right? Now next thing, do some exercises questions inside of this. Functional unit of the skeletal muscle. Who is the functional unit of the skeletal muscle? Tell me. Functional unit of the skeletal muscle, that is sarcomere. All are component of thin filament in skeletal muscle except that is your myosin. Myosin always lives alone. Number 3 is in skeletal muscle, actin attached with the sarcolemma with the help of protein, that is your dystrophin. When skeletal muscle contracts, all things can be happen except but A band is always remain constant. So this is wrong answer. Minimum strength for strength we use the word Rio base. So this is your Rio base. So this is how you should identify each and every question, बेटा. Do the practice exercises every time. हमेशा क्वेश्चन सॉल्व करने हैं. हमेशा आपको जब भी आप उस चीज को पढ़ रहे हो बेटा, तो आपको सेम डे उस चीज का रिवीजन करना है, उसके क्वेश्चंस लगाने हैं, तभी आपको चीज समझ पाएंगे. अदरवाइज वो चीज आप कभी भी नहीं समझ पाएंगे बेटा, क्योंकि हो सकता है कि अभी तो दिमाग में आपके वो फिट बैठ जाएगी, बट लेटर ऑन व्हाट कुड बी हैपन बेटा, यू विल फॉरगेट दोस थिंग्स, एंड दैट इज व्हाई द रिवीजन इज की सक्सेस. दिस इज द की फॉर पासिंग दिस FMG exam, not only the FMG exam, even any exam. So that is why revision is most important factor. Please do revise on daily basis. Please don't skip every single day because exam is very nearby to you guys. Till now date is not confirmed, but definitely the date will come out very soon. So please focus on your study बेटा. लगातार पढ़ते जाओ. ये मत सोचो कि एग्जाम की डेट कब आएगी, क्या होएगा, कैसे होएगा, क्योंकि अपना काम वो नहीं है. अपना काम क्या है? अपना काम है भी अपने आप को प्रिपेयर करना. डेट का क्या होना है, क्या नहीं होना है, ये एनबी वालों के ऊपर है बेटा. तो क्यों ना अपन ये चीज एनबी वालों पे ही छोड़ दे. जो उनकी टेंशन है वो उनको लेने दे, जो अपनी टेंशन है वो अपन ले. और अगर आपने इतना सा कर लिया ना बेटा, तो आपका एग्जाम हमेशा पॉजिटिव ही आएगा. अगर आपने ये चीज कर ली, डेफिनेटली विल पास दिस एग्जाम बेटा. बट अभी आप जैसे घर पे हैं बेटा, और घर पे ही क्या होता है कि अपने बहुत सारे रिश्तेदार, कई बार घर के काम, कभी वो कभी ये, बेटा अभी वो टाइम आपका बिल्कुल भी नहीं है। अभी क्या करना है आपने कि आपको अभी पूरी पढ़ाई के ऊपर फोकस करना है, सारे लाइफ से डिस्ट्रक्शन हटा देना है और अभी सिर्फ और सिर्फ आपको अपने लिए टाइम देना है बेटा, क्योंकि ये एग्जाम आपका लाइफ मेकिंग एग्जाम है। नेवर एवर थिंक कि ये एग्जाम आप जिंदगी के अंदर पहला है और आखिरी है, ऐसा नहीं है बेटा। आप ऐसे एग्जाम ऑलरेडी पहले देके आ चुके हो, लाइक 10th बोर्ड 12th बोर्ड, बेटा आपको पता था आपकी कॉपीज कहां चेक होंगी, आपके लिए पेपर कहां बनेगा, आपका सेंटर कहां आएगा, बट स्टिल यू ऑलवेज वाज वेरी कॉन्फिडेंट कि यस सर आई विल गेट अराउंड 60%, 70%, 80%, 90%। व्हाई? बिकॉज आप अपने आप में इतने कॉन्फिडेंट हुआ करते थे क्योंकि आपने उस हिसाब से प्रिपरेशन करी हुई थी। यहां पर भी बेटा वही है, एग्जाम के अंदर क्वेश्चन आपके सामने है, आंसर आपके सामने है, आपने इतनी प्रिपरेशन करके जानी है बेटा कि आपको उस चीज का दोनों का मैथ समझ में आने लगे। अगर आपने बस इतना सा कर दिया, आप एफएमजी पास हो जाओगे। एंड बेटा ये एफएमजी कोई इतनी बड़ी तोप नहीं है एग्जाम में दुनिया में, ये सिर्फ एक एग्जाम ही तो है बेटा। लोग यूएसएमएल निकाल लिया करते हैं, तो क्या अपन एफएमजी निकाल सकते हैं? अपन जरूर निकालेंगे और इस एग्जाम को फोड़ेंगे, निकालेंगे नहीं बेटा, आप इस एग्जाम को फोड़ोगे अगर बशर्ते आपने इस हर चीज को बेसिक से पढ़ लिया, उसके कॉन्सेप्ट को अपने दिमाग में डाल लिया, आपका काम हो गया। क्योंकि जो भी चीज है एग्जामिनर जो भी पूछ रहा है बेटा, आज से 100 साल पहले भी अपन ऐसे ही दिखते थे और आज से 100 साल बाद भी अपन ऐसे ही दिखेंगे। सोच के देखो इन 300 सालों में कितने तरीके के पेपर हो जाएंगे, तो क्या चीजें बदल जाएगी? तब भी डीएचपीआर ही रहेगा, राइनोडिन ही रहेगा, कैल्शियम ही रहेगा, सोडियम पोटेशियम भी रहेंगे। देन हाउ द एग्जामिनर विल आस्क यू बेटा, he will ask you with change of the language. So now you have to understand, or now you have to get the language of that question, then only you can able to answer that question. So I hope that you are getting my points, and this is my personal advice to you guys. Please be always positive, right? Be confident with yourself, try to motivate yourself on day to day basis, because you are at home, so no one is there with you right now beta. So that is why please study alone. Then if you are studying alone, then please be honest with yourself. अगर आप पढ़ रहे हो तो बेटा पढ़ना ही है, ऐसा नहीं करना है कि पढ़ाई के नाम पे आपने मोबाइल तो चला लिया लेकिन आप उसको देख नहीं रहे, राइट बेटा? सो पढ़ना है, अच्छे से करना है और पास होना है, और पास हो भी जाएंगे बेटा, दैट इज माय गारंटी, राइट? सो लेट्स स्टार्ट विद द नेक्स्ट टॉपिक एंड दैट इज जीआईटी. That is GIT. Today we'll finish the GIT beta, and once we will finish the GIT, then after we'll start tomorrow class beta, and next class, whenever the next class will be there, then only we'll start it. Okay? So I think my iPad get hang. Give me a moment beta. Yes beta, now it's functioning. So let's coming into the system. So beta, for understanding the system in part, first you already know that beta, that what is the system. So system is a group of organ, right beta? So now this is general part of physio is complete, and beta, one more thing I would like to tell you, that's this most difficult part of the physio we already finished, and that is the general physio. Now whatever is remained beta, this is all the systemic part, and if you have understood all these basic fundamentals, then this system is going to be very easy for you, because in each and every systems this is made up of different type of organs, and these organs are made up of different type of tissues, and these tissues are made up of different type of cells. So in every system you have to study the different type of cells, what are the function of these cells and how they regulate, right beta? So now in the GIT systems, what is the function of GIT beta? What is the function of GIT systems? As you already know, the function of the GIT is digestion. So whatever you are eating, this food is going into this GI canal. This is complete your food canal, and in this food canal, when this all part is going inside of your body, then there is some digestion is occurring, and after this digestion, all this required material will absorb into this blood, and through this blood it will circulate into the body. So this is how this function of the GIT is. So GIT function is nothing; it is simply digest the food. And what we are digesting, what we are digesting? Again, the three things only we are digesting. Number one, that is your protein. Number two is lipid, and number three is carbohydrate. Remember beta one thing, we have to digest only these three products, right? And what is the end product of these three things? From protein will make the amino acid, from lipid will make the fatty acid glycerol, and from carbohydrate will make the glucose and fructose. And then after this, we will absorb all this material. Now it will go into the circulation, then into the cell. Now question comes, first of all, you should understand, beta, all the enzymes, we require the enzymes for digest of these three things, these proteins, lipid and carbohydrate, we require the enzymes. And the enzymes which actually plays with this protein digestion, lipid digestion and carbohydrate digestion, we are using the word. And this is called as any enzymes which actually digest this protein, we use the word proteolytic enzyme. What? We use the word proteolytic enzyme. So now onwards, if anybody will say what is proteolytic enzyme, proteolytic enzyme is nothing, the enzymes which actually digest the protein. Next is lipid. So for lipid digestion, we use the word lipolytic enzyme, and for carbohydrate, we use the word amylolytic enzyme. Okay, so three enzymes will use the word proteolytic, lipolytic, and amylolytic, right beta? And what is the end product? End product you already you can see, amino acid, fatty acid, glycerol plus glucose and fructose, right beta? So coming to the GIT, this GIT is, first of all, we should understand what are the layers present into this GIT. So this GIT actually starts from this oral cavity and it ends up to the anal. And now what is happening beta? There are the layers which is present inside of this periconia. There is some extra layer called as serosa. So now coming from inside to out, there are total 4 layers are there. How many layers? 4 layers are there. The innermost layer, which we use the word for innermost, we use the word beta, endo. What we use the word? Endo. So this is endothelium, or either this is called as mucus. This is called as your mucosa. So this is your mucosa. First of all, coming from inside, that is number one, mucosa. Below the mucosa, for anything below, we use the word sub, and anything above of it, we use the word epi. So this is below the mucosa. So for below, what we use the word submucosa. So mucosa, then submucosa, then we require the muscle for contraction of this intestine. So there is a muscular layer. So first of all, mucosa, submucosa, muscularis, and then it segregates from the other organs. That is made the serosa. So 4 layers are there. Coming to inside to out. Number one, that is revise with me beta. Number one, that is mucosa, then submucosa, then muscularis, and then serosa. Mucosa, submucosa, muscularis, and serosa. Right? So now what is happening, beta? Actually, this mucosa is not like this. This mucosa is actually like this. Right? Why this is like this? Because this area is having only this much portion, and now into this much portion, what we want, beta? We want complete digestion and complete reabsorption, complete absorption. We want. Now what is happening when you are going outside from the country? You have to pass this immigration counter area, and you have seen how the line is making on this immigration counter. This is making something like this, right? And making this something like this. This is like this, like this. Why? Because in a small area, more number of person can be accommodated. So this is similarly inside of this mucosa. There is a like this presence of this increase the surface area, and this is called as villi. What we call this villi. There is a more projection over there like this again for increase the surface area, and this is called as microvilli. What we call this microvilli. So what is the function of this? Mucosa is simple columnar epithelium. Simple columnar epithelium, presence of villi and microvilli to increase the area. Increase the area. Right, beta? For increasing the area. Now remember, beta, one more things. As these are layers, as you can see, this is your mucosa. This is your this is your mucosa, right, beta? Then after this is your submucosa. This is your submucosa. This is your submucosa, right? And after this is your muscularis. This is your muscularis, right? And after this is your serosa. This outer layer is serosa. So now what happens, beta? There are two things. First of all, whatever nerves, whatever lymphatic drainage, whatever blood vessels will be present inside of these layers. These blood vessels, nerves, anything, they all will present where, beta? They will present into submucosal layer, MCQ. So at which area or in which layer all these nerve fiber vessels are present into this anteric system. This is the GI systems. So there it is the presence of in submucosa, right? So all the secretions, whatever secretion will be occur inside of this GI systems, all the secretion will be start from where? It starts from the submucosal area. Then muscle contraction is required for the movement of the food particles, right, beta? Called as peristalsis, and for that we require the muscle. So now again for regulation of this muscle we require the plexus, we require some nerves, and these plexus is present into this muscular layer. So the plexus which is present, there are two types of plexus into this enteric nervous system. So in this GI system there are two types of plexus. Number one which is present into the submucosal area, so called as submucosal plexus. Number two, the plexus which is present into the muscles. For muscle we use the word myo, and there is called as myntéric plexus. Another name of this submucosal plexus is Meissner plexus, and another name of this myntéric plexus is Auerbach plexus. That is Auerbach plexus. So now the question comes: what is the function of submucosal plexus? Submucosal plexus function is nothing; it secretes all and each and everything inside of this GI systems. What is the function of myntéric plexus? Myntéric plexus is contraction, and you can say like this is motility of this GI systems. So this is the function of myntéric plexus. So submucosa is having vessels, nerves, lymphatic and glands. Muscularis is smooth muscles, and there is a function is contraction and motility. Then serosa for attachment to the adjacent organ. Coming to the next, and in the next, that is the different parts of the GIT. In different parts of the GIT, always remember, first of all, that is the tongue. This tongue function is provide the taste sensation. Now question comes, how the attachment of this tongue is present inside the body. So when you will, in upward, doing the upward of your tongue, you can see there is a ligament is there on the beneath of this tongue. This is called a sublingual ligament. And this actually, where it is going, there, which bone this is, beta? Which bone this is? This is your mandible. So this mandible is coming, and there is a suture like this over here. And these sutures are at your midline of this midline of this mandible bone, right, beta? And what happens, beta? Whenever these ventral attachment, ventral means front side, dorsal means back side. So ventral, front side, dorsal, back side. Always remember. What happens, beta? These ligaments, these lingual ligaments, sublingual ligaments, actually they are coming over here and they are actually fixing into this mandibular suture like this, and that is how it protects to roll back of this tongue. So this is a ventral attachment called as frenulum linguae, made up of skeletal muscle, called as frenulum linguae, and dorsally it is attached with the hyoid bone. And hyoid bone already you know it is the only one which do not attach with any other bone. So what happens whenever this midline fracture happens of this mandible, this frenulum linguae can be removed from here, and once this is removed from here, the person's tongue can be rotated back, it can be rolled back, and it can be obstruct the airway, and this person can be die immediately. So that is why in midline fracture of this mandible, this is always very risky. Immediately you have to secure the airway obstruction or airway pass of this patient. So ventral attachment is due to frenulum linguae, made up of skeletal muscle. Then dorsal attachment is with hyoid bone. Coming to the taste part, better remember, most sensitive is bitter taste. There is some taste called as umami. Umami means the non-veg person who eats the meat and all. So what is the taste of the muscle? The muscle taste is actually umami, and this is actually functions with monosodium glutamate. Then salt is sodium or either potassium. Sour is by HIN plus acid. Sweet via GPCR, G protein coupled receptor, and this is also GPCR. What is GPCR, beta? GPCR is nothing. There is a one receptor like this functioning. As you can see, beta, this is like a snake-like structure. So this is called as serpentine receptor. This is also having a 7 calyx over here, so called as 7 helix receptor, and this is also made up of the protein, so called as G protein coupled receptor. This is called as G protein coupled receptor. G protein coupled receptor. Coming to the next, that is salivary gland, beta. In the salivary gland, number one, that is three major salivary gland. Number one, parotid. Then below the mandible, this is submandibular, and below the tongue, that is called as sublingual. So now coming to the first of all, parotid. The duct of this parotid is called as Stensen's duct. Stensen's duct. This is located up to this cheek, and this inflammation of this parotid gland actually called as mumps. What we call this? Mumps. So this is open at second molar, and this is main enzyme producer, and this is biggest salivary gland. Inflammation of this called as mumps, and this is also lead to inflammation of testis, called as orchitis. The next is sublingual. The duct of this duct of Rivinus. This is smallest duct, open at near incisor, located below the tongue, and this is the smallest salivary gland. The smallest salivary gland. Number third is submandibular. means below the mandible. This is called as below the mandible, and this is duct is called as Wharton's duct. This is the longest duct, open just behind the incisional lower. Maximum saliva producer, more than 70%. More than 70%. Now coming to the next bit, that is called as Wilder's ring. I hope that everybody have noted it. So this is the smallest salivary gland called as Wharton's duct. Nitesh, you are typing beta enough for today. No, beta, today we are going to finish the GIT as well. So today already the schedule you have seen. Just today the chapter was cell, and then next is body fluids, and then the muscle physiology, and in the last there is the GIT beta. So this is the last chapter for today. That is the GIT, and today we'll finish the GIT as well. Don't worry, we'll take the break in between this. And once if you have understood very well the cell and body fluids and muscle physiology, then beta you will get a very much interest into studying on the systemic part. So let's talk about this Wilder's ring. So what happens, beta? What is the Wilder's ring? What happens? So suppose if you are eating the food, right, beta? So suppose you are eating the food. What happens, beta? This is your hard palate. What is this? This is your hard palate. And after this, this is your soft palate, right? Above this area is called as nasal cavity. Below this area called as oral cavity. Beta. Remember, beta, this is your two tubes. There are one opening and there are another opening. This is your called as gullet. And what is this? This is your esophagus. So opening of the esophagus is called as gullet. Then this is your trachea. This is your trachea. And opening of this trachea is called as glottis. Right? This is your glottis. Now, beta, remember. As you can see in the picture, this trachea is in anteriorly side and this esophagus is in posteriorly side. And this is how I hope that your surgery is finished. So posteriorly, this side, this is the esophagus. So there is, if any outpouching is there, this is called as Jenker's diverticulum. It is always present to the back side. And this is our trachea. And this trachea opening is called as glottis. Above the glottis, whatever is this above the glottis like this. So for above, we use the word epi. So this is your epiglottis. This is your epiglottis. Right? Now, question comes, what is the larynx? But here is one box is present over here like this. This is called as your larynx. What we call this? Larynx. And this is also called as sound box. This is also called as sound box. Now what happens? Question comes, what is the pharynx? So beta, this area, here is your ear, like this, and this complete area, this is called as your pharynx. What we call this? This is called as your pharynx. So this is your pharynx. Now in the pharynx which is present inside of this nasal cavity, this is present over here at the nasal cavity, this is called as nasopharynx. And the pharynx which is present over in this oral cavity, this is called as your oropharynx. And the pharynx which is present near to this larynx called as laryngopharynx. So this is how this pharynx can be divided into three types. So this is all the complete open area called as pharynx. This is called as your nasopharynx, oropharynx, and laryngopharynx. Right, beta? Now what happens? Suppose you are eating any food and this food is getting inside. Now what happens? If you are eating this food from a non-hygienic place, so there are a lot of microbes are there, and these microbes should not enter inside of your body. What could be happen? If these microbes will enter inside of your body, you will become sick. So now one army should be present over here, and this army should kill all these microbes which is present inside of this food. And this army, the one army is present near to this tongue, just behind of this tongue. And this is called as your lingual army. And for army, we use the word tonsils. But we use the word tonsils. So tonsil is nothing that they are the army which kills the bacteria. So one army will be present on the tongue called as lingual tonsil. Then one army will be present on above side, that is the palate. So palatine tonsil. One army, similarly, if you are taking the air inside, which is having a lot of microbes. So ideally these microbes should also be killed over here. So one army will be present over here, and this is called as area is nasopharynx, so called as nasopharyngeal tonsil. And one more army which is present over this ear area called as tubular tonsil. So there are total 4 army: lingual tonsil, palatine tonsil, nasopharyngeal tonsil, and tubular tonsil like this. So 4 this side and 4 this side, and this is making a ring like this, and this ring is called as Wilder's ring. What we call this? Wilder's ring. So Wilder's ring is nothing. This is a ring of lymphoid tissue found in the throat, and in this, number 1, that is lingual tonsil. Number 2 is palatine tonsil. Number 3, that is nasopharyngeal tonsil. Or also called as adenoids. And number 4 is tubular tonsil. This is your Wilder's ring. So next is stomach. Now coming to the stomach beta. So stomach is having 4 parts. Number 1, that is fundus, pyloric, body, and cardiac part. And remember, this is the widest part of alimentary canal. And there is a present of the sphincter, which surgery you have already covered. GS, gastroesophageal sphincter, lower esophageal sphincter. And the food which will store over here. What is the function of the stomach? It will store the food and start the digestion. So food will store over here for 4.5 hours approximately. And remember, the fat, you are eating the 3 things only: protein, lipid, and carbs. So the fat is always longest stay in stomach. And that is why beta, whenever you eat the samosa, kachori, and everything, जब भी आप ज़्यादा खाते हो, तो बेटा आपको ज़्यादा fullness नजर आती है. Why? Because this fatty food will remain for a longer period inside of your stomach. Now coming to the cell, beta, inside of your stomach there are different type of cells, and these cells, number 1 is chief cells, and the function of these chief cells is release the pepsinogen, and prorenin. Beta, this prorenin, this is RENNIN. Tomorrow we'll discuss one more renin, that is renin, and this renin is secretes from the JG cell, which is actually for the RAS, renin angiotensin aldosterone systems. Tomorrow we'll discuss about it. Right. Number 2 is goblet cell. This is the mucus production cell. So this is for the mucus. Then there is the oxyntic or parietal cell. Function is HCl secretion, HCl and Kessel intrinsic factor. Then the G cell. This is for the gastrin. Enterochromaffin cell. This is 5-HT or serotonin beta. One more cell is there, that is called as enterochromaffin like cell, and this produces histamine. This actually produces the histamine, right? So chief cells pepsinogen and prorenin, goblet cell is mucus secreting cell, oxyntic parietal cell is HCl and Kessel intrinsic factor, and G cell is gastrin. Enterochromaffin cell 5-HT or serotonin, and enterochromaffin like cell. This is beta enterochromaffin like cell. Okay, this is your histamine. This Kessel intrinsic factor function is beta to allow the vitamin B12 absorption in terminal item. What could be happen if this Kessel intrinsic factor is not secreting from oxyntic or parietal cell? This will not allow for the vitamin B12 absorption inside of this terminal ileum. And if due to this Kessel intrinsic factor no vitamin B12 will be absorbed, this will lead to the pernicious anemia. But if Kessel intrinsic factor is there, but vitamin B12 itself is not there, this will lead to megaloblastic anemia. Always remember. Coming to the next part, that is small intestine. Small intestine, but nothing. Remember, this is a small diameter, but length is more, and this is around more than 6 meters. There is part number 1 is duodenum, number 2 is jejunum, number 3 is ileum. Better remember. Now this food is entering from the stomach. From the stomach, it is entering where? Into this duodenum, right? Once it will enter into this duodenum, this area for the maximum digestion. So now the question will come, the maximum digestion will be occurs where? The maximum digestion occurs will be into the duodenum. Now question will come, what is the next area? The next area is your jejunum. So this is your jejunum. Beta. Now question will come, maximum absorption into the intestine, that is in jejunum. So whatever maximum absorption will be there, this will be into the jejunum, and what is the maximum digestion will be there, this will be into this duodenum, right beta? So this duodenum is absorb 2 things more as well. Now what is happening beta? Suppose digestion is occur over here. Now all the digestion is done. It means from protein amino acid is ready, from lipid fatty acid is ready, from carbohydrates glucose is ready. Now they will absorb, and where they will absorb? They will absorb into the jejunum. Now what will be happen? Suppose this area is always remember beta, this area is also for 2 things absorbs maximally in this area, MCQ, and that is iron and calcium. So this duodenum is maximum digestion area, and here is the iron and calcium absorbs maximum. Then in jejunum, maximum absorption area, except B12, vitamin B12 and bile salts. MCQ. Vitamin B12 and bile salts. In ileum, it is the largest part of small intestine, and bile acid, partial and minimum digestion happens, and bile acid. This is bile salts, and vitamin B12 maximum absorbs. So in ileum, bile salts and vitamin B12 maximum absorbs. Coming to the next part, that is large intestine. In large intestine, but nothing. In large intestine, there is first of all cecum, then colon ascending, transfer descending sigmoid, then rectum and anus. And remember, all the digestion is already completed in duodenum. All the absorption is completely done in the jejunum. Now, this large intestine actually maximum absorbs the water and few electrolytes. Right, brother? So this is how all these things happens. Coming to the food digestion. So in saliva, where there is a 99% is water, and then there is enzymes, 99.9% lysozyme enzyme. And always remember, beta, in every secretory things, every secretion inside the body, there is which antibody is present? MCQ, that is IgA antibody is present. IgA antibody. And always remember, beta, for buccal cavity. Now remember one thing: the enzyme which is digesting this lipid, we can use the word lipase, but we use the word lipase. For amylolytic, we use the word amylase. So all the carbohydrate is digestive enzyme is amylase, and all the protein is protease. So this is how you should always remember, beta. Right? Lipase, amylase, protease. So now, just if any amylase is secreting into the saliva, we use the word salivary amylase, gastric amylase, and intestinal amylase, or pancreatic amylase. So this is how we always use the word, right? Now, remember, out of these 3 things, inside of this buccal cavity, maximum absorption always for the carbohydrate, and this is for mainly carbohydrate digestion. And this is from sucrose to maltose. Maltose is a disaccharide. So from this sucrose is polysaccharide, maltose become disaccharide, and then it will also convert into glucose and fructose, that is monosaccharide, right, brother? And this is with the help of chlorine ion, and also due to the salivary amylase. And this is also called as ptyline. What we call this? Ptyline. Then the esophagus, but no digestion occurs, only mucus is secreting. And also remember, in this saliva, there is also, now the newer study is also saying, there is some salivary lipase is also secretes that digests the lipid, and it releases from Ebner's gland. Secretes from Ebner's gland. MCQ. This is from Ebner's gland. Next is stomach. But in stomach, there is mainly protein digestion is occurs. Mainly protein digestion. And this is around 50% protein will be digested. And this protein will be digested with the help of pepsinogen. But whenever we use the word GEN in the end, this GEN, right? This GEN means this is not an active form. Once you will remove this GEN, this will become the active form. Like from angiotensinogen. it will become the angiotensin. So this is GN will be removed. Now it will become the activated. Why it is beta non-activated part always present inside the cell? Because this enzyme is actually used for the protein digestion, and inside of this cell or inside of the body, everything is made up of what? Everything is made up of the protein. So if this enzyme will be into the activated state, what could be happen? This enzyme will start lysis or start digesting on its own organ or itself on the cell. So that is why in the cell it is always present into the inactivated form. Once it will come out, then only it will become the activated with the help of activator. And this activator inside of this stomach is HCl. So this pepsinogen will become the pepsin, and this will with the help of HCl. With the help of HCl. Similarly, for prorenin will become the renin, and this renin actually for the milk digestion, milk protein digestion. Similarly, lipid digestion is also occurs over here 1%, and this is with the help of lipid again for the lipase, and here this lipase is secreting from the gastric part, so called as gastric lipase. Gastric lipase. Then the milk digestion is there for the casein protein, starts with the renin. Coming into the duodenum, 50% protein is digested, around 70% carb is digested, and 98% is lipid is digested. And in the school days, you have remember this is your duodenum, this is your duodenum, this is the first part, this is second part, and this is third part. But in the first part, there is some depression like this present over here, and actually this depression is called as crypts of Lieberkühn. What we call this? Crypts of Lieberkühn. So this is presence of crypts of Lieberkühn. And in the crypts of Lieberkühn, what is happening? Inside of this stomach, lot of HCl is there. Now what is happening? This HCl is getting out from this stomach from this food with the help of this food. And what could be happen when this HCl is start coming into this duodenum? What could be happen? This duodenum is not so very familiar with this acidic environment, and now always remember inside of your body there is a bicarbonate. Inside of body is bicarbonate, and bicarbonate property is always and always that is alkali. That is always alkali. So whenever the acid will come into duodenum, this more and more bicarbonate will be secreted inside of this duodenum to neutralize of this acid. Right, beta? So this is present mucus secreting submucosal gland. Submucosal means into the mucosa. We already discussed about all the glands, secretory gland, where they will present into the submucosa, and at this area, the submucosal gland which is present over here is called as Brunner's gland. That is Brunner's gland. And now this Brunner's gland present over here. And remember, in duodenum there is no pseudo enzymes. All the digestive enzyme either it will come from the pancreas or either the bile acid will come from the gallbladder. So right. So in main, this is the main digestion occurring area. Main digestion occurring area. And here is also fat emulsification is also occurs. Fat emulsification is also occurs. What is the meaning of emulsification? This is a fat. If you will do the pieces of this fat, this is called as emulsification, and this is done via with the bile acid. Then there is a pairs patches. Suppose this RB cannot control or cannot finish all the microbes which is present over here. Now these microbes is entering into the alimentary canal. What is happening in these GI systems? Again, these microbes should be killed by someone, and those are called as pairs patches. What we call this? Pairs patches. So pairs patches is nothing. They are intestinal tonsils or lymphoid tissue present at the ileum. Then there is bile juice. The bile juice is yellow green in color. No enzyme is there, and every day there is 700 ml per day is the production. 97% is water and pigments, is bile pigment, salts, bicarbonate, and cholesterol. Then next is pancreatic juice. All the digestive enzymes. This is your stomach, duodenum, and this is your pancreas. So all the digestive enzymes will come into this duodenum, but they all will come from this pancreas. So this is how this pancreas helps for most of the digestion is occurring into the digestion into this duodenum, right? Now the pH of this 7.6 to 7.8, and pancreatic juice every day 1 to 1.5 liter per day secretes. There are enzymes present called as this is secreted by CCK. All the enzymes, digestive enzymes, they are secreted by CCK. CCK stands for cholecystokinin. And later on we'll discuss there are three functions of this cholecystokinin. Number one function is it will decrease the gastric motility, it will increase the gallbladder motility, and it will increase the pancreatic digestive secretions. Right. Second is water is there 98%, and bicarbonate will also come from this pancreas because still if there is more acidic food is there into this duodenum, this acid again will be neutralized with the bicarbonate, and these bicarbonate will be secreted via secretin. This is via secretin. So these digestive enzymes, in the gastric enzyme, there are three enzymes. Number one that is called as your prorenin. Number two is pepsinogen. Number three is gastric lipase. And two are supporting centers. Number one is HCl. Number two is Kessel intrinsic factor. Next is intestinal enzyme. In intestinal enzyme, there are total 11 enzymes. No need to remember of these all the enzymes. Just remember this enterokinase. This enterokinase is quite important for your exam purpose. Then coming to the pancreatic enzymes, there are total 6 enzymes. Nuclease for digestion of nucleic acid, lipase for lipid, amylase for starch, and here is the chymotrypsinogen for protein. Trypsinogen again for protein, procarboxypeptidase for protein. So chymotrypsinogen is for protein, trypsinogen again for protein, procarboxypeptidase for the protein. Now this protein digestion, how this protein digestion is occurring? Number one, only 3 digestion there will be. Number 1 protein, number 2 is lipid, number 3 is carb. So how the protein digestion is occurring? 50% protein digestion into the stomach, that GJ stands for gastric juice, pH is 1.8, highly acidic, and this pepsinogen converting into HCl and into pepsin with the help of HCl, and this will become into large polypeptides. This large polypeptide will again convert into dipeptides, then monopeptides. 50% protein in duodenum, and in duodenum it is helping, it is doing, first of all the trypsinogen is coming from the pancreas, and then in intestine there is an enterokinase. This will converting into this trypsin. Now there is one more thing, chymotrypsinogen is also coming from the pancreas, and now this chymotrypsin will be converting with the help of trypsin into chymotrypsin, and now this chymotrypsin is actually effect of this digestion on this 50% part of this duodenum, right beta? Then the next is now suppose this large polypeptide is ready now, right beta? Now this large polypeptide will convert into dipeptides plus amino acid, and this is how it will be happening with the help of this pro-carboxypeptidase. This pro-carboxypeptidase will convert into trypsin with the help of trypsin into aminopeptidase and carboxypeptidase, and then it will convert this large polypeptide into dipeptide. Coming to the milk protein, this milk protein is casein, again in stomach, this prorenin converting with the help of HCl into the renin, and then this renin will converting this milk protein into the caseinate. This will becoming the curdling of the milk. And beta, remember, whenever you have seen a small baby, and this small baby, whenever the small baby having the breastfeeding, after sometimes when this baby vomits, why this milk is coming out like a curdling? Why this is like a curdling milk? Because inside of this stomach, when this milk is reaches over there is starting of the digestion, and this digestion of this milk protein is making curdling of this milk, right beta? Next is lipid. This lipid is actually first of all always emulsify. This is a bigger molecule, emulsify with the help of bile. So bile function is nothing. It is just making a small particles of this bigger lipid particles, and this is called as emulsifications. Then this is entering into the pancreas, entering into the duodenum, and from duodenum it will be coming into the digest side. How it will become a digest side? Pancreatic lipase. Lipase means lipid digestive enzyme, and intestinal lipase is functioning over here, and they both are converting this emulsified lipid into diglyceride, then monoglyceride, fatty acid, and then glycerol. So this is how this fatty acid is already there. Now remember, this salivary lipase is present only in the infants, and gastric lipase is less than 1%. Gastric means stomach, and lipase means for the lipid. And now the lipid digestion is 80% occurring into pancreatic lipase, then 18% due to intestinal lipase, 1% due to gastric lipase, and 1% due to salivary lipase. Salivary lipase. Next is carbohydrate. Next is carbohydrate. Next is carbohydrate. So in carbohydrate, you already know carbohydrate 30% starch is already starting converting into the oral cavity, and this is due to the salivary amylase. Salivary amylase. This is into the maltose. Then 70% into the duodenum with the help of pancreatic amylase. Pancreatic amylase again into maltose, and this will produce the 2 unit of glucose. This will produce 2 unit of glucose. Then this lactose beta converting with the help of lactase into glucose and galactose, and sucrose again with the help of sucrase into glucose plus galactose plus fructose. Now the phases of digestion. We will complete these 3 phases digestion, and then we'll take the break. After the break, we'll finish all this GIT part today. So I hope that today class we can finish up to the 6:30 only. So at 6:30 our class will be finished today, GIT part, and in the next class we will starting with this excretory systems and respiratory systems. Okay, beta? So phases of digestion. Number 1 is cephalic phase. In cephalic phase, when with the smell of the food, when your all the mouth will get watery, and all some digestion enzymes is also secreted. This is called a cephalic phase. So this is with smell of food. This is a parasympathetic activity, beta. This will increase saliva secretion and increase HCl secretion. Then the gastric phase, when food in stomach, increase the gastric secretion, and this is due to the vagus nerve. So vagus stimulation beta, and in this phase maximum HCl secretion will be occur. MCQ, remember question will ask maximum HCl secretion occurs which phase of the digestion? That is in the gastric phase. Then intestinal phase, when this HCl reaches into the duodenum. So when HCl reach into duodenum, and this is also due to vagus stimulation, and here is a decrease in HCl and increase in bicarbonate secretion. Definitely beta. More HCl is coming from the stomach, and now this duodenum wants to neutralize it. So during this intestinal phase there is a lot bicarbonate will be secreted, and this is with the help of CCK. VIP stands for vasoactive intestinal peptide, and secretions. This is not secretion, this is secreting. This is secreting. This is secreting. So I hope beta that's you are okay with the pace, because as your exam is very nearby beta, we are also trying to finish our course as much as possible as soon beta. So I understand that you have not studied so much in a single day, but beta, there is no choice for you, even for you and even for us. So that is why today we are finishing all this 4th chapter, and the remaining part we will cover in the next upcoming classes, so that more time we will get it to study, and all the material you will get it as soon as possible. बेटा मैं भी नहीं चाहता कि मैं आपको इतना सारा एक साथ पढ़ा दूं, लेकिन बेटा अभी आपके पास भी समय नहीं है, आपको भी पढ़ना है और आपको भी एग्जाम पास करना ही करना है। और जब आपको एग्जाम पास करना है बेटा, तो डेफिनेटली ये 2 महीने आपके लिए इस तरीके से होने चाहिए बेटा कि इसमें कोई ब्रेक नहीं, कोई इफ नहीं, कोई बट नहीं, आपको सिर्फ पढ़ना है और पढ़ना है। इसलिए बेटा many students are writing right now, these are enough for today, but beta I understand this is not enough for today beta, because please increase your ability. ऑफलाइन क्लासेस के अंदर भी हम लोग बच्चों से यही कहते हैं कि प्लीज पुश योरसेल्फ, एंड राइट नाउ यू आर सिटिंग एट होम बेटा, यू आल्सो प्लीज पुश योरसेल्फ, देन ओनली यू कैन पास दिस एग्जाम, अदरवाइज बेटा इट गोना बी वेरी डिफिकल्ट फॉर यू टू पास दिस एग्जाम. So please start pushing yourself, please be patient and try to understand the things, try to make the concept. Okay beta, I'm giving you my number, this is my WhatsApp number, so if any problem, you find any problem, you can message me on this WhatsApp 750-360-7854. Dr. Arun, I'm from Jaipur, Rajasthan beta. So 7503607854 बेटा कोई भी बात हो कुछ भी हो आप मेरे को इस पर मैसेज कर देना बेटा. I will try my best to reply you as soon as possible. But sometimes it happens बेटा that my class is going on and some discussion is going on, so might be on the same time I cannot reply you back. But whenever I see the message, I will try to reply you as soon as possible. So this is my number. Now we'll take the break, and after the break, only 1 hour classes remain for today, and then we'll finish the today's class. So this is break for 20 minutes. We will start the class at 5:25 p.m. Thank you very much. Yes, my friends, welcome back after the break. तो बच्चों आप चाय वाय पी के हो गए आप एकदम बिल्कुल फ्रेश. Shall we start now? So let's start with the absorption. So till now we have seen अपन ने अभी तक क्या देखा बेटा कि आप जो भी कुछ खा रहे हो वो क्या खा रहे हो बेटा. What you are eating only that's either protein, carb or lipid. And what is happening when you are eating everything? This is going inside of your stomach, then into duodenum, and in duodenum there is a maximum digestion is occurring of these substances. Now what is happening better? When you are eating all these three products, now the end product is coming out already. You know, from protein this is amino acid, from lipid that is fatty acid, and from carbohydrate this is glucose. Now these three products are ready. Okay, we have seen that's how the different enzymes are there and how these enzymes are functioning differently. Now what is happening better? That's now these products are ready. Where they are ready? Suppose this is your gut. Okay, this is your gut. And your gut is actually made up of like this cell. This is the cell because inside the body everything is made up of the cell, isn't it? So this is your cell. This is your cell, and this cell name is enterocytes. So now this food is over here. Amino acid is ready. Now fatty acid is ready. Glucose is ready. Right, beta? Now what we want with these all substances, we want to absorb them, and where we will absorb them? First of all into the enterocyte. Then where it will go, beta? It will go to the back side. There is a blood vessels like this. So now what is happening, beta? First of all, this will absorb, and this is which wall? This is your apical wall. Which wall? This is your apical wall. So now this amino acid, fatty acid, and glucose. First of all, where it will be absorbed? It will be absorbed from this wall. Yes, from this wall. And after coming on this wall, these all substances will come out from this basal wall. So this is the basal wall, and then it will enter where, beta? Then it will enter into the blood, and through the blood circulation, it will going up to the liver for synthesis. So now we will study about the absorption. This process is called as absorptions. And in the absorption, you know, I already told you that this is a cell. So in the cell, it means there is some transport channels are there, different transport channels are there. And through these transport channels, the absorption of these all the ions will be happen over, all the substance will be happen inside of the cell. And then through the cell from this basolateral side, this will coming into the blood. So this is how the absorption will occur inside of your GIT. Right, brother? So coming on the topic, absorption in the oral mucosa. The first area is oral mucosa. So in oral mucosa, there are few drugs like nitroglycerin and fentanyl. Then in the gastric mucosa, there is alcohol, aspirin, PCM, water, and some glucose. Then in the duodenum, already we have discussed about duodenum, iron and calcium. Iron and calcium. Then in jejunum, maximum absorption area, maximum absorption site. Then in the ileum, beta, 2 B's, vitamin B12 and bile salts will be absorbed. Coming to the next part, that is colon. And remember, in the colon, beta, in the colon, only the water and some minerals. In the colon, only some waters and the minerals, because whatever food is there, whatever food is there. Look at the diagram, beta. Look at the diagram. This is your stomach, right? Then this is your duodenum. Then this is your jejunum. Then this is ileum, and then beyond this area, this is your colon, right? This is your colon. So remember, first of all, at this area, iron plus calcium will be absorbed. At this area, this is your jejunum. So in jejunum, maximum absorption, and in ileum, vitamin B12 and bile salts. So this is how, this is iron and calcium area, this is maximum absorption area, and this is vitamin B12 and bile salts area, and this is your large intestine. And in the large intestine, remember, there is water plus minerals. Water plus minerals will be absorbed. So this is how inside of your body all this absorption will be occur. Now in this colon, water, mineral, and all the food which is over here right now, this is the undigested food. So suppose already the maximum digestion occurs over here, right? Now what is happening? In this area, the minimum digestion is also occurs in this terminal area. But now when it enters into the colon, in this colon, which the food which is coming over here, this is not going to further digest, and the food which is present over here, we are using the word undigested food. So the food which is present inside of this colon, we call that undigested food, right? So now this undigested food, so there are long chain fatty acid. Always remember, long in short and short in long. So this is in small intestine, and short chain in large intestine. And this is the question of 4 June 2023. 4 June 2022, the short chain fatty acid is maximum absorbed in which intestine? And the right answer of that question was large intestine. There is something called D-xylose test. Some of the children, they are having the sensitivity for the glucose, gluten, and this sensitivity means there is no properly absorption of the carbohydrate, and they are sensitive for this, and we know that disease called as celiac sprue. That is called as celiac sprue. Now remember, this fructose is from GLUT5, F for fructose, and F stands for GLUT5. So this GLUT5 is a facilitated type of diffusion. So this actually absorbs with the facilitated diffusion. Glucose and galactose, they are from secondary active transport from SGLT1. Remember, beta, MCQ. Amino acid again from the facilitated diffusion and some of the secondary active. And hexose is the small glucose, hence it's easily absorbs. Coming to the next, beta. Now fat is ready. Now fat digestion will be occur. So how the fat digestion will be occurs? Remember, beta. First of all, I would like to ask you, what is the lymphoid systems? What is lymphatic systems? What is lymphatic system? Question comes in the mind, what is the lymphatic systems? But remember, this is your blood vessel, right? And this is your cell, right? Now we already know maximum fluid will present over inside of this intracellular fluid, right? And minimum is in only in plasma, and middle portion is in interstitial fluid, right? Now what happens? Beta? When this side of area, this is, we also know that inside of the cell, through the metabolism, what is also manufacturing, or what is also producing inside the cell. So now these pipelines is having its own limitation to carry the fluid. Now what could be happen if you will drain more and more fluid inside of this pipeline above the limit? What could be happen? This pipeline can be ruptured, isn't it? So what happens, beta? As draining of this area of fluid, this is happening with a special or another pipelines which is present over here, and these pipelines are called as lymphatic vessels. What we call them? Lymphatic vessels. Now what happens, beta? Actually, due to the presence of RBC, WBC, these are called as blood. But now the fluid which is present inside of this interstitial fluid, there is no RBC, WBC, so we cannot call it as a blood. So now we call it as only the fluid. Now this fluid is clear in nature. This is like a water. Now what happens? This extra fluid actually drain into this lymphatic vessels, and then it carry up to the inferior vena cava or vena cava area, and then it drain into this vena cava. So this is how it gets back into the circulation. Now what happens, beta? Inside of this cell, if any pathogen will come, there is a lysosome is present. So this lysosome will kill that pathogen. And inside of this blood vessels, what happens, beta? When this vessel, any pathogen comes inside of this vessel, there is a WBC. So this pathogen will be killed by the WBC. But what about if any pathogen come inside of this interstitial space? Now there is no any cell, no any organelle that will kill this pathogen which is present over here like this. Now what could happen, beta? Suppose the pathogen which is present over here like this pathogen can travel back into this lymphatic vessels like this. And if this pathogen is going into this lymphatic vessels, it means there is nobody there to check this pathogen, and this pathogen can lead to a lot of infections. So what happens, beta? In this lymphatic vessels, actually there is a different checkposts are there like this. And these checkposts are actually called as lymphatic nodes. What we call this? Lymph node. So these are actually lymph nodes. So anytime if this pathogen enters inside of these vessels, these checkpoints will check this pathogen, and this pathogen will be killed by this checkpoints and called as lymph node. And that is why you have seen whenever the person is a chronic illness is there, the size of the lymph node is increased. Similarly, what happens, beta? If these vessels are blocked due to some infection, due to something, if these vessels are blocked, what will happen? Now this extra fluid cannot go into this area. And now what will happen, beta? This extra fluid will start accumulating into this area, and in this area you will know that any fluid accumulates inside of this area, this will lead to the edema, and that is why in lymphatitis means any problem, any inflammation of lymphatic vessels lead to the edema. And you know, beta, there is a filariasis mosquito which causes that Japanese which causes a lymphatic filariasis. And in filariasis, what is happening? There is a lymphadenitis. It means there is inflammation of lymphatic vessels, and it may lead to elephantitis. This much size of the foot happens due to the blockage of this lymphatic vessels. Right, beta? So this is the function of this lymphatic vessel. Similarly, what is the cancer? What is tumor? When uncontrolled growth of cells start producing more and more number of the cells. So what is happening? These cells can travel into these lymphatic vessels as well. So now what is the treatment of the cancer? Just remove that all area. Now suppose if any cell is already entered inside of this lymphatic vessels, what could be happen? Now this can travel through this fluid onto some another area, and that is why in the cancer we all the time we check the lymph node is spreading. Why? Because these cells can enter inside of this cell and inside of this vessels, and then it can travel. So that is why we are using the word lymphatic spread. Right, beta? So this is how the basic funda is. This is only the blood vessels, cell, and then the interstitial fluid. Right, beta? So now what happens? In this fat absorption, first of all, this fat will be emulsified. This fat will be emulsified. This is the lipid droplet coming to the bile salts from the gallbladder. This will emulsify, making small particles. Then it will digest with the help of lipase. After digestion, this will become the monoglyceride, bole to fatty acid. Now what happens? This fatty acid actually binds with the bile salts. And when this bile salts attach like this, what happens, beta? Now this is called as a micelle. What we call this? Micelle. Now this micelle will travel up to this wall of the intestine, and now what happens? This bile salts cannot enter inside. These bile salts cannot enter inside, and this bile acid, this bile salts is always stay outside, and only fatty acid gets inside. Once this fatty acid gets inside, what happens? This fatty acid actually binds with the protein, and now it will becoming the chylomicrons. What we call this? Chylomicrons. And now this chylomicron come out from the cell, and then the lymphatic vessel is there, and then it will mix into the lymphatic vessels, and through the lymphatic vessel, this is going into the circulation. And name of this lymphatic vessel is called as lactate. This is called as lactate. So this is how this fat absorption is occurs, beta. This is how this fat absorption is occurs. So look at over here. This fat absorption, first of all, fatty acid plus glycerol, bile salts, and phospholipids will bind, and then it will become the micelles. Simple diffusion into enterocyte, then it will become the chylomicron, and absorbed by the lymph capillaries called as lactate. This is called as lactate. This is your chylomicron. Coming to the next, beta, that is iron absorption. So now, which area the iron maximum absorbed? Beta? That is in the duodenum. So in duodenum, maximum iron will be absorbed. Now look at over here. What happens? This iron is actually Fe3 plus. So first of all, what happens? This is iron. So this Fe3 plus actually convert into the Fe2 plus. This iron convert into the Fe2 plus. So now it is becoming from trivalent into the divalent. Why? Because this is from trivalent into the divalent. Now what happens, beta? When look at over here, this is the intestinal lumen, right, beta? Now what happens? This iron is converting into the divalent, and this is a type of metal. Iron is a metal. So this is a transporter name on the apical side of this enterocyte, and this transporter name is divalent metal transporter, DMT, divalent metal transporter. This divalent metal transporter absorbs this iron from this intestinal lumen, then it enters into the enterocyte. Now why this iron is required, beta? As you know, this iron is required for formation of the hemoglobin, because in the hemoglobin there is 4 chains are there, alpha 1, 2, beta 1, 2, and this iron is actually mixed with this chains, and then it binds the oxygen, and then it carry for the transportations, right? So this iron is maximum absorbed into the enterocyte. Now what happens? When this iron is not required into the blood, because if there is a more number of the iron, then it is also the problematic. So we are not allowed to go this iron. And so now this iron will start storing inside of this enterocyte in the form of ferritin. But now suppose if this iron is required and this iron is need to get out from this enterocyte from the basal wall. So this is the side. This is your basal wall. This is your apical wall. So from basal wall, there is a bit called another transporter or another channel that is called as ferroportin. So this iron comes out from this ferroportin from this basal wall and then it enters into the blood. And once it enters into the blood, it mixes with the transferrin. It binds with the transferrin. And when it binds with the transferrin, then it transports into the liver and it goes into the liver and then there is a synthesis of the hemoglobin. What happens? When there is enough hemoglobin is there, enough iron is there. So now what happens? This liver sends another enzyme called as hypestin and this hypestin will come and this hypestin will block this ferroportin. So now this iron cannot come out from this enterocyte into the blood and now it starts accumulating inside of this cell. Right, beta? So this is how this iron transport occurs. How this iron transport occurs, beta? I hope so now it's clear to you. So now write it down. This iron is absorbed by DMT1 into enterocyte. DMT stands for divalent metal transporter. This is on apical side. Then ferritin coming out through the ferroportin from basolateral side enters into the blood. In blood it binds to the transferrin. And this is how this iron travels inside of this lumen. Right, beta? So I hope so. Now it is clear. So for iron absorption, maximum iron absorption into the duodenum, and in duodenum there is a DMT, divalent metal transporter 1 on the apical side of this, and then it binds with the transferrin inside of the blood and goes into the liver. Coming to the next GI hormones. In GI hormones, you already know that there is the G cells which produces gastrin, S cell secretin, and I cell is CCK. Now action is gastrin, beta, it increases stomach motility and it is also increase gastric secretion. Secretin we already talked about, it decreases gastric motility and it increase pancreatic bicarbonate secretion. It increase pancreatic bicarbonate secretion. CCK having three functions. Number one, it will increase motility of gallbladder and pancreas, decrease gastric motility, and it secretes pancreatic enzyme. There are some more enzymes are there like ducrenin, beta. Ducrenin, this is in duodenum, again from the Brunner's gland. Brunner's gland releases. Then GIP, gastric inhibitory peptide, enterokine, inserting gut peptides like GIP and GLP-1. Now coming to the next topic, that is GI movement. And in GI movement, what is happening, beta? In GI movement, there are total 5 types of GI movement. Listen to me very carefully. First of all, there are total 5 types of GI movement. Number one GI movement, that is called as 4 are into the feeding state and 1 is into the fasting state. So when you are eating the food, there are 4 types of GI movement, and once you are not eating any food, there is 1 type of GI movement. So total there are 5 types of GI movement. Number one, very careful. First, you are eating the food. It is going into the food pipe, GI systems. Now this food should go ahead. And now this food is going ahead. This is with the help of peristalsis. So now you know what is happening in peristalsis, beta. In peristalsis, first of all, this area should be contract and this area should be dilate. So this can lead to this move, this bolus or this food particles to the forward side only. This is how it is happening. So this is peristalsis. Inside of this peristalsis, always remember, beta, for constriction, you require some neurotransmitter because this muscle contraction is not directly can be caused. So for muscle contraction, you require the neurotransmitter, and the neurotransmitter which helps to constriction of this muscle at this retrograde side, this is called as acetylcholine and substance P, MCQ. For relaxation, this is your anterograde side, and for relaxation, you require this VIP and NO. VIP stands for vasoactive intestinal polypeptide, and NO means nitric oxide. So remember, nitric oxide is always doing the dilation. And similarly, in this intestine is also dilated with the help of nitric oxide and VIP. This question is asked many times. That who is doing the contraction, acetylcholine substance P, who is doing the dilation? This is VIP plus nitric oxide. So number one, that is your peristalsis. Number two, what happens? Now this food is coming, and now food suppose enters into the duodenum. Now in duodenum there is a lot of digestive enzyme is coming. Now this digestive enzyme should be mixed with this bolus. This digestive enzyme should be mixed, isn't it? Now for mixing of this digestive enzyme inside of the body, there is no spoon is there. Means you are preparing. आप एक सैंडविच बना रहे हैं और जब आप सैंडविच बना रहे हैं तो उसको आपको मिक्स करना है सारे मसाले डाल के. How you will mix it? With the help of a spoon. But there is no spoon inside the body. So now how the body can mix all the digestive enzyme with this bolus? And this mixing of the food, beta, is actually happens like this. Means you will constrict this area and you will constrict, beta, this area. This both area is constrict. Means also you can constrict this area. And now what is happening, beta? This area constrict, this area is constrict. Now what could happen, beta? Now you are doing one side. There is a contraction, relaxation. And this process, when you are doing only a single place, all this contraction and relaxation, this is actually helping the mixing of all the digestive enzyme with the bolus. And this actually movement is called as segmentation. What we call this? Segmentation. So first peristalsis, second segmentation. Now what happens, beta? All digestion is done, all absorption is done. Now this food is entering into the colon. And once inside of this colon, suppose this food is entering into this colon. And now in this colon, beta, this food is having a lot of water in it. This undigested food is having a lot of water and few minerals is over there. And for removing the water from your clothes, how you will do the clothes, beta? You are doing like this, all the clothes, then only the water will come out. And similarly, this movement is called as hostration. So this movement is called as hostration. And this hostration is done in where? In the colon, in the large intestine. And that is why, beta, you have seen that a person is having a constipation during the defecation time, passing of the stool. How the stool will come out after 2 or 3 days? This will become very solid. Why? Because maximum amount of water is already absorbed from this feces. Now this feces become very solid part. And that is why in the constipation, the feces will come very tight. Okay, beta? So this is called as your hostration. So number one, peristalsis. Number two, segmentation. Number three, hostration. Now what is happening? Now this food is coming into where? This food is coming into this rectum area. And in the rectum area, beta, what is happening? You want to defecate out. Defecate means passing the stool. So you want to defecate it out. And once you are going for the defecation, beta, all the undigested food should be get out from your gut. Right? And now for getting out all this food, you require movement, and this movement is called as mass movement. What we call this? Mass movement. And that is why, beta, when you are going for the freshen up, with the help of this mass movement, all the undigested food gets out within 2 or 3 minutes. Right? Now this 4 motility already you have seen. Number 1, peristalsis. Number 2, segmentation. Number 3 is hostration, and number 4 is mass movement. Now what happens? Suppose now no food is here. Now this machine is completely empty, and if this is something type of machine, this machine should be cleaner for the preparation of the next meal. And for cleaning process, what happens from here, beta? Some like this movement gets out from this area, and this is for the cleaning of this complete gut area. And this cleaning process is called as migratory motor complex, MMC, migratory motor complex. Right, beta? So this migratory motor complex, this is during the fasting state. So number one, that is your peristalsis. Peristalsis. Number two, segmentation. Segmentation, and this is into the large. This is into the small intestine. This is into small intestine, small gut. And these two are into the large gut. That is mass movement and prostration. In fasting state, beta migratory motor complex, bole to MMC, and this starts after 4 hours of the last meal. So peristalsis is the reflex activity, stretching of the gut, propulsive movement, and this is coordination action of enteric nervous system. ENS stands for enteric nervous system. Right? Beta? VIP stands for vasoactive intestinal peptide. NO stands for nitric oxide. Right? Then the mass movement. Mass movement is 3 to 4 times after meal in large gut. This is for the 3-minute stretch in colon, and this is also called as gastrocolic reflex, then duodenocolic reflex, or also called as defecation reflex. Then segmentation is non-propulsive, and this is a type of pendulous movement, like a pendle. This is a pendulous movement. Rate similar to BR, but BR stands for basal electrical rhythm. This is 12 per minute in gut. Cut it, beta, this. And MMC is electrical plus motor activity. This is for 90-minute interval. Migrate from stomach to distal ileum, increase secretion. And when food enters into this gut, this stops the MMC and starts the peristalsis. Okay, beta. Coming to the next part, that is SCL secretions. For this SCL secretion, beta, if you will understood this SCL secretion, you will understood how this SCL is forming, then you will be understood what is the function of PPI, proton pump inhibitor. Remember, this SCL is secreted from which cell? This is oxyntic or parietal cell. Oxyntic or parietal cell. Now remember one thing, beta. What happens? We already know that. Suppose this is your cell. Due to the metabolism inside of the cell, CO2 is produced and water is produced. We already know. When they both combines with the help of carbonic anhydrase, it makes H2CO3. This H2CO3 is actually very weaker substance and dissociated to H ion plus HCO3 minus, and this is called as carbonic acid. What we call this? Carbonic acid. Now this H ion is already formed, bicarbonate is formed. Now H ion is always acidic in nature. This is acidic in nature, and this is always alkaline in nature. This is the chemical property of that substances. So now what happens? Always remember, on this cell, this is the plasma. This is your plasma side, and this is your gastric lumen side. Always remember, beta, during the salivation, there is a more amount of potassium is secreted from the saliva. So now what happens from the plasma? Through this plasma side, beta, there are total 3 receptors present on this basal side of the cell. So number 1 receptor, M1, H2, and gastric. So there are total 3 receptors present on the basal wall of this parietal cell. Now this M1 receptor is for acetylcholine. This is for acetylcholine, beta. This H2 for histamine, and this is for G cells produce gastric. So now what happens? Whenever there is you have started to consume the food, beta, so vagal stimulation is there, vagus nerve will be excited, and once the vagus nerve will be excited, what will happen? क्या होगा बेटा? जैसे vagus nerve को पता चलेगा कि आपने खाना खाया है, vagus nerve acetylcholine release करेगी, it will release acetylcholine, and this acetylcholine actually activates this M1 receptor of parietal cell. Once this M1 receptor will be excited, this M1 receptor start functioning inside of the cell. Similarly, it will also activate this H2 receptor, and it will also activate this gastrin receptor. Similarly, these 2 receptors also individually can be activated via the hormones like histamine. So if there is more histamine, this will activate this H2 receptor, and if there is more gastrin, this will also activate this gastric receptor. Now what happens? When these 3 receptors are combinedly function together, this will lead to more and more HCl production. And what happens, beta? Whenever these receptors are activated, these receptors will tell to the inner part of the nucleus that nucleus, please form the HCl. There is a message for you from the brain, please form the HCl. And now what happens, beta? When the message is reached into this nucleus from all these 3 receptors like this, so now what happens, beta? Now what happens? This nucleus will start producing the HCl. And now how the HCl will produce? Look at over the screen. What happens? This H2CO3 binds together, makes H2CO3, and dissociating into HCO3 plus H ion. Right, beta? Now what happens? For positive ion we use the word protons, and for negative ion we use the electron. Now what happens, beta? Suppose if one positive ion is getting out, so there is a one channel, and this channel, what channel leads to? This channel will open, and this potassium, this hydrogen starts getting out. This hydrogen start getting out. Which side? To the gastric lumen side, to the intestine. Now this H ion is getting outside over here, beta. Now if one positive ion is coming from outside, one positive ion should go inside, and inside already there is a lot of potassium is there. So for sending this potassium from outside to inside, we require the pump, and this is the pump. Now this pump, what is happening? 2 positive ion is moving inside and outside, and positive ion we use the word proton, and that is why this pump is called as proton pump. And this proton pump actually helps to get out this H ion from the cell and takes inside to this potassium, and that is why we use the word proton pump. Now what about the HCl? This chlorine, because H ion is already there, with the H ion there should be the chlorine ion should be bind, then only it will form the HCl. Now what happens, beta? When this H ion gets out, similarly this bicarbonate also gets out from the cell, and where it will go? It will go into the blood. Now this is the alkaline property, and now this bicarbonate is going into the blood. What will be happen? If one negative ion is getting out into the blood, one negative ion which is already present into the blood, that is chlorine, because you already know in ECF most abundant anion is chlorine. So this chlorine should enter inside of the cell. So if one negative going out, one negative should get in, and then this chlorine ion get out from this apical wall, and then it also enters into the gastric lumen. Now what happens? H ion was already there, chlorine is now came, and they both binds together, and it forms the HCl. So this is how beta HCl actually forms. Now what could be happen? You don't want to produce this HCl. Look at on the pharma. You don't want to produce this more and more HCl. What you can do? Either you can block this channel, either block this, either block this. And now what happens? This you cannot block it properly. This you can block via ranitidine. This you can block via ranitidine, so called as H2 receptor blocker. But still, even if you have blocked this H2, still this is working, this is working. So still a lot of HCl can be produced. So that is why beta, this ranitidine now outdated. We no more use of this ranitidine. Now we are not using this ranitidine for this HCl production. So now what else you can do which is more effective? Just forget about these receptors and just block this pump. And which pump this is? This is the proton pump, so called as proton pump inhibitor. And those inhibitors are pantoprazole, rabiprazole, and everything. So now, how much these can be activated? If this pump is not functioning, this HCl cannot be formed, and that is why this is most effective. Right, brother? That is why this is most effective. So this is how I hope that now you know that how this HCl is formed. There is one more thing called as Zollinger-Ellison syndrome. What happens in Zollinger-Ellison syndrome? In Zollinger-Ellison syndrome, this is a type of tumor of the gastrin, G cell, gastrinoma. Now more and more gastrin will be produced, and this more and more gastrin will be produced, and it will activate this gastrin receptor, and this will lead to more and more HCl production. So in Zollinger-Ellison syndrome, there are the ulcers. There are ulcers. So this is how you should understand. There is one more thing called as postprandial alkaline tide. Called as postprandial alkaline tide. What is postprandial alkaline tide? What happening when you are eating the food? Your blood will become more alkaline. Ideally, your blood pH should be 7.35 to 7.45, but it is increases little bit more for fewer times. And this process is called as postprandial. Postprandial means after food consumptions. Alkaline. Alkaline means this is more alkaline in nature, and this is like high tide in the ocean. This is similarly postprandial alkaline tide. Why? Because this more and more bicarbonate is entering into the blood, and this will lead to more and more alkaline nature of your blood. So that is why this is called as postprandial alkaline tide. Right, brother? Coming to the electrical rhythm of the GIT. This electrical rhythm of the GIT is produced by the special cells called interstitial cells of Cajal. And there is a resting membrane potential from -60 to -40 millivolt. This is produced by slow wave BR, basal electrical rhythm, and this is not the action potential. Remember, this is not action potential, right? So what happens? This actually -60 and going -40, again -60, -40. So it means this is not generating complete electricity. That is why we use the word basal electrical rhythm. But what happens? Whenever there is any stimulus, this negativity going more positivity like this, actual action potential of your GIT. So action potential is known as your spike wave MCQ. Spike wave. Peristalsis produces contraction. For example purpose, BR a different side. Maximum BR you should remember, that is in duodenum, 12 per minute, and minimum in cecum, that is 2 per minute. So just remember this and this. Coming to the next part, that is enteric nervous system. In enteric nervous system, we have talked about the two plexus. Number one, which is present into the mucosa, submucosa, called as submucosal plexus or Meissner plexus. So this is your submucosal plexus and Meissner plexus. Another plexus which is present inside of your muscle layer, and this is called as Meinrich plexus or Orbeck plexus. Now you already know what is the function of the submucosal layer: secretion. So Meissner plexus function is secretions, and Meinrich plexus is for your contraction. Now tell me what could be happen if there is no Orbeck plexus inside of your body, that area. It means that intestinal area will not contract, and if it will not contract, the food which is coming over here, it will stop over here, and now this area is completely a open pipeline like this. So no peristalsis is there, no forward coming of this food, and this disease is called as Hirschsprung disease. This disease is called as Hirschsprung disease. Now it is a defect. Now you can't do anything with this defect because it is already not forming this plexus. So it means now what is the treatment? Whatever the egg embryonic portion, you just cut it, remove it, and whatever is remain, just anastomose. So this is how this Meinrich or Orbeck plexus is there, right? But similarly, this is in ecclesia cardia. Similar thing happen. There is no Orbeck plexus. Now, about this Meissner plexus, this is helping for the secretions, all the digestions, all the pancreas, everything it is secreting with the help of this submucosal or Meissner plexus. Right, brother? So, brain of gut, it is a type of second brain, brother. It's independently works million of neurons, and all these movements are there. Peristalsis, distance, and GIT reflex GIT secretions, and more than 30 neurotransmitters are there. Like acetylcholine, VIP, nitric oxide, substance P. This is not sub, this is substance P, serotonin, dopamine, kappa. Now, this influenced by the ANS. ANS, we already know, it is divided into two parts, sympathetic and parasympathetic. And this actually, in the gut, this is supplied to the both plexus, both the part. Sympathetic, you know, it activates when? It activates when into the stress condition, fighting condition, and this is supplied by the celiac ganglion, superior mesenteric and posterior mesenteric. And whenever you are fighting with someone, beta, is there any craving to eat something? क्या जब किसी से लड़ाई कर रहे हो तो आप कुछ चाहते हो बेटा खाना? लड़ाई करते करते बोलते हो रुको, एक बार मेरे को गुलाब जामुन खाने दो. क्या ऐसा होता है? No. So that is why this is always decrease motility, decrease secretion, and this is inhibitory for GIT. And parasympathetic, vagus nerve till transverse colon, and this is post transverse colon, pelvic nerve. And parasympathetic beta always increase the desire for or craving for the food. So that is why when you are feeling relaxed watching the movie, then only you eat the popcorn. तभी तो अपन खाते हैं popcorn, samosa, सब कुछ movie देखते हुए. Why? Because we are feeling more and more relaxed, and it will increase the motility, increase the secretion, and this is excitatory for GIT. So beta, this is how you should understand this GIT. And there is ulcers. What is ulcers? This is discontinuation of epithelial lining called as ulcer. If this discontinuation happen into the oral epithelium, this is called as oral ulcer. In gastric area, gastric ulcer, and in duodenum area, called as peptic ulcer. So suppose that there is disruption of postproductive air, and this is a gastrinoma. Gastrinoma treatment is surgery. There is called as one bacteria which actually produces the toxin, and this toxin actually stimulates this parietal cell. And once this parietal cell will be stimulated, what would be happen? It will lead to more production of the HCl. So this is how the ulcers can be happen with the H. pylori, and treatment is antibiotic for the triple therapy. And said, then stop, then said, and use the antacid. Then there is ranitidine, cimetidine, that is H2 receptor inhibitor. And pantoprazole, rabiprazole, this is proton pump inhibitor, or another name. This is hydrogen potassium ATPase. Why we are using the word? Because exchange of this hydrogen and potassium is happening with the use of ATP. So this is also the proton pump inhibitor or hydrogen potassium ATPase inhibitor or PPI. So this is all about this GIT. I hope that you have understood this GIT very well. Now Coming to some exercises question, beta. Protein is digested by the proteolytic enzyme of the stomach, which cell produce pepsinogen in stomach. Beta, remember, pepsinogen, this is the main function, बड़ा काम है, और ये बड़ा काम कौन करता है? चीफ लोग करते हैं. So this pepsinogen is secreted by the chief cells. Hydroxytryptamine, this hydroxytryptamine is nothing but this is 5-HT. So this 5-HT is produced by which cell? Enterochromaffin cell. Enterochromaffin cell. CCK is secreted by which cell, beta? CCK is secreted by which cell? This is your eye cell. This is your eye cell. Then in digestive tract, maximum digestion occurs at duodenum. Which of the following substance absorption takes place maximum in duodenum? That is your iron and calcium. So in duodenum, maximum iron and calcium. In terminal ileum, that is vitamin B12 and bile salts. Rest all in jejunum. Rest all in jejunum. This is in jejunum and this is in terminal ileum. In duodenum, during digestion process, pancreatic enzyme chymotrypsin converted into active form of chymotrypsin with the help of beta. So chymotrypsin into chymotrypsin. This is with the help of beta trypsin. This is with the help of trypsin. Always remember. And this trypsinogen into trypsin. This is with the help of enterokinase. This is with the help of enterokinase. So this is how beta the question is there. Now one of the students asked me, sir, in the body fluids part, how this 10 liter came from the interstitial fluid. This was the question, beta. 30-year-old male having 70 kg identifies interstitial fluid. And answer of this 10 liter. Beta, remember, out of the 70 kg, 60% is water. So total body water is 42 liter. Out of this 42 liter, beta, 2/3 is ICF and 1/3 is ECF. So ICF is 28 liter and ECF is 14 liter. This 14 liter is ECF. This ECF is having two compartments. Number one, interstitial fluid, and number two is plasma. Now I already told you, interstitial fluid is 75% of total ECF or 3/4, and plasma is 25% of total ECF. Now you tell me, beta, how much out of this 14 liter? What is the 3/4 of 75%? That is around 10.5 liter, and this is around 3.5 liter, right? इस 14 का अगर आप 25% देखेंगे तो 3.5 लीटर हुआ, और अगर 75% देखेंगे तो 10.5 लीटर हुआ. And this is how this interstitial fluid, what is the question is asking? Question is asking this interstitial fluid, and this interstitial fluid is 10.5 of total interstitial fluid of total ECF, and this is how this question is formed. So now I hope that beta you have cleared this doubt. Now if anything more doubt if you are having, please let me know. And now beta always remember, एक चीज हमेशा ध्यान रखो बेटा, जब भी आप शरीर को पढ़ो, जब भी ह्यूमन बॉडी की स्टडी आप करते हो, तो बेटा ऑलवेज ट्राई टू मेक द कॉन्सेप्ट. अभी आप क्या करते हो बेटा? अभी आप 70% चीजों को रखते हो और 30% चीजों को समझते हो. आपको करना क्या है? 70% चीजों को समझना है एंड 30% चीजों को रखना है. Then only beta it's going to be very easy for you to pass this exam, because it is quite impossible for us. ये बेटा हम कर ही नहीं सकते हैं. ये आप कैसे सोच सकते हो कि आप 70% चीजों को रख के एग्जाम के अंदर जा सकते हो? बेटा this is quite impossible. You are also a human being. I am also a human being. I understand beta. I understand it very well. It's quite impossible. So try to make the concept. कॉन्सेप्ट बनाना कोई ज्यादा भारी काम नहीं है. अगर आप उस चीज को बनाने की कोशिश करोगे, इट्स गोना हेल्प यू अ लॉट बेटा. आप हर जगह उस चीज को अप्लाई करना शुरू कर दो. आज से आपको पता चल गया कि अपनी बॉडी के अंदर जो भी कुछ टारगेट एरिया है, जो भी कुछ अपन खा रहे हैं, जो भी कुछ सांस में ले रहे हैं, कहां पहुंचना है बेटा? सेल में पहुंचना है. किसके थ्रू से पहुंचेगा? ब्लड के थ्रू से पहुंचेगा. Entering into the cell, cell के अंदर भी कौन टारगेट एरिया है? That is nucleus. And now this cell membrane is having lot of channels, receptors over there. So now जो हमको पढ़ना होता है इन सब चीजों के अंदर, वो सारी चीजें बेटा यही पढ़नी होती है. चाहे आप कोई सा भी सब्जेक्ट पढ़ लो, चाहे आप मेडिसिन पढ़ लो, चाहे पैथो पढ़ लो, चाहे फार्मा पढ़ लो, चाहे सर्जरी पढ़ लो, चाहे पीडिया पढ़ लो, होना क्या है बेटा? सब में यही चीज होनी है, सेल ऑर्गन सेल ऑर्गन सेल ऑर्गन. सो नाउ ट्राई टू अंडरस्टैंड द बेसिक कॉन्सेप्ट. और अगर बेटा आपने ये चीज पकड़ ली, तो बेटा आप किसी भी क्लिनिकल सब्जेक्ट में मात नहीं खाओगे. दिस इज माय गारंटी. दिस इज माय गारंटी. सो रिमेंबर माय वर्ड्स बेटा. एज अ फैकल्टी आई एम गिविंग यू माय सजेशन. रिमेंबर माय वर्ड्स. अगर आप लोग बेसिक कॉन्सेप्ट अच्छे से बना लेंगे, इट्स गोना हेल्प यू फॉर द क्लिनिकल पार्ट. एंड बेटा इन द फर्स्ट पेपर इफ यू हैव स्कोर मोर देन 90, इफ यू हैव स्कोर मोर देन 90 इन सेकंड पेपर, बेटा दूसरा पेपर तो आपका है. दूसरे पेपर के अंदर तो आप वैसे ही नंबर नहीं छोड़ोगे. दूसरे पेपर के अंदर तो आप 70-80 नंबर वैसे ही लेके आ जाओगे और हो गया अपना काम. पहले पेपर में 90-80 नंबर और दूसरे पेपर में 60 नंबर, 140 नंबर अगर आपने बेटा एक्जैक्टली सही कर दिए, आपको पास होने से कोई रोक रहा है? कोई भी नहीं रोक रहा है आपको पास होने से. लेकिन थिंग इज दैट स्मार्ट स्टडी, स्मार्ट कैलकुलेशन एंड ऑलवेज बी ऑनेस्ट विद योरसेल्फ बेटा. ऐसा नहीं कि मैंने अभी आज पढ़ा दिया, आपने इस वर्कबुक को बंद करा, घर पे आराम से बैठ गए और कल सुबह जब क्लास शुरू होगी तो उसके बाद ही आप अगली बार इस वर्कबुक को खोलेंगे. नो बेटा, दिस इज नॉट लाइक दिस. सो ऑलवेज ट्राई टू रिवाइज ऑन डे टू डे बेसिस. एंड वंस यू हैव रिवाइज्ड इट बेटा, प्लीज मेक श्योर दैट आपने एटलीस्ट यू हैव सॉल्व अराउंड 20 और 15 एमसीक्यू जस्ट बिफोर गोइंग टू द बेड. सोने से पहले 15 से 20 एमसीक्यू आपको करने ही करने हैं. और अगर आपने ये चीज कर ली तो बेटा आपको ये पता चल जाएगा कि किसी भी टॉपिक से एग्जाम किस तरीके से एग्जाम के अंदर क्वेश्चन किस तरीके से फॉर्म होता है. बिकॉज दिस इज नॉट योर थ्योरेटिकल एग्जाम, दिस इज एमसीक्यू पैटर्न. एंड फॉर एमसीक्यू पैटर्न यू शुड अंडरस्टैंड हाउ द क्वेश्चन इज फॉर्मिंग. एंड फॉर अंडरस्टैंडिंग ऑफ दिस फॉर्मिंग ऑफ द क्वेश्चन बेटा, यू हैव टू मेक इट प्रैक्टिस. यू हैव टू डू लॉट ऑफ प्रैक्टिस टू सॉल्व दिस एमसीक्यू. क्योंकि एक शब्द से क्वेश्चन का आंसर बदल जाता है और उसका मतलब बदल जाता है. सो दैट्स माय सजेशन टू ऑल ऑफ यू गाइस. प्लीज ट्राई टू सॉल्व at least 10 to 15 एमसीक्यू ऑन एवरीडे बेसिस जस्ट बिफोर गोइंग टू द बेड. एंड दिस इज मेक द हैबिट टिल द एग्जाम डे. बेटा अगर आपने ऐसा कर लिया, मैं कहता हूं आपसे, आप ये सारी चीजें आप तब तक इतने एमसीक्यू सॉल्व कर चुके होंगे, इतने एमसीक्यू सॉल्व कर चुके होंगे, आपको बेटा इन एमसीक्यूज के अंदर जो सिली मिस्टेक्स होती है, जो एक हर एफएमजी के साथ में ये होता है कि कम से कम आप 5 से 10% सिली मिस्टेक करके आते हो, ये सिली मिस्टेक नहीं होगी बेटा. ये सिली मिस्टेक कभी भी नहीं होगी. सो दिस इज माय सजेशन टू ऑल ऑफ यू बेटा. एंड दिस इज ऑल फॉर टुडे. सो टुडे वी हैव कंप्लीटेड द सेल, वी हैव कंप्लीटेड बॉडी फ्लूइड, वी हैव कंप्लीटेड मसल फिजियोलॉजी एंड जीआई बेटा. सो प्लीज ट्राई टू रिवाइज द थिंग्स टुडे itself only, because tomorrow you are having another class and day after tomorrow again the physiology will start, and next day next class we will cover the excretory systems and respiratory systems. So it's going to be very difficult for you if you will not finish all this topic on the time, right beta? So I hope beta you have learned this physiology with a very good interest and you have enjoyed today's class. Might be you are studying beta those things first time, or might be you are studying the physiology first time. One more thing I would like to tell you guys. Every time student thinks from the physio only 5 or 6 questions comes. Yes beta, only 5 or 6 questions comes. But no, I will tell you. Every time from physio beta this is not true. Every time from the physio there is more than 15 questions comes. And why? Because you guys check only the recall session on the YouTube. Beta what will be the recall? What is the recall? रिकॉल वही तो होगा जो आप एग्जाम के अंदर क्वेश्चन देके आए. आपने मेरे को बताया कि सर फिजियो के ये क्वेश्चन आए थे. अगर बेटा आपने आज तक प्रोनेक्स रियो बेस पढ़ा ही नहीं, क्या उस चीज का रिकॉल होगा? कभी आप रिकॉल दे पाओगे? आप कभी भी उस चीज का रिकॉल नहीं दे पाओगे. एंड दैट इज व्हाई यू नेवर फाइंड these recalls on the YouTube session. So much maximum number question you never find on any single recall. But beta just watch my last recall of this FMG exam 20th Jan 2023. There are direct question around 15 or 16 question was there, and indirect FMG in FMG exam indirect physio question was 21. So remember these points. And every time this is happening, even the same thing, around 15 or 17 questions is going to come in your this June exam as well. So don't ever think like this, that this physio is quite for the less number of the subjects. No, beta. There is more than 15 questions will come in each and every exam. In the last PG exam, on this 5th March 2023, there were direct 13 questions was there. And can you believe it? Out of these 13 questions, around 12 questions, not 12, this is 11 questions. Direct 11 question was from your workbook. Can you believe on this? PG के अंदर आप जिस वर्कबुक से पढ़ रहे हो बेटा, उसमें डायरेक्ट 11 क्वेश्चन आए थे. तो ऐसा हर बार होता है. लेकिन अपने को क्या लगता है कि फिजियो ज्यादा इंपॉर्टेंट सब्जेक्ट नहीं है और अपन इसको स्किप कर देते हैं. बेटा, अगर आपने माइलिन शीट का नॉर्मल फंक्शन नहीं समझा, क्या आपको समझ में आएगा कि मल्टीपल स्क्लेरोसिस क्या है, क्या गुइलेन-बारे सिंड्रोम क्या है? अगर आपने नॉर्मल सा फंक्शन नहीं समझा कि माइंट्रिक प्लेक्सिस क्या है, मीसर प्लेक्सिस क्या है, तो बेटा क्या आपको हिस्टोमोलाइसिस एक्लिस या कार्डिया समझ में आएगा? कभी नहीं आएगा. So that is why always try to understand first of all the basic concept, the basic functions of the body. Then only you will realize, you will tell, no, this is not a normal condition, this is a disease condition. Okay, beta? So this is my suggestion to all of you, beta. And thank you very much for listening to me today. And my number is I already gave you to you, beta. This is my number, 750-360-7854. My name is Dr. Arun, beta. And if you find any query, any questions, beta, please write me on the WhatsApp on the same number. And this is all for today, beta. Thank you very much. Thank you very much, beta. And God bless you, beta. Till the next class. In next class, we will start with the new systemic part, that is XTD systems. So wish you a good luck, beta. Please keep revise things. Keep focus on the study. Please be honest with yourself. We always with you. Missed always with you, beta. So please study hard and just try to crack this exam in this June 2023. Thank you very much.